All right, great. So welcome everybody. So after 3 years, this is the first time we're having an in-person meeting, and we picked a great day. As I was coming down this morning, it seems it's a jacketless day. It's going to be 70 in Fahrenheit, and we have a great lineup of speakers. Thank you all for coming from all over the world. I mean, I think we have a speaker from Singapore, so that's the exactly the other side. So really great to have you here. So I want to spend a couple of minutes and tell you a bit about our center, a few slides. So we are a science and technology center funded by the National Science Foundation. And so the idea of these centers, so these are the biggest investment that NSF makes to support their vision of advancing discovery, innovation, and education beyond the frontiers of current knowledge. And typically it's at the interface of two disciplines. So in our case, it's the physical sciences and biology. So we were funded in 2016 and were renewed last year. Yeah, so we got nearly 5 more years on the center. And so in the first part of the center, we were the efforts were focused on understanding these 3 questions that I have here. How do cells sense? How do they adapt and remember? And these cut across mechanobiology of both plant and animal systems. So we study both plants and animals and the role of mechanical force. Now in the second part, we are focusing on mechano-intelligence, right? How many of you visitors have heard this term? If not, this is the first time you're hearing here, right? So what's mechano-intelligence? So mechano-intelligence is understanding and using mechanical force to engineer the working memory of cells, right? Working memory of cells for enhanced fitness and function. So it really involves sensing, adaptation, learning, and memory, which occurs in the nucleus. So in our second part, nucleus, which is the central control center of the cell, is a big a focus area. So we really want to understand how forces impact chromatin organization, gene expression, and how that feeds back. So again, I don't have any time to talk about research, but I'm just going to highlight two of our projects. One really looking at how chromatin is organized in 3-dimensional space. Say when you take a mesenchymal stem cell and plate it on different stiffness substrates, and really involves integration of imaging theory, data analytics, etc. And another project here, so here cells are feeling the environment, but in non-alcoholic fatty liver disease, which is very prevalent throughout the world, simply because we are eating more and exercising less, there are lipid droplets that accumulate in the cell, and this can lead to compression applied by these lipid droplets. That changes gene expression. It causes a malfunction of the hepatocytes and eventually leads to HCC. So these are just 2 examples. And what I really want to highlight here is that this involves 5 labs. This involves 4 labs. So really, our center is a very multidisciplinary and involves investigators from almost all our partner institutions. So we also have educational and outreach programs for everybody. So we have programs for kids through the Franklin Institute. So this is so this is the Philadelphia Science Festival, and we are also developing a museum exhibit in collaboration with Franklin Institute, which is one of the oldest science museums in the country. Also, our faculty are engaged. If you don't go to science fairs, we'll get you in the pub, right? So we have talks at science fairs. We are reaching high school students through our research experience for teachers program, as well as our undergraduates are involved in developing modules for high school students. So we have funded textbooks with strong mechanobiology content. Phil Nelson, I mean, as you know, is pretty much is part of our center and very much involved in this. And we have a network of educational partners, which includes HBCUs, MSIs, as well as 4-year colleges. and we are bringing mechanobiology modules to them. So currently we have a mechanobiology module, we have a workshop in at Puerto Rico, Mayaguez. Diversity at all levels is a priority of the center. We have programs to recruit undergraduates, master's students. So we have a fellowship where master's students at MSIs can come and work at one of our partner institutions, fully funded for 2 years. And we have a future leaders in mechanobiology program. So if you know a student or a postdoc or a early career faculty member with a great result, let us know. So we have so once a month we meet on Zoom, and a lot of these videos are pretty well downloaded quite a bit on YouTube. So it's on our YouTube channel. And our students are very much involved in organizing town halls, workshops. So that's an example. So with that, I'm really happy to see all of you here and look forward to a great symposium. So with that, we'll start the first session. Yeah, we are a couple of minutes late. That's fine.
Good morning, everyone. I have the pleasure of introducing our first speaker, Verena Ruprecht. She's speaking to us as a group leader from the Center for Genomic Regulation in Barcelona. Title of our talk is Morphodynamic Cell and Tissue Plasticity in the Early Embryo. We'll take about 5 minutes for questions afterwards, and I'll ask the next speaker come up during questions. Now the floor is yours, Verena. Thank you so much for being here.
Okay, thank you very much again for the invitation to this exciting symposium, and actually my apologies that I cannot be there in person. So I still hope that we will have some sorts of interaction, and please don't hesitate to reach out also afterwards via the chat or sending me also an email. So what we are interested in the lab is in fact how the organism forms from a single fertilized egg. And as you can see in the movie on the right, this is a highly dynamic process that really involves very dynamic behavior at the single cell level, where cells have to constantly divide, change their shape, they have to migrate. And all this needs to be integrated with cell fate specifications. So cells dynamically change their states and also morphodynamic behavior. And in the lab, we are really interested how the physical tissue environment and also mechanical forces shape cellular dynamics and how this integrates into mechanochemical signaling pathways. And the focus that I want to place on the talk today is actually that early development is something we see it here in a very stereotypic and robust manner, but it is actually prone to cellular errors at the single cell level. And this is something that is very important to consider also if we think of mammalian, especially human development, where we know even in the earliest stages of the blastocyst formation, single cells can undergo cellular errors that potentially lead to a reduction in cell fitness, lead to, and ultimately even lead to cell death. And we know that many of these errors also occur due to aberrant cellular divisions that can cause aneuploidies and result then in cell death, apoptosis. So the interesting aspect for us here was that it's actually very poorly understood how in this early stage embryo that has a very simplistic tissue architecture, how an embryo can cope with these aberrant cells or cells that are ultimately dying in the embryo. And we know that these problems at the single cell level are really an ultimate cause also for pregnancy failures in human development. Yeah. And so if we think about the adult body, how are apoptotic cells handled in our adult organism? We know that we have a class of professional immune cells and phagocytes like macrophages, neutrophils, or dendritic cells, and they are masters in clearing our tissues, and they are so efficient that we can hardly detect any apoptotic cells in our tissues because they are cleaned up so rapidly. But if we think of the embryo in these very early stages, and here we compare the mouse and the zebrafish embryo, as I said, they have a really simplistic tissue architecture. So you have the inner cell mass that will form the embryo proper, and the embryo is surrounded by an epithelium. There's actually a squamous epithelium of flattened epithelial cells. Yeah? So we set out the question, how are these cells with reduced fitness or that ultimately undergo cell death actually handled in this early embryo? And is there a mechanism that allows also in the physiological condition an embryo to detect, sense, and cope with these cellular errors, and how does the embryo do so? And in fact, we do see using the zebrafish now as a model system that has the advantage of being perfectly compatible with live cell imaging and high throughput methods. We do see spontaneous events of cell death in the in vivo tissue of the embryo. And this is one example where you can see the crowded environment of the embryo and the single cell undergoing spontaneous apoptosis that has these typical hallmarks of cellular fragmentation. You also see in the marker that is lighting up, that is an Exin 5, which specifically labels phosphatidylserine at the surface that is exposed from the inner to the outer leaflet when a cell undergoes cell death. Yeah? And doing backtracking of these cells and their origin, we also see that many of these cell death events in fact occur due to errors in mitosis in these early developmental stages. So to study this process and identify mechanisms, what happens to these dying cells in the embryo, we set out to establish a sort of embryonic stress test assay. And this was done by a fantastic postdoc in the team, Esteban Hoijman, who is now also professor at the University of Barcelona. And so we set up this stress test assay for studying how embryos cope with the barren cells in terms of being able to overexpress pro-apoptotic proteins, in this case PAX, at varying levels in the embryo. And then we can perform long-term live imaging. And when you look at the movies that we typically obtain in such experimental assays, bear with me, so I will guide you through this movie. On the right, you have the lower part, which is actually the embryo interior. And here in red, you can see these cells that will actually undergo cell death, that are initially highly abundant in the interior of the embryo. And in the upper panel, what you can see is the surface epithelium. And what you can recognize, hopefully over time, is that the embryo interior is cleared of these apoptotic cells, while a lot of this debris from these apoptotic cells piles up in the outer epithelial layer. And if we look into this process quantitatively, what we realized is really that these fragments of the dying cells, they specifically end up in the outer epithelium tissue layer. So the epithelium has a specific capacity to both recognize, but also take up these apoptotic targets from the embryo interior. While the cells in the inner cell mass, they cannot interact, uptake, or recognize those dying targets. Yeah? And that was a very curious observation for us, because it indicates that the epithelium has a very specific clearance function at the earliest stages of development in the developing organism. And we also were able to show that this is not only happening in the zebrafish embryo, but we can also work with systems like self-organized embryoid bodies or gastroloids that spontaneously epithelialize. And also in those systems, we see that this clearance takes place in the outer epithelial layer of the embryo. And we were also able to show that a similar mechanism is there in the mouse blastocyst, where we can see that when we transfer apoptotic embryonic stem cells or surrogate targets of apoptotic cells, which are mimicking the surface by exposure of these phosphatidylserine lipid, these are very efficiently cleared by the outer epithelial layer, which is the trophoblast, in this case, in the mouse embryo. So this led us to conclude that epithelial tissue, the epithelial tissue that is actually the first tissue formed in embryo development, has a very specific role in conferring an innate phagocytic immune function that allows to recognize and clean apoptotic cells from the interior of the embryo. And this is this was pretty interesting for us to see, because it is known that epithelial cells also serve as non-professional phagocytes in the adult body, especially in tissues that have low accessibility to phagocytes like the retina or the testes, but they also serve as additional phagocytes in tissues like the intestines or the breast tissues, where abundant cell death can occur, and where they have also been shown to play an important role in regulating inflammation. So epithelial tissues are it's a kind of have a conserved mechanism across species that they mediate apoptotic clearance. But very interestingly, they do also establish one of the first innate immune functions that we have in embryo development. And now turning to especially the zebrafish as a fantastic model system now to study the mechanism of this phagocytic clearance in more detail. This is where the zebrafish comes at hand, because we can perform a live imaging really at the single cell level to study this process in more detail. And this is an example of a single epithelial cell here on the right that is ingesting an apoptotic cell in red that is forming these dynamic protrusions, a phagocytic cup with a high actin enrichment. And you can appreciate this is dynamically wrapping around the target, generating also forces during this ingestion process until the target is ultimately ingested and phagocytosed. And this is very similar to the steps of the formation of these actin-rich protrusions is very similar to what has also been shown in professional phagocytes like macrophages or dendritic cells. So the morphodynamics of these cell-target interactions is very much conserved. And in addition, we were also able to identify the molecular players underlying this process, which I already mentioned. One of the key recognition factors to trigger that process is a phosphodiesterase ligand that is exposed at the target surface that triggers then the remodeling of the cytoskeleton in the phagocyte. And we can also interfere with this process by covering or shielding the PS lipid via annexin 5. So that we know this is really a specific ligand that is necessary, but also sufficient to trigger the response. And we were also able to identify the downstream signaling components that involve RUC1 and also PF3 kinase. And also this molecular pathway is very much conserved between non-professional phagocytes as epithelial cells and professional phagocytes. Yeah. So do epithelial cells then have the absolutely same capacity as professional cells to clean, and or is there any difference in the mechanisms they employ now, and that we can also study now in the physiological tissue context of the organism? So in fact, here's another example where we used mosaic labeling of epithelial cells, where we see they do form these dynamic protrusions that are the phagocytic cups. They are triggered upon target interaction, and they help to clean and ingest targets from the underlying tissue. But curiously, what also our in vivo imaging showed us is that these epithelial cells can form a very different type of actin protrusion when they interact with the targets. And this is an example shown here on the right, where instead of an epithelial cells getting in contact and taking up the target, it seems to rather interact with this contact and then manipulate it in a way that the target is actually moving away. And since this was for us reminiscent of grabbing an object, handling an object, we called these kind of protrusions dynamic epithelial arms, in contrast to these phagocytic cups that are formed during ingestion. Yeah? So we do see that epithelial cells, and these is protrusions that are frequently formed, so at similar frequency and throughout the clearance process, that there is a plasticity in terms of how far epithelial cells interact with their target, and how far protrusions and which type of protrusion is formed during these target interactions. Yeah? So clearance is really mediated or involves two different types of actin-rich protrusions that are formed. And it was surprising for us to see when we turned to live cell in vivo microscopy that these protrusions that originate from the epithelial cells, they are generally associated with all apoptotic targets in the tissue that are at the same time showing motility. And this was surprising because we did not expect that apoptotic cells in fact show a motile behavior in the in vivo context of the developing embryo here. And when we performed a correlation between the apoptotic targets and these protrusions that are formed, we clearly saw that these protrusions were always associated to the rear of these moving targets, so opposite to the movement direction. And this is kind of giving a hint that in fact these protrusions are strongly correlated and involved in the target movement itself, either by triggering apoptotic movement in the apoptotic cells, or directly leading to a mechanical force generation that would move apoptotic targets in the tissue. So there is polarized orientation of these epithelial arms at the rear of apoptotic targets. So, but which mechanism is now true? Does it trigger autonomous motility in the apoptotic cells, or is it that it's involving active force generation to translocate these apoptotic cells in the tissues? And this is a more global view of this apoptotic movement that we see in vivo. And you really see this involves large scale, long distance movements in the in vivo environment. And so when we interfere now specifically with pathways that are involved in cell motility in the apoptotic cells only, for example, by targeting Roche DBAs or interfering with contractility or the actomyosin cortex in cells per se, what we saw is that we did not affect the these motile patterns of these cells at all. Yeah. So we were not able to target cell migration of these apoptotic cells just by interfering with the motility machinery in these apoptotic cells. But when we interfered with RAC1, which is a very important Roche ATPase in regulating protrusion formation, actin polymerization, when we interfered with RAC1 activity at the tissue scale, then in fact we were able to see that these apoptotic cells lost their migration pattern, as is visible on the right. And in fact, they're having a very correlated, consistent movement, which is actually just indicating these overall gastrulation movements that they are following just the tissue movements during embryo development. Yeah? And we were also able to show that it was sufficient to specifically interfere with RAC1 activity only in epithelial cells. We were seeing a similar pattern, so that we were able to block the movement of apoptotic cells. So this in fact led us to conclude that we require RAC1 activity and this protrusion formation specifically in epithelial cells to trigger apoptotic motility. So, and this implies that epithelial cells are in fact able to trigger movement of apoptotic cells by generating active forces that are acting on these targets. And to unequivocally prove that, we turned to an assay where we used surrogate synthetic targets based on lipid coating of beads that are mimicking the surface of these dying cells, again exposing lipids, including phosphodiesterine. And also in this case, as you can see on the movie, we do see target movement, and we do see uptake of these particles, which really tells us, first of all, phosphodiesterine is sufficient to trigger the response in epithelial cells, but it, in fact, this movement involves active force generation, and epithelial cells are having the ability to interact with these targets in two ways: phagocytic uptake plus mechanical pushing that allows to translocate targets in the tissue. And of course, this leads to the immediate question of why and how is this plasticity and protrusion formation triggered? What triggers engulfment versus mechanical pushing? And we were able to dive into the regulators at the actin cytoskeletal level, specifically formins and ARCT3, to identify their relative contribution in these protrusion formations. And I will briefly guide you through the data that we obtained here. So if you look into this is now the surface epithelium, if we interfere with ARCT3 function, which is a major important actin polymerization factor that mediates actin branching, we no longer see any uptake at the epithelial level, although the embryo is full of dying cells at that moment. Yeah, and in comparison also with formins, we have a very similar phenotype, which shows that the phagocytic efficiency is massively compromised if we reduce ARCT3 or formin function, which means both are really relevant for the process of phagocytosis. So phagocytic cup formation requires the presence of both of these factors. But interestingly, when we look into the deep tissue level, where we saw this apoptotic motility going on, we saw that with interfering with ARCT3 massively stopped these dynamics at the apoptotic cell level, while formins, the inhibition of formins did not have any major consequences. And this was showing that this second type of protrusion that epithelial cells can form that is leading to the pushing is really dependent on actin branching, while formins seem to be dispensable for this type of protrusion formation. And as a final aspect, of course, it was very curious for us to ask then, why are these epithelial cells actually making two types of protrusions for the clearance? Why is it not sufficient just to ingest the target and have the tissue cleaned up in the most efficient way in that case? Yeah, and what you can see is that these protrusions, they do evoke a motility pattern, as I said, and this is really, we can describe this movement in the tissue of apoptotic cells as persistent random walk behavior that leads to a dispersal of apoptotic targets underneath the epithelial layer. So we turn to mathematical, very simple mathematical modeling to actually ask, so is these dual types of protrusions of mechanical pushing plus the phagocytic uptake now positively affecting clearance efficiency, or is it dispensable? Yeah? And so, to just guide you through the simple assumptions that we made in the model, assume that the epithelium is confronted with a local mass of apoptotic cells. Yeah, so we have very few parameters in this model: the uptake efficiency, the uptake probability, and the pushing probability, and a certain speed with which these targets can be translocated in the tissue. And yes, I'm wrapping up now. Yeah. And just to guide you through the results of this modeling that we performed, in fact, it turned out that it is beneficial for the epithelium itself to mechanically translocate targets. And this becomes evident if you think of another very critical parameter, which is the maximum loading capacity an epithelial cell has. So intuitively speaking, if an epithelial cell is confronted with a large mass of targets, if it would have an unlimited clearance capacity, you just ingest everything at once and the clearance is done. But under physiological conditions, epithelial cells do have a limited clearance capacity. And as soon as saturation is reached, it is beneficial to disperse targets to your neighbors. Yeah? And we were able to see that epithelial cells, being non-professional phagocytes, in fact have a very low clearance efficiency, and already they reach a saturation below a volume of an apoptotic cell. So this is also a mechanism that gets relevant already at low numbers of apoptotic targets to allow for efficient clearance. Yeah? So they saturate at low cardio volume, and for that, it is beneficial to have a mechanism that engages your neighbors in the uptake process, and they will stop cargo uptake at the higher cargo load. And this is a process, if you think of professional phagocytes, typically if you have a site of wound or infection, also professional immune cells operate in groups, which is usually mediated by chemical cooperation. So in our system, epithelial cells, in comparison to professional immune cells, they are not motile by themselves, so they are sessile in the tissue and they cannot move collectively towards a local source of apoptotic targets, for example. But what we propose is that they kind of inverted the process, and by a type of mechanical cooperation that involves active mechanical dispersal of targets, they can, in that case, still build up a cooperative tissue clearance by involving mechanical forces that act to disperse targets to multiple epithelial cells and then collectively are involved in the clearance process. And this is also illustrated here in this movie on the right, where you can see you have an initial target group of yellow targets that interact with a small pool of epithelial cells, but over time, due to dispersal, many more epithelial cells can participate in this clearance, and this overall will positively impact on the tissue clearance efficiency. So non-motile phagocytes can optimize tissue clearance by a mechanical cooperation that is mediated, in fact, at the single cell level by this plasticity of protrusion formation. And just as a brief outlook, so this is really the exciting aspect of epithelial tissues that have multiple functions that they have to integrate. We all know that they are important in terms of building tissue barriers, regulating sensation and absorption secretion, but epithelial cells, they are also highly dynamic at the single cell level, and they need to undergo shape changes, divisions in development, but also during tissue homeostasis in the adult organism. And phagocytosis is another important example. And the cells utilize the same cytoskeletal machinery to mediate all these different types of processes and maintaining the physiological functions. And I think it opens a lot of exciting future questions as well to see how epithelial cells integrate these different functions. With that, I would like to close. Thank you very much for your attention. I already highlighted people involved in this project. Hanna-Maria Häkkinen was also one of the key people in the lab contributing to the work that I showed, thanks to the funding and agencies and to collaborators and also the core facilities. And I'm happy to take a few questions now.
Thank you, Verena, for an amazing talk. So we'll take questions from the audience now.
Yeah, that was a great talk. Thanks. So I was wondering, the protrusions that you showed that were chasing and in the second type, the less common type, seems like it had significant volume. So is water pumping into the cell? I mean, it seems it was maybe 20% of the cell volume, right? So is that an important factor?
That is a very, very interesting question, I have to say. We haven't looked into the volume regulation yet, but I could imagine that it does play a role. We know also that epithelial cells, the basal surface of epithelial cells, it's still underexplored, right? And we know already in the beginning, they do have a lot of basal ruffles and there is excess membrane area. So I think getting a volume calculation or excess membrane or surface calculation is not as easy. So we would have to take very accurate measures to also get a good measure on surface and volume in those cells. But I could imagine that it also plays a role, yeah. Mm-hmm.
Other question?
Thanks so much for a lovely talk. And I was curious about the size of the apoptotic cells and whether that matters in terms of their relative motility versus propensity to be phagocytosed.
Yeah, that's also something that we're looking into right now, because in fact you do see that if you look into phagosome diameters that are so the uptake and targets in the epithelium, we know that they have a size that is much smaller than the size of an apoptotic cell, and this is due to fragmentation during the process. And we are now setting up a synthetic system that allows us to explore the size dependency, and we do see an optimum. So they have a maximum size they can handle and ingest. So target fragmentation is an important step of the whole process to allow for efficient clearance. Yeah, so without fragmentation, the clearance will not work. Yeah? So it is very important to consider, and it also involves active force generation for target fragmentation. That's also an interesting aspect that we are studying right now.
Great, thank you.
We'll take one more question.
Great talk, thank you, Claire Rose. And I was wondering if the cell die from something else than apoptosis, is there another program to take care of these dead cells?
That's also a good question. So we have only focused on apoptosis so far, I have to admit. Yeah, so stereotypical apoptotic features as phosphodiesterone exposure. We haven't explored other sorts of cell death yet, or whether also other clearance mechanism for pathogens would be acting in a similar manner. That's something we still have to explore. I cannot answer it yet.
Thank you for the talk. It reminds me of listeria, and I'm wondering if you see any organization of the expulsion machinery, symmetry breaking, that gets done to direct it.
Yeah, so these kind of protrusions that are pushing targets also reminding us of actin comets that are formed intracellularly, yeah? And we are looking into these, let's say, biophysical regulation as well, how far these different protrusions are formed and how far it also involves symmetry breaking. So we definitely know so phagocytic cups and these pushing protrusions, they originate from the same signals, but something is different in the initial trigger that and propagation, how these protrusions are formed, yeah? And we are still trying to wrap the data together and wrap our head around it, how this actually works, yeah.
Thank you again, Verena. Excellent talk. Thank you for the questions.
Thank you very much, yeah.
So our next speaker, Vinayak, is a graduate student in the Shenoy lab. He's going to be presenting on how does microenvironment affect the chromatin arrangement, a polymer perspective.
So, yeah, hello everyone, and thanks for attending my talk and being here. So, I'm a member at the Shenoy lab. I'm a PhD candidate. So today I'll be presenting how the microenvironment of the cell affects the 3D chromatin organization using a polymer perspective. So the outline for the talk would be that we have seen through experimental evidence mainly that the nucleus has characteristic chromatin domains, and these are dependent on the microenvironment of the cell. Inspired from this, we decided to build a polymer model which would show the maintenance of these domains as well as how these domains change in response to the microenvironment, or as we say, methylation level. And then finally, we try to look into the question of how the chromatin can have a memory of the microenvironment. This is an important question because for a big case, it's for example, in cancer, when the cell moves through different environments, we need to know how each environment is affecting it. And then in your day-to-day lab activities, if you culture cells on substrate and then you change them to some other substrate, then you also need to know how long the memory of the first substrate would last, or some treatment would last. So the first thing the first motivation is that we see these characteristic chromatin domains across cell lines, and these domains have a size of about 100 nanometers. They are also known as clutches, and you can also see a very distinct lamina associated domain, which is about 200 to 400 nanometers. So the central question we ask here is that what gives rise to these scales? Not only is the chromatin, we see these scales, but also the chromatin organization changes when the microenvironment changes. So for example, if say the substrate stiffness changes, then the actomyosin activity of the cell would change, which as shown by our lab previously and some other people and other labs as well, can lead to epigenetic shuttling. For example, I'll just stick to HDAC or EZH2 in this case. So HDAC or EZH2 would, so the in blue here is chromatin and red it's heterochromatin. This is euchromatin, this is heterochromatin. So HDAC or EZH2 help to convert euchromatin into heterochromatin. So on a stiff substrate, when HDAC or EZH2 move out of the nucleus, you would expect a resultant decompaction, which is what we see in the experimental setup as well. So you can see there are larger domains on the softer substrate and smaller domains on the stiffer substrate, which shows decompaction. So based on all of this, we ask how do the domain sizes scale? As you can see that the domain sizes are changing here with the microenvironment. And then what is happening at the genomic scale? Because it's a polymer model, we can try to answer those questions. So we consider the following dynamics in our model. So firstly, there is diffusion of density, which is basically that the polymer is moving in a background of nucleoplasm, or the chromatin is moving in a background of nucleoplasm through Brownian motion. Then we have diffusion of epigenetic marks, which is just that euchromatin again in blue and heterochromatin in red. So euchromatin likes euchromatin and heterochromatin likes heterochromatin. So the lower configuration is much more preferable than the top configuration. So if we have just these two things in the system, and we start with a random red and blue polymer. So again, red is hetero, blue is U. And we see a ripening effect wherein all the heterochromatin goes to the center and the euchromatin comes out. So definitely this is not enough to answer the questions we asked, the scaling or the emergence of these domains. So we add another mechanism to it, which we call epigenetic reactions, which is basically that any part of the heterochromatin or euchromatin can be converted to the other form using these probabilities of acetylation or methylation. So these are dependent on the microenvironment. So this is where the role of microenvironment comes. And if we add this to the simulation, then we see that now we start getting stable domains, as is seen here. And you can see through the simulation as well, these domains are stable in their radius. So the basic physics behind why these tables are why these domains are stable now is that the diffusion, which is the first two processes, the diffusion which comprises of the first two processes, would try to make the domains grow, which is what you saw in the last slide, while the reactions, specifically the acetylation part of the reaction, would try to make these heterochromatin domains decrease in size. So a balance between the two would give you these characteristic domains. So that was the first question we had asked, what gives rise to these domains? The next was, how do these domains scale? So for that, now we can change the acetylation levels in our model and see what's happening. So this is chromosome 20 in a cell line. So you start with an initial polymer, and as you increase the acetylation, the euchromatin regions grow, and you can see that they are mainly increasing to their boundary. Even in the 3D space, they are increasing at their boundaries, and the euchromatin is increasing at the boundaries of these domains. So most of the action here, we just want to show that most of the action here is happening at the domain boundaries, which will become relevant a little bit later in the talk. So now, for coming back to the question of scaling, we can do these simulations at different acetylation levels and get different domain sizes, and we get a very nice relation where the radii is directly proportional to the root over rate of methylation upon acetylation, a relation which we got analytically as well in a previous publication. So how does this relate to experiments? So as I already showed you in the motivation, that when you increase stiffness, the domain sizes go down. And just to match that, we find that the simulation shows a similar behavior as the experiments. Instead of doing such a difficult experiment, which is changing the substrate stiffness, we can just do TSA treatment as well, so TSA is an HDAC inhibitor. So in the case of soft to stiff on a stiff substrate, HDAC moves out of the nucleus, and which is the same as kind of inhibiting HDAC through TSA treatment. So we expect that the domain sizes would go down here as well, which is what we see in the experiments, and we then match it with our simulations. So that covers the first two questions we had asked. The first was, how do these scales occur and how do they scale with the changing microenvironment? The third was, what's happening on the scale of the chromatin itself. So for that, we refer to Hi-C experiments. So just to give a very brief background on some information I would need in the next slide. So this is a typical Hi-C map of chromosome 19. And what you can do is you can get a lower dimension representation of it using the first eigenvector. So without going into how it's obtained, the only main thing to remember here is that anything above 0 in this plot will give you A compartment, which is euchromatin, and anything below 0 will give you B compartment, which is heterochromatin. So now we take cells and then we do TSA treatment on them. So TSA again would inhibit HDAC, and then we see these changes. So I'll just explain these changes a bit more. So on the background, you see the first eigenvector, which I just explained, and then you see these lines, which I plotted over them. So the basic idea is that any blue line corresponds to a domain which changed from B compartment to A compartment, which is basically going from heterochromatin to euchromatin. And then any red line is going from A to B, or U to hetero. So and the other thing is that, okay, so that explains the plot basically. There are two things to note here, that firstly, there are more blue lines than the number of red lines, which is you are getting more euchromatin, which is what you expect through TSA treatment. And the next thing, which is very important and goes back to a point which I had mentioned, all these changes are occurring at the domain boundaries. So you can see that all these changes are where this gray line is touching 0, which is what we had shown through our model as well, that all these changes would occur at the domain boundary. So then we do simulations and we find that the changes which we see in the simulations align well with these changes, which we see in the experiment. So how does this tie with the memory question which I had asked initially? So now we see that all the changes which are occurring in the nucleus, in the chromatin configuration, are happening at the boundaries. So just keeping that in mind, now we can do a simulation process. We can do a simulation wherein we can take a cell from a soft to a stiff substrate and back to a soft, which would correspond to acetylation and methylation in case of our in case of our simulation. And the interesting thing we see, even though the 3D conformation itself is interesting, the more interesting thing is what is happening on the chromatin thread itself. Just to quantify the different types of behavior we see on this thread, we have conserved beads which don't change their epigenetic marks. Then we have beads which get a memory of the substrate, and then we have recovered beads which basically change from a soft epigenetic mark to a stiff and back. So these are just numbers to give you an idea of how the distribution is there. But mainly the mechanism is that we see are the main things we want to highlight. So you will see basically 2 different types of behavior. One is for larger domains and the other is for smaller domains. For the larger heterochromatin domains, you see that it's mainly the things at the boundaries which open up during acetylation and then close back again. For the smaller domains, which I have shown here in the blue box, the domains can actually go away or occur de novo. So these are the 2 types of things which can give rise to, say, any type of differences you see in epigenetic or genetic regulation. And the main idea behind we want to convey through all of this is that everything is happening at the boundaries for the larger domains, and smaller domains can change altogether. We also have some results for LADs, but I won't go into the details. So this is just that if you change the lamin interaction with the nuclear periphery, then the contact angle changes and so on. I can discuss this later during the poster session. So with that, I would like to summarize my talk, that we explain the presence and scaling of these clutches. We reconcile the imaging with the Hi-C observations, which is something which is new to the field and very new to the field. And then finally, we give a mechanism for mechanical memory formation. With that, I would like to thank all the members of my lab and our collaborators. And thank you for your attention, questions, and do catch me at the poster session if you have any extended questions.
Questions?
Very interesting work. So I don't know where I missed this. When you model this chromatin organization, did you consider the charge of DNA nucleic acid itself, or just mainly focus on histone?
Yeah, so yeah, that's a very good question. That's something which we should model, but the point is that at this point, our model is extremely coarse-grained. So each of the beads in our model, which we have, is about 90 nanometers or so, each of the single units of the polymer. So we are, for this model, we are assuming that the charges aren't that relevant at this scale, but they are definitely very relevant, and we are working on that as well in the theory as well as the simulation. So yeah, we don't consider it explicitly in this model because of the coarse graining effect, but yeah, that is something which we need to work on to get better, more accurate results. Yeah.
Nice talk. What do you think of as the energy source that's driving this process? Is it these chemical reactions, just simply thermal energy, or something else?
So yeah, again, a great question. So I'll just go quickly back to. So basically, the first two processes which I showed, the movement of the polymer as well as the diffusion of the epigenetic marks, both of them are energy independent, as in they conserve energy. The only thing that needs energy is the last process, which is the epigenetic shuttling. So the only thing that needs energy is the last one. So and the energy for that is mainly coming from, so when you have changes in the microenvironment, you can get actin polymerization, which we showed correlates well with the shuttling of these epigenetic factors in and out of the nucleus. Mainly coming from the changes in the microenvironment, as in how the cell reacts to it through actin polymerization and so on. The first two processes are energy conserving, so you don't need any. Does that answer?
Yeah. Thank you very much. Our next speaker here today is Kun Du. He's joining us from the National Institutes of Health, where he is a PostBaccalaureate IRTA fellow. The title of his talk is Nanoscale Viscoelastic Surface Characterization of T lymphocytes during formation of the immune synapse.
Can you all hear me? All right, cool. So thank you for the introduction. Hi, my name is Kundo. I am a second-year post-baccalaureate fellow from the NIH with the National Institute of Biomedical Imaging and Bioengineering. Currently work in the section on mechanobiology with Dr. Alexander Cartagena-Rivera. And today I'm going to be talking about our progress that we've made with characterizing viscoelasticity of T cells at the nanoscale length scale during formation of their immune synapses. So before I continue, I'd like to start by first outlining some great pioneering work that's been done looking at T cells and their overall mechanical properties. Around 2015, labs in France led by Claire Hibois and Atefas Nasios used single cell microplates to indent myeloid and lymphocytes in inflammatory conditions and resting conditions to calculate their global elastic modulus, as well as the viscous loss modulus. And a couple years later, another study led again by Hibois and Julien Husson, this time used a different technique using micropipette force probes to sort of suspend T cells and indent them while they're in contact with these activating antibody-coated beads. And they were the first to sort of see and characterize that these soft cells, which are around 100 pascals, so quite soft, became a lot more rigid while being activated. And so it became pretty clear that not only looking at the mechanics that occur at the immunological synapse is important, but also looking at the dorsal side of these T cells and the cell's overall mechanical properties would be beneficial to understanding how these drive T cell activation and understanding generally, how pathogenesis works, and as well as the T cells response and development, which is quite important to us and to us as our lab. We're quite devout AFM practitioners. This became a really interesting and fascinating question for us. So if you're not familiar with atomic force microscopy or AFM, basically it functions using a small cantilever as a sort of force transducer. And there's also a laser that follows this cantilever. And when it deflects, or when the cantilever deflects by coming into contact with a soft sample, this gets picked up by a photodetector, which is able to track this change and does it at very sensitive scales. And our lab in particular specializes in leveraging AFM and the different modalities that it has, combined with also coming up with theoretical frameworks to determine the material properties of very soft biological samples, often at very high spatial temporal resolution. And sort of our repertoire includes things like cells, of course, viruses, as well as whole tissues, which is quite exciting. And one of these modalities is using something called tipless AFM. So using an AFM technique developed by my PI in 2016, we used a very soft, flat tipless cantilever to very lightly deform the spherical non-adherent T cells, as you can see on the figure in the bottom left there. And from that, we can calculate the surface tension from the cell cortex from the corresponding AFM curves that you get. And using this framework, we were able to measure populations of both pre-activated and naive CD4 and CD8 T cells, and found that this cortical tension is actually much higher in T cells that had already been activated than these naive cells in these non-stimulatory conditions. And seeing that there was this increase in cortex tension between unstimulated, naive, and activated cells at different sort of stages in differentiation gave us a lot more motivation to look at the mechanical properties while these T cells are actually being stimulated firsthand. And so for this, we approached this problem using a different methodology that would not only take into account the cell's dynamic shape and its heterogeneity while it's being activated, but also taking into account the cell's viscoelasticity. And previously, this is shown to be the case with many cells, including leukocytes. So with that in mind, a postdoc in our lab, or former postdoc in our lab, Cameron Parvini, we implemented a newer methodology for extracting the viscoelastic information that takes individual force curves in each pixel of a map that we take with AFM. So it's the very bottom left image there as like a point-by-point acquisition. We then translate the time series observables, basically what the AFM sees. So it's things like the force as well as the indentation as a function of time. We translate that into the complex Z domain and analyze them directly using transfer functions such as the relaxants or its inverse, the retardants, which gives us a very nice derivation of the elastic as well as the viscous portion of the viscoelastic behavior given by the storage modulus, which is analogous to the elastic modulus of the cell, as well as the loss modulus, which is an indicator of the viscosity. And so the nice thing about this model is that unlike traditional viscoelastic models like generalized Maxwell or Kelvin-Voigt models, we get to avoid relying on iterative parametrization and optimization to calculate these viscoelastic quantities, which can take quite a long time. And in the case of AFM maps that we take, which can be tens of thousands, if not hundreds of thousands of force curves in a single map, this becomes almost computationally too heavy for anything out there. So to sort of test this implementation, we decided to try it first on Jurkat cells fixed with 4% PFA. So we could take AFM scans with very high pixel density and collect as many force curves as we can at a singular time point without having to worry about the cell being too dynamic that we can't capture the entire cell throughout the AFM's capture time. So we fix these Jurkats after about 5 or 6 minutes of spreading on antibody-coated glass with anti-LFA1 control and another surface with anti-LFA1 and CD3. And actually saw that cells on the surface coated with anti-CD3 and saw this activation, saw an overall increase in the viscoelastic behavior and showed very unique heterogeneity in viscoelasticity, especially at the sort of distal and central region of the cell at high frequencies versus the sort of peripheral region in between, which seemed to be less stiff and less fluid. So once we were able to sort of flesh out our model, we were then able to implement this actually using live Jurkat cells, where we observe very similar heterogeneity in the cell's mechanics. And unlike fixed cells, these cells are actually very dynamic. So in order to measure these cells in a way where you can at least capture reasonably the early activation of these T cells, we used a smaller map size and a smaller pixel density, therefore less force curves taken through our AFM to measure the entire cell within about 5 minutes or a little less. And again, interestingly, we see the similar behavior like we did in fixed cells, telling us that this viscoelastic behavior seems to be preserved both across fixed and live cells, which may indicate something about the underlying actin-myosin structure being changed in activation. So just to summarize at least the results that we came up with, we developed 2 unique AFM methodologies. One using tipless AFM, where we measured naive T cells and activated T cells, and showed that the activated T cells were actually stiffer in cortex tension than their naive counterparts. We also developed a sort of model-free viscoelastic method that's compatible with our high-resolution AFM to measure the viscoelasticity at multiple timescales at the nanometer length scale for both fixed and living T cells. And we also observed when we applied this methodology is that we observed this viscoelastic heterogeneity at higher frequencies for these T cells while they're being activated. And so just to, I guess, summarize that, we did see a higher stiffness as well as viscosity at two regions in particular, the central as well as the distal region of the cell, as well as the peripheral transition region being more fluid and soft, which is quite interesting. And it brings a lot of questions to mind that we'd like to pursue further. So of course, we would love to apply this to more relevant T cell models within a more physiologically relevant setting. So we actually have in our bucket list, so to speak, using these primary human lymphocytes. So if effector CD8, CD4 cells, Tregs, and we want to try applying that not only just on antibiotic coated glass, but hopefully to apply that to a more mechanosensing environment using polyacrylamide gels or some soft medium of varying stiffness. But we've actually taken some steps towards that using actually cytotoxic lymphocytes. So yeah, so we have CD8 cells being spread on anti-LFA1, anti-CD3, as well as the co-stimulatory anti-CD8. And yeah, I think we see some interesting behavior with of course, seeing the similar trend in viscoelasticity heterogeneity in the CD8 cells. So hopefully we want to apply that to a more broad scale. We've also started to look at ways to run sort of unsupervised learning, specifically clustering algorithms like k-means or k-medoids, what have you, to individual maps to see if we can better define mechanically, variable regions of interest and characterize their viscoelastic profile. And shown around the bottom right is just an example of an implementation of k-medoids clustering on one of our fixed cells using the storage modulus and loss modulus. And you can see that the algorithm is able to quite nicely organize spatially where it sees different viscous and elastic properties, which I think down the road would be good for profiling viscoelasticity in a more unbiased manner. And so further down the line, and we've actually made some progress on this front as well, as to run a global clustering sort of regime to see if we can extrapolate these trends to not only single cells at singular frequencies, but also to entire T cell populations or subsets. And of course, adding different elements that are noticeably absent from the study, like looking at structural components to potentially turn this into not only single label, but multi-label classification problem would, I think, be quite interesting as well. And with that, I'd like to thank the lab back home at NIH, and as well as a few key collaborators of ours, Alex Jommer, Erdem Tabunov, and Arpita Budaya, who have helped answer a lot of the many questions I had on immunology, and sort of were good guides to us into our first foray into immune cell mechanobiology. Also like to thank the NIH BioWolf cluster for running all of the analyses that we did, as well as our funding from the NIBIB and the Intramural Research Program. With that, thank you.
Number of questions. So someone that hasn't asked one yet, looks like we have. I'll try to get this up to you quick. And if the next speaker could come down as well.
Thank you. That was a really wonderful talk. Have you guys considered looking at if there's any sex dependency on the forces generated with the T cells? Like how hormones could potentially affect the viscoelasticity as well as the stiffness of T cells?
We haven't looked at specifically hormones, but I think down the line, I think that'd be an interesting route to take. Granted, I'm not quite versed with hormones and immunology as well, but I think that'd be an interesting route to take, where we can look at certainly things like cytokines might affect the mechanical behavior. I think that's the first thing that comes to mind, and I think that should be a very interesting route to take. Yeah. Thank you for your question.
Thank you.
Oh, Kwasi, yeah. One last question, then next speaker.
Yeah, thank you. That's a really good talk. I was just curious to know, have you tried using peptide MXC for T-cell interaction? So these activations, especially for the primary T cells, were done with anti-CD3, anti-CD28, right, coding. Now, what happens if you do more antigen-specific stimulation where you are engaging the TCR? Do you expect, I guess, similar stiffness and viscoelasticity properties?
Yeah, I just want to make sure I'm answering questions. So you're asking whether if just looking at just TCR engagement versus just TCR engagement co-stimulation would cause an effect in viscoelastic behavior, or?
Because here you are bypassing the TCR, right? You are using anti-CD3, anti-CD28 for pan T cell activation.
Yeah.
What happens when you target the TCR? You use peptide MXCs instead of CD3, CD28.
Yeah, so those are great suggestions, and yeah, that's something we'd like to look closely into, not just anti-CD3, because there's obviously more to the TCR than just coating glass with anti-CD3. But I think that'd be very interesting to look at particularly with primary cells, and we're going to make more progress with that, and hopefully involve a better model of activation and corroborate that with our viscoelastic results. Thank you for your question.
Thank you very much. So our last speaker of this session is Dr. Nader. He's assistant professor, recently joined Penn as a pathology and laboratory medicine at Children's Hospital of Philadelphia. The title of his talk is Compromised Nuclear Envelope Integrity Leads to Tumor Cell Invasion.
Okay, good morning, everyone. It's a pleasure to be here. Thank you for organizing for the opportunity to present my work here. As I said, I just joined CHOP 6 months ago, so trying to get things started in the lab. And I will start with this picture. So I did my postdoc in Paris, and things can get pretty nasty there some days. And as you can see here, there's a lot of mental stress, but I am mostly interested in the physical stress. Okay, so just like us in a lovely morning, the cells also have to deal with physical stress all the time when they're navigating within interstitial spaces or growing within the tissues. Okay, so this is a mouse ear explant. And what you see here in gray is the collagen network, in green neutrophils, and they're gonna inflict a damage to the tissue with laser, and then you're gonna see a very synchronous migration of the neutrophils towards the damaged site right here. Okay, and I like to show this time lapse because this illustrates that cells have to cope with the complexity of the microenvironment in order to achieve their functions, achieve their goals. Okay, so they have to cope, in this case, the very complex meshwork of collagen, and this involves cell body deformation, but also involves nuclear deformation. Okay, and I am mostly interested in looking at and studying nuclear deformations and how the nuclear deformations affect cell fate and cell behavior. Okay, the lab where I did my postdoctoral research, and also now my lab, we have been using different microfluidic and microfabricated devices in order to recapitulate the complexity that cells encounter in vivo. Okay, so here, for example, we have the microchannels, and we use the microchannels to mimic, for example, the channels that cells use to invade in vivo in the muscle fiber. So, for example, here you have in yellow the cell body, in blue we have the nucleus, and I hope you can appreciate here that not only the cell body but also the nucleus undergo high deformations in order to be able to, in this case, metastasize in the tissue. Okay, and here we have this is an in vitro experiment. If I can get my. So this is just the dendritic cell that was embedded in 3D collagen matrix, and it does not play. But what I wanted to show here is that the nucleus gets pinched and gets highly deformed as the cell explores this 3D collagen matrix. Okay, and here we have a cartoon showing the interstitial migration of dendritic cells, which is shown here in a mouse ear explant, and the process when the dendritic cells find the antigen and have to present the antigen to the T cells in the lymph nodes, they have to migrate into the lymphatic vessel, and this involves also extreme nuclear deformations. Okay, and this is actually shown here. So this is a mouse ear explant, and you can see here that the cell, when it travasates into the lymphatic vessel in red here, you see that this involves also high nuclear deformations. Okay, and again, we're very interested in studying nuclear deformations and the consequence of nuclear deformation. So here you can see a cell in the microchannel, and in green is the nuclear envelope, and in red is the chromatin. And you can see, here we're not pushing the cells, okay? So the cells are migrating by their own will. And we can impose more or less stress to the cell by modulating the constriction width here through which they are migrating. And you can see here that as the cell migrates, the DNA extrudes out of the nuclear envelope, meaning there's a nuclear envelope bleb and then rupture. This exposes the DNA to the cytosol, and this generates DNA damage. Okay, I'm not going to get into the details of the nuclear envelope receding mechanism, but there's a nuclear envelope receding mechanism here that allows cells to continue with this constricted migration. Okay, so the cells do not die, but our lab showed that, Matteo Pia's lab showed that this repair mechanism is core3 dependent. Okay, so I'm going to focus on the consequences of this loss of nucleocytoplasmic compartmentalization, which is, I just told you, DNA damage. So in order to study this in a more physiological context, we were trying to study the implications of these deformations in a more physiological context. And then we thought of a scenario where the cells would self-impose strong deformations. And we thought in the tissue overgrowth of solid tumors, and we adopted the breast cancer progression, okay? Because in this model, the cells grow under intraductal confinement, okay? So as the mammary duct inflates, because the cells grow there, they are self-imposing deformation, exactly as I showed you in the first picture of people in the train station. So the first question that we are trying to address here is, okay, so how do the nuclei look like in a human primary breast cancer? Okay, so this is a patient, and you can see here at the tumor bulk, the nuclei look pretty much round. But if we now go and analyze the microinvasive focus, we see that the nuclei are highly flattened, and they are also positive for the DNA damage marker gamma H2AX. Okay, and you can see here better in the inset, they're highly stretched and highly positive for the gamma H2AX, and the nuclear roundness is also quantified here. Okay, so at this microinvasive foci, nuclei are highly stretched and also highly positive for DNA damage marker. Okay, so based on this observation, we made this hypothesis. We asked the question, does increased physical confinement in these dense tissues have a link to cell invasiveness? Okay, so in order to test this question, we have to try to reproduce that now in vitro, right? So how can we impose different mechanical stress to the cells in vitro? And in order to do that, we established a posthumous collaboration with Fred Mercury to put the cells under pressure. And in order to do that, we use what we call the static confiner. Okay, so this is an ordinary 6-well plate, and to the 6-well plate we attach deformable pistons, and to these pistons we attach cover slips. So these cover slips have a layer of microfabricated micropillars. Okay, so these micropillars will be their spacers determining the confinement height to which the cells are going to be subjected. Okay, And here you can see the cartoon of cell confined and the micropillar here. Okay, so we can modulate the confinement height to which we're gonna apply the cells to. So this is how it looks like an actual confinement. So you have the micropillar here. So the cells that are under the micropillar are completely smashed, dead. We're not studying them, but we care about these cells here. And then you can see a lot of plasma membrane blebs because it's like we're pressing on the cells, so they become pancakes, and then the plasma membrane detaches from the actin cortex. So that's why you see these protrusions here, these plasma membrane blebs. And most interesting to me, that's how the nucleus looks like. Okay, so if confined like HeLa cells, this is the HeLa cells confined to 3 microns, you see breaches on the nuclear envelope, and then you see also the DNA extruding out. Okay, so now we'd like to actually determine the DNA damage response to different confinement heights. So for that we use in order to probe the nuclear envelope rupture events, we use a cytosolic DNA sensor here in red called CGAS. Okay, so CGAS has a cytosolic distribution when it doesn't see DNA, but when it sees DNA, it becomes very, it goes to the sites of the rupture in that case. Okay, so the cells are confined at 2 microns, so it means we see rupture. Okay, CGAS is very perinuclear now, and the green foci here means DNA damage. Okay, so as you can see here, cell confined at 4 microns does not undergo nuclear envelope rupture and does not experience DNA damage. On the other hand, cells that experience nuclear envelope rupture undergo DNA damage. Okay, so this graph is actually showing that the DNA damage here is not due to mechanical damage, mechanical stress that we apply on the cells, is really due to nuclear envelope rupture. So here we have movies that show exactly what I'm telling you. And so this is a nuclear envelope bleb, and then you see the bleb is going to burst. And then only after it bursts, we start seeing the accumulation or the appearance of DNA damage foci. Okay, and it's important to mention here that for as long as the cells remain under confinement, the DNA damage levels remain high. And why is that? It's because the nuclear blebbing and bursting events just keep happening on and on and on. Okay, so they appear, they burst, and then nuclear envelope is resealed, and then they appear again and they burst again. Okay, so this is a very dynamic process. So now that we know that 2 microns is a confinement height sufficient to induce nuclear envelope rupture and chronic DNA damage, we're gonna take these cancer cells, the ductal carcinoma in situ cells, we're gonna squeeze them, confine them. Following the confinement, we're gonna remove the confinement, harvest the cells, and embed them into the collagen matrix. Okay, so now we can study their behavior in 3D collagen matrices. Okay, so again, the cells are not confined here. They were confined previously, harvested, and then embedded into the collagen matrix. So now this cell here was confined at 10 micrometers, which is a control confinement height. Okay, so it doesn't cause rupture of the envelope and doesn't lead to DNA damage. And then you can see the cell stays pretty much put on the collagen network. It doesn't explore a lot of the collagen. You see small protrusions. Now, if you confine these ductal carcinoma in situ cells to 2 micrometers, which is a height that induces both nuclear envelope rupture and DNA damage, the behavior become very, very different. Okay, so now it's highly protrusive, explores a lot the collagen network. Okay, so it seems to be a confinement-induced change of behavior here, and we can actually ask the question: does this behavior that we observe here in this collagen, does this translate into increase the collagen degradation? And the answer is yes. Okay, so we have here an antibody that recognizes the epitope of collagen that gets exposed upon collagen degradation. And you can see here that cells that are confined at 2 micrometers exhibited increased collagen degradation as compared to the control cells. Okay, so that's the path we have so far. We have cell confinement leading to nuclear envelope rupture. this leads to DNA damage, and we observe that it seems that this DNA damage response is leading to increased matrix degradation. Okay, but what is the factor or factors responsible for this DNA damage following these nuclear envelope rupture events? So to answer this question, there was this paper that was published by the group of Titia Delanger in New York City, Rockefeller Center, Rockefeller University, and they showed that the Trex1 exonuclease enters the nucleus during to resolve these chromatin bridges that persisted as a result of telomere fusion. Okay, so Trex1 nuclease entered the nucleus, nicked the DNA, and allowed bridge resolution. And at the same time, we observed what the DNA that gets exposed upon nuclear envelope rupture now becomes accessible to cytosolic factors. Okay, so we hypothesize that Trex1 might be one of the factors responsible for DNA damage following nuclear envelope rupture. And so we did a bunch of perturbations and knockdowns and overexpression, and also Trex1 knockouts, and we could show that upon nuclear envelope rupture, Trex1 depleted cells do not display increased DNA damage. Okay, so DNA damage upon nuclear envelope rupture is Trex1 dependent. So what we have so far is that confinement leading to nuclear envelope rupture, the nuclear envelope rupture needs to Trex1 dependent DNA damage, and this might be leading to matrix degradation. So now that we know the factor that is responsible for this DNA damage, we can ask the question: does DNA damage following rupture play a role in increased matrix degradation? So we knock down Trex1, and we confine the Trex1 depleted cells, and then embedded them into the collagen matrix, and we could prevent increased collagen degradation by depleting Trex1 in the cells. Okay, and this is quantified here, and also we do not see increased collagen degradation in two different Trex1 CRISPR knockout clones. Okay, so the path so far is confinement leading to nuclear envelope rupture, leading to DNA damage via Trex1. And I didn't have time to show you today, but this leads to ATM and SNA1 dependent invasive phenotype. Okay. Now, if you do the same things, exactly the same thing to healthy cells, untransformed cells, this leads to cell senescence. Okay. Again, we didn't have time to show you this. So we decided to try to get closer to the in vivo environment. So we wanted to develop the device that would mimic the intraductal confinement that cells are when the cells are growing in vivo. Okay, so we have here the myoepithelial layer, and then we have the tumor bulk where the cells are growing, and then we have microinvasive like breaches on the myoepithelial layer, and then this is already a fully compromised and invasive stage. Okay, so we wanted to recapitulate that by using a microfabricated device. So we developed what we call the mammary duct on a chip. So we have the cells, we have the channels here where the cells are injected. So the channels is actually mimicking the tumor bulk. Okay, and then we have the gates, 10 micrometer wide gates, which would be mimicking the breaches on the myoepithelial layer. And then we have the collagen chamber, which can be seen here, that is mimicking the interstitial space. Okay, the cells will be invading towards this interstitial space. So with this device, we can mimic the intraductal confinement that cells are experiencing this stress when growing. And then you can see here that the cells invade towards the stroma, and then it can get super crowded here. So we can look at nuclear envelope deformation, rupture, and DNA damage in this micro device. Okay, and what you can see here that after long times, 48 hours growing inside this mammary duct, we see more DNA damage. You can see this quantified here. We see increased nuclear envelope rupture, and we see a decrease in nuclear roundness. Okay, so this self-confining duct on a chip is inducing nuclear envelope rupture and DNA damage in these cells. okay? And what happens now if you deplete Trex1 in this context, in the context of mammary duct on a chip? Now you can see here that at 24 hours of growth, since there's not enough cells to inflict stress to each other, we don't see increased DNA damage. But then in the control cells, we see increased DNA damage at 48 hours. But in Trex1 depleted cells, this DNA damage is decreased, okay? And this is shown here in the insets. You see the cells do not exhibit 53 BP1 positive foci, and as opposed to the control cells, okay? And the nuclear envelope rupture levels are the same, are comparable between knockdowns, and control cells. So we can also, like I said, we can also access cell invasion in this micro device, okay? And we mix here two cell populations, the control cells in green and the Trex1 depleted cells not labeled. And we can see here that the control cells invaded in greater number, but also farther into the stroma, into the collagen chamber, okay? And this is quantified here. So DNA damage in cell invasion in a ductile on a chip is Trex1 dependent. So we went further. We decided to now generate mammary interductal xenograft model. So we inject either Trex1 proficient cells or Trex1 deficient cells in the mammary gland of the mice, and then we analyze both in situ and invasive stages of tumor progression, okay? And as you can see here, we observed a lot of DNA damage events, especially against the myoepithelial layer of the parental tumors. But in the Trex1 knockout tumors, we observed we did not observe DNA damage positive nuclei. Okay, so these are two mitotic figures, so are not considered, not counted as a DNA damage positive nuclei. So we could actually decrease in vivo DNA damage by depleting Trex1 in these tumor xenografts. Okay, and this is also quantified here. What about tumor progression, right? So we observed that 100% of the tumors generated with the Trex1 parental cells progress to invasive stages, but now only 30% were invasive in the Trex1 knockout xenografts. Okay, and this is shown here. Most of them remained in situ. Okay, so breast cancer xenografts of Trex1 knockout cells do not progress to invasive stages. Okay, so the final model that we have here is that we have cells undergoing nuclear envelope rupture, and this leads to Trex1 dependent DNA damage, and this leads to, again, I didn't have time to show it today, but this leads to ATM and also SNE1 dependent invasive phenotypes, leading to matrix degradation and tumor invasion. On the other hand, if we do the same thing to non-transformed cells, when these non-transformed cells undergo nuclear envelope rupture and DNA damage, they become senescent. Okay, and this was a work that involved at least 4 different labs. So this was a joint task force. I really have to thank a lot to different people: Imageanalyse, Mathieu Morin, my postdoctoral advisor Mathieu Piel, Philippe Chavrier, Catherine Village, Jean-Louis Viovi, because they also helped us developing the memory duct on a chip, and also Giorgio Citan in Italy in the lab of Nicolas Manel. And thank you for your attention. I'll be happy to take any questions.
Thank you for the excellent talk. And the first question, it's the first hand.
Very lovely talk. Do you know, so Trex1 seems to be a very promising clinical target. Do you know any study that
So we did, yeah, we did we analyzed it, we consulted the TCGA, and in breast cancer patients, the ones that have higher expression levels of Trex1 have poor prognosis. Yeah, that's as far as we went, but obviously now we need to analyze other other types of cancers, and also try to correlate that with DNA damage and nuclear envelope rupture and the expression of Trex1. Yeah. Can you put back my presentation, please? I just want like to see the opportunity that we also. So yeah, we were able to see actually these DNA damage events in human primary breast cancer. So you can see here that the C-gas staining also exactly as we had observed in vitro. You see at the tip of the nucleus. And we also analyze another type of cancer, which is the colorectal cancer. And you see here the microinvasive margin that the cells are more brown, meaning they're more positive for C-gas. But at this level of resolution, you cannot tell really about the localization of C-gas. So is it really nuclear envelope rupture? So we can zoom in at this invasive margin, and to our surprise, we saw exactly the same pattern as we had observed in vitro of C-gas at the tip of the nucleus. So we were able to observe nuclear envelope rupture events in vivo in both colorectal cancer and also in breast cancer. Okay.
Thank you.
Yes.
Yes, thanks. That was a great talk. I have two quick questions. The first is, do you see sting activation and also interferon response related to the C-gas? And then the second question is, do you see DNA repair at all? And what is the kinetics after nuclear envelope rupture?
So yeah, we do not see sting activation. Okay, so the nuclear envelope rupture and the exposure of this DNA is not, this DNA we think is highly enriched for chromatin, and then this prevents the activation of the C-gas sting pathway. Okay, and the other question is, so the 53BP1 that I showed is actually DNA repair protein. So yes, there is constant DNA damage and DNA repair. And the half-life of this foci is around, I would say, 40 minutes. Okay.
Hi, that was a great talk. Earlier you mentioned that it's the DNA damaged cells that are able to degrade the collagen matrix, be able to invade out. Have you looked like at prolonged periods of time to where there's enough degradation to where the non-DNA damaged cells would be able to then have a path to migrate out to further enhance like the invasiveness?
Yeah, so if I got your question correctly, so what we think here, so these are, so for example, this is also mice, this is a tumor xenograft of the mice, and then we see usually enriched areas where we see the C-gas staining, and then of cells undergoing this DNA damage. But we think that it suffices only 1, 2, or a few cells to undergo this stress and this DNA damage, and then to be able to breach the myoepithelial layer and then to invade the stroma. The other cells that are behind, they not necessarily underwent this stress. You see, so now once the gate is open, all the cells are ready to invade into the stroma. Yeah.
Question. Fascinating stuff. So Trex1, of course, if you knock it down, it can lead to pathology with extra cDNA. Do you have any sense of the dynamics over the time ranges that you need to be knocking this down in a patient, or if there are other signals that say precede the need for Trex1 in a cell that could be a trigger for local delivery?
Local delivery?
So the question is on the dynamics. So do you have any sense of what sort of time scale you need to be applying Trex1 for therapeutic effect, or if there are ways to turn it off and on?
Yeah, yeah, because if like you mentioned, so Trex1 dominant negative mutations leads to chronic inflammatory response and autoimmune diseases. So what we are characterizing here is really a nuclear signaling module. So is the role of Trex1 towards nuclear DNA as opposed to its role on the cytosolic DNA. And now how to, if you knock down Trex1, and then would prevent DNA damage, but then at the same time we're gonna like promote this chronic inflammatory response. So I don't have an answer to that.
Thank you.
Let's give another round of applause for Dr. Nader and all the speakers this morning. We'll be coming back at 11 a.m. So now you have a short break and you can engage with the speakers after for more questions too. Thank you.
Hi, everyone. Welcome back. Our second session, we're starting it off with Dr. Andrew Holle, who is an assistant professor at National University of Singapore at the Mechanobiology Institute there. And he will be talking to us about the epigenetic state during and after confined migration.
All right, thank you very much. So I'm super happy to be here. I just want to thank the organizers of this meeting for having someone come all the way from Singapore. I think it represents the fact that mechanobiology is a truly international community. I was discussing with Song Lee just now that we were very lucky that we had amazing talks leading up to this, right? So we've had a lot of very nice introductions from Guilherme and from Vinayak that help explain a lot of things I'm going to talk about today. So hopefully this all connects and it makes some sense. So as she just mentioned, I'm from the National University of Singapore and from the Mechanobiology Institute at NUS. And I run the confinement mechanobiology lab. So we just heard some nice confinement stuff. But today what I want to talk about is maybe how epigenetics might change in confinement, because that's the theme of this meeting. This is a new area of exploration for us. So we're happy to take any input you might actually have. So this is kind of an overview of some of the things my students are working on. We just started the group about 2 years ago. And the stories I'm going to talk about today are from Yixuan, who's very interested in epigenetics, and also Gao Xu, who's interested in stem cell differentiation. So like I said, we saw some of these videos already, thanks to Guilherme, right? So none of this is totally new. We know that cells in vivo are under compressive confinement. And depending on the type of environment these cells are actually in, we see that there's different levels of this confinement. And we can use nuclear shape almost as a readout for the level of compression or confinement these cells are in. These are individual cancer cells migrating out. You see the ones migrating in a single stream are under much more confinement. Now, I'm a biomedical engineer by training, and what we as biomedical engineers do is try to take something complex in vivo and make it much simpler, right? So we can study the important parameters that are involved in this. We also got a very nice introduction to some microchannels already. Again, thanks, Guilherme. This is the type of system that I've used throughout my postdoc and that we're carrying into my new group, which is a PDMS-based, photolithography-based microchannel system where we can watch individual cells enter a confinement, move to the opposite side, and then exit. So very quickly, I'll just summarize some of the things that I learned in my postdoc on cancer cell migration and how individual cells can undergo this mesenchymal to amoeboid transition. We can see that cancer cells moving into wider channels and cancer cells moving into very narrow channels seem to be behaving much differently. Just like as Guilherme showed, when you have not so much compressive strain versus a lot of tight sandwich style compression, cells react differently. And we can see that inside very narrow 3-micron channels, there's this adaptation phase where the cells actually decide to enter, realize that they can't move through the way they typically would, reorganize their cell body, and then move through or permeate very quickly. And of course, we can quantify all this because this is very compatible with microscopy. We don't have to do the fancy intravital microscopy that Peter Friedl is so good at. And we can learn a little bit about how this mesenchymal to amoeboid transition occurs when it's self-directed by the cell, right? When the forces aren't being opposed directly on the cells, but when the cells can decide their force landscape when they move through the confinement. So again, just to summarize the system that we use, right? This is kind of a setup that we see quite a bit where you have it before, like an entry reservoir, cells move into a confinement, and then we can look at them actually afterwards. And this is what it looks like if you hold them in your hand. We're so happy to send and share these chips all around the world. So if you'd like to play around with these, we can ship them to you. We do have ceilings on these microchannels, so it is a 3D confinement as they actually enter. And we also don't, even though it's kind of a microfluidic technique, we don't actually use pumps or anything for the most part, because we keep the fluid column the same before and after, so there's no flow. So the cells that are migrating are doing so in the absence of flow. And just in terms of like the parameters that we use, most of what I'm going to show you today is between 25 microns or 150 microns in channel length. We don't use any chemoattractant. We don't have a pressure gradient. We coat pretty much everything collagen 1. But of course, we can change this. So I wanted to highlight some of the tools, like as an engineer, that we can use to understand nuclear confinement, because this is an epigenetics type meeting. And we saw very nice videos of these pinch point constrictions from Guilherme earlier. So what I want to do is kind of contrast a pinch point constriction with a long sustained microchannel confinement. So both of these are excellent tools for studying how cells behave in vivo, but perhaps they sort of reflect different environments, different microenvironments, and where this confinement is being imposed. So a pinch point might represent more in vivo intravasation, where cells are breaking into a very, very tight space, but they can put a lot of their cell body through to the other side before they rip the nucleus through. And to contrast, literally we saw this exact same picture, so it's very helpful. We know that inside the body, there's long confinements or constrictions like this, where the cells can't actually build big, strong focal adhesions on the opposite side and rip the nucleus through. They have to adapt more. So what we know from a lot of work from Matthew Piel's group, from Jan Lamberting's group, is that these very short passages often induce high levels of DNA damage. A lot of times we study this in terms of chemoattractants, whereas with the long microchannel confinements, we see the DNA damage itself is a little bit less characterized. And for the most part, what we do is look at cells that explore without stimuli. So if I show you these videos, and I was very, I have a lot of videos in this talk. 2 of them didn't survive the translation from Mac to Windows, right? So you're just going to take my word for it on what the pinch point video actually looks like. But again, Guilherme showed nice videos of this as well. These cells that enter these pinch points put big, strong protrusions through, build focal adhesions, and then rip the nucleus through very rapidly. Whereas you see something kind of different with the cell that's entering this type of confinement. There's a reorganization and a pause followed by this long migration step. And we can actually quantify this just by looking at the amount of time spent at the channel entrance, right? So if we have a very abrupt 3 micron confinement, then it doesn't spend much time at this entrance. If the 3 micron confinement is very long, then we get nearly double the amount of time on average for a nucleus to spend at this leading edge. And we think maybe this has important implications in how the nucleus responds to these confinements. If we look at DNA damage, right? So we saw a lot of nice videos of 53BP1. In this case, we're looking at gamma H2AX. Here we see that the cells moving into this short 3 micron confinement. This is their gamma H2AX measuring DNA damage. These are in RPE1 healthy cells, essentially. So there's less of this background you get from cancer cells. And you compare this with cells that are inside long 3 micron confinements, we actually see fewer of these gamma H2AX breaks from the cells that are undergoing this much longer confinement, suggesting that reorganization may be protecting them in some sense. And we can actually take these and look at them along the length of the channel and say, do we have any correlation between how deep into the channel the nucleus is and the amount of gamma H2AX puncta that show up? And we see a very slight negative correlation that the deeper into the channel it gets, we see fewer on average of these gamma H2AX puncta. So a nice feature of these microchannels, something that I really like, is that we can study the cells in multiple different microenvironments in a single chip. We can look at cells that have never entered a confinement. We can look at cells when they're inside the confinement itself, and then we can look at them afterwards to see if they retain any sort of memory of this journey, right? If we look at very simple things, like the projected area or the aspect ratio of nuclei, we see that cells that move through these very tight 3 micron constrictions, they don't change their the nuclear area after they go through, but the aspect ratio does change. This is a much more sticky change in nuclear morphology. This is 2 days after we plate the cells. If we look at volume, which is a little bit more complex to image, right? We can do this either with confocal microscopy. We're also starting to use fluorescence exclusion microscopy to be able to get more high resolution movies at a much higher time scale of volume changes. We see that the nuclei that move through these abrupt confinements, if you look at them on the entrance or the exit, we don't see any big differences in the nuclear volume. Same is true for wider but longer 10 micron channels. But in the longer 3 micron channels, we get a reduction in volume inside the confinement. And this reduction in volume is sticky, right? 2 days later, if you take the measurement of all the volumes of the cells outside, it remains lower. So if we look at how cells dynamically rearrange and respond to this confinement, especially when you compare wide and narrow, we start to see some patterns as well. This is kind of an illustrative, in a sense, chymograph showing, showing how the nucleus is actually positioned inside the body of the cell as it moves through these very tight spaces. And we can then quantify the position of the nucleus as it pertains to the cell itself. How far back within the cell body is it? We find that cells moving through the long, narrow 3 micron channels, the nucleus is much closer to the rear on average, again, suggesting that there's an adaptation. If we just look at all of the cells within the nucleus and plot where the nucleus is across all of our channels, because we can look at hundreds at once, we do find an enrichment of nuclei at the entrance of the channels. Again, confirming that there's a process happening, a dynamic rearrangement of the nucleus that causes it to stall before it actually moves through. And this brings me to the epigenetics of it, right? The focus of this meeting, something I'm not a huge expert on, something I'm so happy to hear constructive criticism and advice moving forward on. So what Ishwan has found, the PhD student working on this, is that chromatin really is dynamically altered during this process of entrance into a long sustained confinement. If we look at this H3K9 AC, which is a marker for acetylation, we see in the cell that this is not the same cell. It's 3 different cells fixed at different points. We start to see a reorganization of where these acetylation marks are actually occurring. And finally, we end up with this semi-polarized signal once the cell's fully inside. So if we know that chromatin's condensing under confinement, we can actually compare how it's occurring in wide 10-micron channels and narrow 3-micron channels. And we find that, and this is well known, Jan Lammerding has showed this as well, that when cells enter a confinement, the chromatin becomes condensed, we get less of these acetylation marks, and this is especially prevalent inside 3-micron channels. If we then look at how this chromatin is organized once the cells are fully inside the channels, not overall levels of the acetylation marks, we find something I think is quite interesting, is that the acetylation becomes polarized at the leading edge and trailing edge of the nucleus. This isn't always balanced, right? But we do, on average, find that there's much less of the hooks at the poles of the cell and much more of the acetylation marks. So these actually are anti-correlated in the very narrow 3-micron channels. So in regions where you have less of the DNA, you've got more of these acetylated regions, more euchromatin. If we look at some of the things that might be playing a role in this changing of epigenetics, if we look at histone deacetylases, things that should remove these acetylation marks, we find that cells entering the confinements, you get more of a translocation from the cytoplasm to the nucleus inside both 10 and 3-micron channels. But then what happens if we actually try to interfere with some of this? So we had a nice talk earlier from Vivek's group that showed that we can use this TSA to actually interfere with this process. So if we look at regular cells and their acetylation marks as they enter the channels, this is all scaled the same way. So you can see how little acetylated regions there are in 3 microns. If we rescale, we see this polar distribution like we saw before. But if we add the TSA, so we get much more acetylation, the cells inside 10 microns have much more of the acetylation than they would before, but in 3 microns, they lose it. So just chemically altering this acetylation pathway doesn't actually change the cells losing so much of their acetylation inside the confinements. Although it is important to note that when you have these hyperacetylated cells, the nuclei then are much bigger, much fewer of them are likely to enter these very narrow confinements. Because we know from Dennis Disher and many other groups that at these regions of high curvature, we would find at the leading edge of 3-micron channels, you get some depletion of different types of lamins. We looked for depletion of lamin B. And this is quite new data. We haven't looked at multiple different antibodies, but in our hands, at least, we don't see any major depletion of the lamin B signal at the leading and trailing edge of the nuclei in 3-micron channels, where you have the most amount of curvature. So the final thing that Ishwan's kind of done, this is a big journey for us to explore, like what's actually happening to lead to these changes in epigenetics, is look at histone acetyltransferase. So in this case, PCAF, which corresponds to HDAC3, we find a strong polarization of this signal as well, that then corresponds to the polarization that we find in the acetylation marks. So earlier I mentioned that we see strong polarization. It's not always on both sides. Sometimes it's just at the entrance or just at the exit. But in this case, the expression of the PCAF always corresponds with the side that we find the cell with more or less of the acetylated marks, right? So there's a correlation between these two things. So what's actually driving this? Is it possible that maybe nuclear envelope permeability, if you read some of Pararuka Kuzak's papers about nuclei under high compressive stress at high curvature regions, allowing for more or less import of different transcription factors, could this be driving this particular response, that we segment more of our PCF at the front? This is then allowed to migrate into these high curvature zones across the nuclear envelope, and that's then affecting our amount of acetylation that we see in these polar regions. But this is ongoing, and we're learning a lot about this. But the last thing I want to talk about today, how am I doing on time? 10 more minutes. Okay, cool. We can take a little bit easy. So I had done most of my postdoc work on cancer cells, and everything I showed you for the most part now was with cancer cells or clean model RPE1 cells. But what about something where nuclear compression might be a little bit more functional for the overall behavior of the cell? So we started to move into adipose-derived and mesenchymal-derived stem cells. And the first thing that you'll notice when you look at these cells compared to a breast cancer cell is that they're much, much bigger. So these are scaled the same way. You can see how huge the adipose-derived stem cell is compared to the breast cancer cell. And the first thing I was a little bit concerned about is, would we even be able to get cells to migrate through these very tight spaces? And luckily find that these big adult stem cells do indeed migrate through 10 micron channels and the very narrow 3 micron channels. So how are they affected? How's the nuclear morphology affected, since we're talking about the nucleus so much today? Well, when you put these stem cells through the very short 25 micron long confinements, these pinch point confinements, we don't see any change in stem cell nuclear area or aspect ratio. But if we start looking at the much longer microchannel confinements, we can compare cells inside the channel that we see here and outside the channel afterwards. We see that changes in nuclear area and changes in nuclear aspect ratio that cells encounter are actually sticky, right? So a stem cell migrating through a 3 micron channel afterwards, so 2 days afterwards, it still has a lower projected cell nuclear area, and it has a higher aspect ratio. And if we then compare the volume, we find that the volume afterwards is the same as it was before. So if the cell's entering, it's undergoing some nuclear changes, and afterwards the volume's the same, but the projected area is lower. Just simple geometry says that that means that the height of the nucleus is actually higher. So these nuclei, when they enter a 10 micron tall channel, they have to become deformed in 3 dimensions, and this deformation actually sticks. So the mechanism for this, we're not totally sure about yet, but I think it's interesting to explore. For us, we're interested in the output, the consequence of this on stem cells. So if we look at the most simple maybe model for adult stem cell differentiation, this is osteogenic differentiation, differentiation down a bone lineage. And here we're looking at RUNX2 or CBFA1, which is one of the earlier transcription factors that gets turned on in osteogenic differentiation. If we send cells through these shorter pinch point confinements, we don't really see any of the signal. If you have a very, very narrow 3 micron channel, you start to see some of this signal pick up. But through the longer 150 micron long confinements, 10 microns is not sufficient to really induce any change in this signaling. But 5 microns and 3 microns, we really start to see a big spike in CBFA1 expression. And if we quantify this, it's quite significant. And actually, when we compare this to cells that are chemically differentiated, the magnitude of the signal is approximately the same, right? So we're getting a very robust response just from this migratory journey that the stem cell might take. We can also look at other transcription factors involved in osteogenic differentiation at different times. For this one, Gao-Shu looked at Osterix. This is 14 days after the migration through the channels. So the cells are robust, even grown for 2 weeks inside this system. And we see, again, a big enrichment of Osterix in the nucleus, in the cell, and in translocation. But what I think is really interesting, right, and I actually really love these pictures, because this kind of summarizes a lot of the work that we do in one single image, right? We see cells that have never undergone this confining journey. We see maybe sometimes cells that are still undergoing the journey itself, although the journey only takes 3 hours. And finally, cells that have gone all the way through. And if we stain for the right thing, we can see actually the effect that this journey had. If we look at those same acetylation marks that we were looking at in the nucleus for the cancer cells, when they're in confinement, they have a big reduction in acetylation. But these stem cells, 7 days after they've made this journey, their level of acetylation signal is much, much higher than the cells that actually never went through it. Okay, so there's more genes being exposed for transcription. Presumably there's more activity going on, driving it down this differentiation pathway. And this expression of H3K9AC is also confinement and length dependent, where we see the biggest and most robust response in the very long 150 micron long channels, and especially at the very narrow 3 micron widths. So overall, the questions, at least as they pertain to the nucleus, right? You saw there's a wide diversity of things we're interested in terms of confinement mechanobiology. But for the nucleus, we're very interested in what's causing these things to happen. We know the nucleus is heavily deformed inside a confining microenvironment. I love this nucleus because it looks like a micropipette aspiration, where you're imposing a ton of force on a nucleus. In reality, this cell chose to do this, right? In its adaptation to this extreme confinement. And when it does this, I mean, work from Shivashankar has shown that these different regions of the nucleus, when it becomes more compressed as cells become terminally differentiated, you get different regions that become in contact with each other. And how this might change signaling is also a big open question. And comparing that to this more active compression where the cell puts these big, strong focal adhesions through and then rips the nucleus through very quickly, how much of a role is this playing? I think a lot of the work that Vivek has done and is doing about telling us how chromatin is organized in these spaces and how we can consider it from a polymer perspective could help answer a lot of these questions as well. But the thing we're doing right now as well, although I don't have time to talk about today, is if we sever the link complex, where supposedly these strong forces are being exerted on the nuclear envelope, how will this change that resultant differentiation behavior? So overall, we want to know, does nuclear adaptation to these different spatiotemporal scales of confinement drive epigenetics, the purpose of this meeting, phase separation, and differentiation? And I mentioned phase separation just because I wanted to quickly highlight this paper that we just put out through some collaborators, which I think illustrates that confinement is a really excellent tool for people with very wide and varying biological questions. With this particular collaborator who had actually discovered a phase-separated condensate called perispeckles in the nuclei, when we put these cells into microchannels, we find an enrichment of these perispeckles, but only at very specific widths, only very specific confining widths. And this is another one of the videos that was a victim of the transfer, but I can tell you exactly what happens. In this particular nucleus, we see no perispeckles. It's a live cell reported for perispeckles, but as it enters, we can see during the course of its confinement here, you get an increase in perispeckle expression before it ultimately exits and eventually reaches an equilibrium post-confinement. So this phase-separated change is not sticky. When you look at the phase-separated condensates afterwards, it returns back to the equilibrium. Okay, so again, I just really wanted to advertise this as well. I'm sure Guilherme also has very nice tools that you can play around with. But confinement, I think, can be looked at in many, many different biological contexts. So talk to your local friendly confinement mechanobiologist for more information. Yeah. With that, I'd be happy to take any questions. I do want to thank my funding sources, the National Research Foundation, and highlight that in both my lab, the Confinement Mechanobiology Lab, and many of the labs at the Mechanobiology Institute, we are actively searching for postdocs as well as new independent group leader candidates. Thanks.
We have some time for a few questions.
Hi, thank you. That was a beautiful talk. I was just wondering towards the end, because we were talking about the link complex as well. I was wondering if you had looked at all, or are you thinking of looking at curvature sensing bar proteins at the protrusion tips? Because there's a lot of beautiful work showing that they can play a big role in linking protrusive activity to the cytoskeletal elements, to the link complex. They really exist at this interface of protrusive and cytoskeletal activity. So I think it would be something interesting to study if you're not already looking into that.
Thank you so much. No, we haven't considered looking at this yet. We're taking more of a more brunt approach, I guess you could say, just by using this DN cache Nesprin that Jan Lamberting and Brian Burke have developed that actually forces out endogenous Nesprins into the cytoplasm. So theoretically, you can't have this forced transmission across the nucleus. And then if we see this differentiation response afterwards, we know it's just because of the compression of the nucleus on its own. If we don't see it, then we know that it's because force at the link complex actually exposed maybe different regions for transcription, and that's what initiates the program. The nice thing is, like, from my perspective, I don't care what we get. Either answer is interesting.
Thank you.
Hi, it was a nice talk. I kind of have a technical question. So, like, how did you come up with 10 micron and 3 micron dimensions? And I was just wondering, like, if for other dimensions, would you expect other responses from the migrating cell?
Yeah. So really just for shorthand in this case, because if I show every single channel width that we've worked with, which ranges from 20 microns at the absolute widest down to 3 microns is kind of the lowest, but 3 and 10 seem to provide the nicest contrast between a cell behavior that's kind of unconfined, but still interacting with the walls of the channel. Because if it's 30 microns wide and the cell's just on 2D, then you're losing this kind of more 3D stimulus. So that's why we kind of choose this. In terms of the shapes that we do use, this is a whole other project one of my students is working on, is can we look at a much more high throughput approach to many different types of confining stimuli, whether it's just widths, changes in periodicity, changes in curvature in particular spots, and how do we look at thousands of cells going through many of these different things without having to kind of build a new chip for every single new design. Yeah, so that's definitely an engineering challenge that we're taking on.
So, thank you very much for your nice talk. So usually like the channel that you confine the channel is way steeper than the other tissue, right? I mean, so people try to understand the other channel like a gaps or like a size of this, how the gaps like a regulated epigenetic event or something. What do you think about the other, if you change the stiffness of the wall or like a channel, and then how can you expect that the other?
Yes, yeah. I think that actually was one of my major goals during my postdoc, and I completely failed, right? I wasted like a year of trying to build polyacrylamide systems in these particular gels. And actually Kostas Konstantopoulos has kind of beaten us to this. He's just published a paper last year with soft-walled microchannels. And essentially at these very narrow confinements, and even the 2 micron confinements that Matthew Pales explored, you need this push outwards. You need this chimneying response. And it seems like in these very soft microchannels, if you do this, the cells can exert pressure outwards, but not sustain it enough to build the friction to move forward. So I think the papers I've seen that have soft 5 micron channels, the cells don't move so well.
Yeah, you show the decrease in acetylated chromatin, right? When you put in the 2 micron channels. Yep. But you also show that it gets accumulated towards the tip or the region of high curvature.
Yep.
Why is it there?
Yeah, I would love to know. So like this, we found this because we're just curious about how we had the stem cell results that we get major changes in acetylation after the cells move through. We knew that Jan had showed there's a big change in methylation. So we started looking at acetylation inside the nuclei. And this is when we found this polarization type signal, where sometimes we get perfect 50-50 front and back. Sometimes we get strong front, sometimes we get strong back, but we almost always see this increase in acetylation at the edges. So this sparked the question, right? Like, what's causing this? Is it just curvature? Is it some change in the import kinetics? Because theoretically, the center region of the nucleus is not exposed to cytoplasm, so things can't really get in and out so well. But if something is moving in, then maybe only these regions can either remain or become more acetylated in response to that curvature. But we don't know. That's why I told this sort of incomplete story, so that we could have a scientific discussion about it and hopefully.
The acetyltransferase you never saw inside the nucleus.
The what?
The acetyltransferase that you did the co-staining, but yeah. You saw the vicinity, but you never saw inside the nucleus. Right?
It's tough to say whether it's just maybe crossed a little bit, right? Mostly we see it accumulate in the cytoplasm, but whether it's not deep into the nucleus, we could say.
Okay, let's thank our speaker. There'll be plenty of time over lunch to ask more questions. Next up is Professor Song Lee. He's the Chancellor's Professor and Department Chair of Bioengineering at UCLA. And he'll be talking to us about mechano-epigenetic engineering for cell reprogramming.
Great. Good morning, everyone. I want to thank Vivek and CEMB to organize such a wonderful symposium. I have learned a lot this morning from every talk. And today I want to talk about some of our recent work on mechano-epigenetic engineering. The research in our group is focused on cell and tissue engineering. On the one hand, we are interested in understanding how microenvironmental factors, and especially biophysical factors, can regulate cell functions together with biochemical signals. And on the other hand, we're trying to apply what we learned in vitro and combine that with biomaterials engineering, drug delivery, etc., and use that for in vivo tissue regeneration or disease therapy, such as cancer therapy. So today I want to focus on some of our in vitro work in the mechanobiology area. And you have seen many examples that mechanical forces everywhere in the body at different levels. Our focus is at a cellular level, and we want to understand how mechanical factors can regulate cell functions. And on the other hand, we want to use these knowledges to engineer cells in vitro. And you are going to see how we do that in one of the examples I talk about. In a microenvironment, there are so many physical factors, right? So many years ago, I worked on the effect of fluid shear stress in the cardiovascular system, how that can regulate cell signaling from a receptor level to intracellular cytoplasm signaling transduction, and finally regulate gene expression. In the past 20 years, we have focused on the extracellular matrix in all the substrate in cell culture, how these physical factors such as the stiffness, viscoelasticity, the micro-nano pattern, or the ligand distributions, or even the dynamic mechanical loading, all of this together could regulate cell functions. So in our recent work, I just want to use one slide to show the sensing part. And this was motivated by the needs to enhance the long-term immunity to have more effective vaccine for COVID. And this could also have an application for cancer vaccine as well. So in this case, we use microfluid channel to make alginate microbeads with different sizes. And you can use a different cross-linking mechanism to manipulate the stiffness and viscoelasticity of these beads. And then we manipulate the cell surface chemical presentations and also the release of the cytokines. And the goal is to in vitro demonstrate that we could increase the production of T memory stem cells, which is very important for long-term immunity. And we also simplify this system and demonstrate that in vivo, indeed, you can enhance T memory stem cell formation together with other vaccine components. So the other area we have focused on in the past 10 years or so is mechanical regulation of epigenetic changes. So we know that mechanical forces can regulate signal transduction and gene expression, but in many cases, all of these changes are transient. It could go up and down and then go back to the basal level. And how the mechanical factors can regulate permanent changes of cell memory in the long run is very important because it's related to stem cell differentiation and cell reprogramming and all other phenotype changes in vivo. You have seen the example. I don't need to go into the detail. I just want to point out that in the body, we have heterochromatin, euchromatin, and all of these are regulated by various things. And we don't know the exact hierarchical structure, how this is organized in vivo. But we do know a lot of random commands here. For example, the regulation by all of these enzymes. So we're going to show some of these changes in our mechano-stimulated samples. I will show you in a few slides. So the model we use is cell differentiation and cell reprogramming. And the reason is that during cell reprogramming or differentiation, these changes could happen within days. For mouse cells, less than a week, you are going to see geometric reorganization of the chromatin. In the humans, may take a little bit longer. So here I want to focus on a couple models. One is the conversion of fibroblasts into pluripotent cells by using Yamanaka factors. And the other one is the direct conversion of fibroblasts into neuron. So the hypothesis is that during cell differentiation or cell reprogramming, and here, as you see, this Wellington landscape, and every point here represents a transient state. We know that the chemical signals can regulate this process, and after the cells differentiate, they'll settle at some stable state. And the hypothesis is that if you have mechanical cues appropriate for the cells, around cells or inside the cells, and this could regulate the epigenetic state and finally help the cells to be converted into one or the other, all the way back to the pluripotent state or into a different distinct cell types. So how can this happen? So one hypothesis is that through these modification enzymes, so for the methyltransferase or acetyltransferase, etc., or demethylase and deacetylase. And the other one is that through the mechanical coupling. We know when the cells attach, and it could be connected through integrins to extracellular matrix or cell-cell adhesions. And all of these are coupled to actin filaments or other cytoskeleton, and they can be transmitted. The signal can be transmitted through these actin filaments into nucleus. In connection with lamin or lamin AC or lamin B, there are a lot of chromatin directly affiliated. So in in the nucleus, people have found, for example, heterochromatin are mostly concentrated at the nuclear periphery. And you have seen in the previous talk from Vivek's lab. And also, most of these heterochromatin are turned off in certain cell types and organized in certain way. So the hypothesis is if you deform the cells or deform the nucleus in certain way, you can perturb this nuclear organization of the chromatin and cause either a movement or opening or closing for a certain part of the chromatin. And this part is unknown because it could be a random process. This could be a stochastic. It could be some definite result as well. So 10 years ago, we had one observation is that when you culture cells at different shape, in this case, you have to change the nucleus as well. Only changing the cell shape is not sufficient. And what we found is if you have cells cultured on the 10 micron microgrooves for days, there's some chronic change in the chromatin. And you could also culture cells in individual matrix islands with different shape. And what we found is when the cells have elongated shape, it seems there's an enhancement of this H3K4 methylation and histone acetylation. And this is a global change. But if you examine some of the specific sites that are relevant to cell reprogramming, for example, if you want to turn the fibroblasts into iPS cells, at a promoter of SOX2 and NANOC, you can use a cheap qPCR to show that indeed you have increase specifically of H3 acetylation and also this H3K4 methylation at certain sites. So these could be useful for specific cell phenotype change. In a recent study, we use reprogramming of fibroblasts into neuron as a model. In this case, we introduced 3 transcriptional factors. These are discovered by Warnick's group at Stanford. And we introduce these factors into cells, and we do different kind of mechanical treatment to see how this affects the reprogramming process and efficiency. So one simple thing we found is that when you add these factors to cells, at an early stage, there's actually an increased cell adhesion, cytoskeleton, etc. Actually, that retards cell reprogramming. And then we did some experiment to disrupt these stress fibers in the cells with blebstatin treatment. You can see here there's a dose dependency. If you treat the cells at an appropriate concentration of a blebstatin, you can boost reprogramming efficiency to a certain level. And if it's too much, actually it's detrimental to cells. And we examine a lot of histone marks and also DNA methylation. What we found is that if you treat the cells with blebstatin, in this case to relax the intracellular tension in actin, we could increase ACH3 in H3K4 methylation. And this actually is accompanied by the changes of activity, for example, histone acetyl transferase, and then there's a reduction of HDAC. So these could, we have done a further experiment to show these actually are related to the increase of acetylation. And on the other hand, for the H3K4 methylation, we also see corresponding changes of these enzymes. So that shows that this chronic exposure of blebstatin, which disrupt the intracellular tension, can induce enzymatic changes that can help the chromatin reorganization. And we also perform genome-wide accessibility assay by use ATAC sequencing. And here you can see, actually we have a genome-wide map. You just want to show a few examples of the neuronal gene. And for example, tubulin, ASCL1, and MID1L. And you can see in some of these regions, you do have increase of these peaks, which indicates the increase of accessibility. This is the result of disruption of intracellular tension. So there are many studies, if you've seen in the literature, that extracellular matrix cell adhesion, all of these can induce some changes in the cells. However, many of these microenvironmental factors cause some confounding results, and sometimes even conflicting results. You can see some reports shows the increase, and the others shows the decrease of certain epigenetic marks. So one thing we tried to do a few years ago is to remove all of these extracellular mechanical signals. Rather, we just perform a single deformation of nucleus. And with this approach, you can directly examine what happened in a cell. In this case, there's no cytoskeleton, cells are in suspension. And there are many other approaches people have tried to do single cell study. You have seen some examples earlier. For example, micropipettes, AFM, or even optical tweezers. And what we did was to use microfluidic channels. You can use different sizes or lengths of the channel to apply the deformation to cells. Actually, we have done some optimization, show that a certain aspect ratio of these channels is important to apply this compressive force to cells. In addition, you can also have this cyclic constriction and relaxation of the cells to have a dynamic deformation of nucleus. So for neural reprogramming, we use this protocol to perform the experiments. And here, what we did is first introduce these reprogramming factors. And when these factors are already introduced into the genome, and we induce the deformation of the cells. And this actually is a high throughput microfluidic channel. And when the cells go through this channel, it's very fast. It's very different from what Andrew just showed, in only millisecond deformation instead of hours, just like flow cytometry. Then we collect the cells and culture them again and examine the cell reprogramming after days or even weeks. So here shows some example. We did a lot of optimization and to address the issue, for example, the DNA damage, as you've seen in the early session, and also a rupture of a cell membrane or nuclear membrane, etc. And what we found is that for mouse fibroblasts, and this is, by the way, cell type specific, a 7 micron white channels in the depth is about 30 micron, actually shows very good cell viability. And in addition, that we can find the enhanced cell reprogramming. So after millisecond squeezing of cells, you do see change of mechanical property for these cells. In actually, this is probably the nuclear change of stiffness, and you can see that this change is transient. Related cells attach after a few hours, and gradually this recovers. So the mechanical softening of nucleus is a transient response to this millisecond mechanical deformation. So what happened to the specific genes of interest during this cell reprogramming process? In ALCL1 is a pioneer factor that regulates reprogramming. And what we found is that if you do a PCR, you can see with the squeezing, you have this very robust induction of these neuronal transcriptional factors in comparing to static control. And this, of course, also includes the forced expression of ASCL1 as a reprogramming factor. And then we use ASCL1 promoter-driven reporter to demonstrate the endogenous ASCL1 expression. Indeed, we have over 10-fold enhancement for this neuronal transcriptional factor expression in the cells. The other question is, when you apply this mechanical loading, whether you can accelerate this cell reprogramming process with the chromatin changes. And we use this probe developed by Pak Wang's group at Penn State. And here is a nano probe, can detect the mRNA expression, which is earlier than protein expression. So with temporal monitoring by microscopy, and you can see that indeed for neuronal gene expression, and when you have mechanical squeezing, you have early induction of these neuronal genes after 12 hours. So what about epigenetic changes? And we examine over close to 10 different kind of histone marks and also DNA methylation. And what we found is the most dramatic changes is H3K9 ME3, which is a heterochromatin mark. After squeezing, what you found is this large foci of heterochromatin actually kind of disrupted and become diffuse in the nucleus and gradually recover after 24 hours. Then we wonder how fast would this happen? Because for epigenetic state changes in general will take hours or even days. And it's a very slow process if you have enzymatic reactions. So we use a FRAP probe developed by Peter Wang's lab at UCSD, in this case, to detect directly the H3K9 trimethylation in real time in this channel. Since this forced cell deformation only happens within milliseconds, we slow down this process by applying a lower pressure in the microfluidic channel. So this process is kind of prolonged for minutes. And with this FRAP probe, you can see that this is the same cell. When the cell gets into the channel, the red color indicates high level of H3K9-ME3. And then when you get into the channel, within minutes, you can see a decrease of H3K9-ME3. And this is very fast compared to previous reported enzymatic modulation of H3K9 trimethylation. And the other thing we notice is that DNA methylation also decrease. So this is also correlated to H3K9 trimethylation because the methylation of DNA is correlated with the heterochromatin enclosed structure. And similarly, we found a decrease of these focal loci of DNA, and gradually you will recover. So what is the global effect and also local effect of this mechanical deformation of nucleus? We perform ATAC-seq, and we have shown some of these interesting changes following mechanical squeezing, and this is unpublished results. You can see here, this includes many sites in the nucleus, and we can also relate this to specific promoter or enhancer of various genes. So following squeezing, about two-thirds of the sites in the genome actually has a more open structure. And then on the other hand, about 1/3 of them has a decreased accessibility. And we look into the gene, the promoters, enhancers related to the neuron reprogramming for ASCL1 and tubulin 3 here, you can see that mechanical squeezing increases the peaks. That means the accessibility of this promoter or enhancer. And the other question is that with this transient mechanical squeezing, how is that compared to the enzymatic reactions? And we compare this with a couple chemical inhibitors. One is the H3K9 methyltransferase inhibitor, and the other one is DNA methyltransferase inhibitor to address this observation that we show a decrease of DNA methylation and decrease H3K9 trimethylation. So what we found is that indeed, actually, if you add this chemical inhibitor, you can enhance cell reprogramming efficiency, but not necessarily to the level of mechanical deformation. And we did a dose titration and showed that to the level that you reach a plateau, otherwise you probably can kill the cells. To a certain level, you cannot beat mechanical factor in this case. And we also try different combinations with different ratios and percentage of these two inhibitors. And the best we can get actually is close to the mechanical deformation. So what is the mechanism of mechanotransduction? And we know that there could be enzymatic reaction. For example, when you culture cells for long term, regulated by stiffness, viscoelasticity, or with the change of intracellular tension. But with this very transient mechanical deformation, what happened? And we think this probably is mostly related to the mechanical effect in a coupling to chromatin through the lamin. And we examined the lamin structure after squeezing. And what we found is that you can see there are a lot of wrinkling of nuclear lamin. And also there's a partial disruption on a nuclear periphery. And we know that a lot of the heterochromatins affiliated with the the laminar through the lamin-associated domains. And we did a staining. In this case, we used M6A tracer, in this case green, to label this chromatin lamin-associated domain, and the red color is lamin B1. And what we found is that after squeezing, you can see in some of the co-localization of the lamin B1 and this chromatin is decreased. That suggests there's a partial disruption of this affiliation between chromatin and nuclear lamina. So if this disruption of nuclear laminar could help reprogramming with this partial mechanical detachment of heterochromatin, in this case, if you disrupt lamin, you're supposed to increase reprogramming efficiency. So we did that experiment to knock down lamin AC, and indeed you can see with the squeezing, it's not as strong as siRNA effect on the lamin organization, but after you knock down lamin AC, and you have similar effects on the H3K9 trimethylation and DNA methylation. You have decrease of these heterochromatin marks. Indeed, if you examine a long-term effects on cell reprogramming, you knock down lamin AC, you do increase the efficiency to convert the fibroblast into neuron, similar to the effect of mechanical squeezing. So that shows the effect of mechanical deformation of nucleus on fibroblasts in the reprogramming. And the other question you ask is whether this is true for other cell types. Can we generalize this to any other cell types? If you deform the nucleus of macrophages, what will happen, right? So we did an experiment. For macrophages, we deformed this in a high throughput manner, and we try to convert this macrophage into neuron, and we got a very similar trend, and you have a induction of reprogramming efficiency, even though this is much lower than fibroblast conversion. Because for macrophages, it seems it takes much longer, and also the overall efficiency, the basal level was low. And then if you convert the cells into other cell types, whether you are being enhanced. So in this case, we use the Yamanaka factors again and see whether you can increase the fibroblast reprogramming efficiency into iPS cells. And indeed, in this case, we observe enhanced reprogramming efficiency when you squeeze the cells. You have more iPS cell colonies formed after a couple of weeks. And now we are trying to do is to scale up this process. We are trying to make a device that can process millions of cells with minutes, just like flow cytometer. In this case, we can mechanically prime the cells and make them easier to be converted into other cell types. In this case, could be cell reprogramming or cell differentiation. So in summary, what I have shown is that the intracellular tension is important. So that's one component of mechanical transduction from cell surface to nucleus. In addition, if you directly deform nucleus and partially disrupt the connections between nuclear laminar and heterochromatin, you could also induce increase of accessibility in chromatin, enhance cell reprogramming. And we actually are exploring a lot of these applications for cell engineering and for email cell engineering as well. And we believe that the high throughput device could enhance all of these applications. And with that, I want to thank the lab members, collaborators, and funding sources. Jennifer Sato did most of this work on the intracellular tension, and Yang Song did work on the microfluid deformation of cell nucleus. Thank you.
Any questions from the audience?
Very great talk. Do you think when people sort cells with flow cytometry, they also inevitably reprogram their cells?
I don't think so, because the microfluidic system, the channel is pretty big, much bigger than the cells. you do have force, shear force, or other compression force on the cells. And we've done this experiment, the enhancement of reprogramming is very low, maybe less than 2/3. So what we think is that the deformation of cell, rather than the force applied to the cells, plays an important role. So when you have this flow cytometry force system, you apply the force, but the cell deformation is not much. And actually, most of this force is probably absorbed by the cytoplasm in non-nucleus.
Yeah, thank you. The talk was very impressive. I wonder, I have a question and it goes in the same direction. What do you think are the underlying mechanisms of mechanotransduction? If you say the displacement is more important than the force, so are you making use of the elastic properties of the lamina or the viscoelastic properties? So is there a distinct temporal frequency that you're targeting with these very brief displacements? Because if it, I mean, you know, I guess if it is an elastic regime, this is more like in a millisecond type of scale, right? Or is it actually the viscoelastic properties which should be on a second type of scale?
That's a very good question. We haven't done detailed study, so I think this mechanical deformation, indeed, you can do a gradual deformation versus a sudden step change, right? So I think that definitely makes difference. You need to have a rate. The rate constant may be important. So if it's too slow, the cells can adapt to that, and probably you are not going to induce significant reorganization of chromatin. And the cells could, for example, the passive migration through the microchannel will be a good example. And this will allow a gradual reorganization of chromatin. So probably it's different from the step change. In that regard, I think viscoelastic property in this case, how you apply this mechanical deformation will be important.
Hi, my question is if somebody saw if under confinement the cells change by RNA-seq the transcription of different genes to see mechanism or something, because we saw that changing the nucleus or in chromatin. I don't know if somebody saw by RNA-seq.
Yeah. Regarding gene expression, we are doing single cell sequencing and also box sequencing, depends on what kind of cells we use to look at the mechanical changes. And what we think is that if you do not introduce all of these reprogramming factors, you just deform fibroblasts or deform macrophages, whatever, you're going to introduce some noise in the chromatin. And this can go back to the basal level after hours. So you need to capture this time window. For example, some genes become more accessible. And we are actually doing experiment. We have evidence that if you use CRISPR, you can directly target precisely to the site you think there's opening. And we did prove that. So this enhancement of a mechanical epigenetic, whatever, modulation is site-specific. We do have global changes, but we need to know the sites. So a lot of this staining we have shown are global, right? You show the global increase, but even though in some case you don't see a global change of histone marks or DNA marks, but if you do have specific local changes, that could be effective as well. So I think indeed it's gene specific in this case, and chromatin specific. Some chromatin may be more sensitive to this, some chromosome, I mean here, and some chromosome and maybe less. And it's also dependent on the location of this chromosome in a specific cell type.
Just a really short one, please.
It's okay. So long, thanks for flying across the country for that talk. So one of the really exciting big picture takeaways from your talk is really evidence that cytoskeletal tension is related to epigenetic state. And how confident can we be yet that it drives epigenetic state? Is it that the features that correlate to cytoskeletal tension also correlate to changes in epigenetic state, or does one drive the other?
I think the cytoskeleton tension, it is important because it regulates the actin cortex around nucleus. And people also talk about nuclear actin as well. And we don't know what's going on inside the nucleus yet because there are a lot of monomer actin there. They are very important for mechanotransduction in transcriptional regulation. I think it's not just a correlation, there's a direct connection. So there are many ways to modulate epigenetic state. The change of the intracellular tension definitely has effect on the epigenetic, on a chromatin organization. And even the localization of these enzymes that modulate the chromatin. For example, some of these, for example, DNA methylase, they're localized as nuclear periphery. And there's a strategic goal here is to maintain the heterochromatin stability. So when you disrupt the mechanical structure, you also change the distribution of these enzymes, as you've seen in the previous talk by Andrew as well. When you deform, you can see the difference in some of these enzymes as well.
Okay, let's thank Dr. Lee for his talk. The next two presenters were awarded the highest scores for their presentations from the CEMB Postdoc Preview Day, which was held yesterday. And this event provided applicants with the opportunity to learn about training opportunities, meet faculty and staff, and explore resources at all CEMB institutions. This program focuses on exposing underrepresented populations to the cutting edge research of our center and encourages them to explore with us. This year, we had 10 potential postdoctoral fellow participants, and we look forward to holding this event again, so please nominate your trainees. So the first talk will be by Deepti Rana. She's a postdoctoral researcher at Dankook University.
Yeah. So hello, everyone. Okay. Hello, everyone. I am Deepti Rana. I am a biomaterials researcher, and today I'll be talking about my PhD work, which I completed back at University of Twente, Netherlands, at the Viscoelastic lab. As the name of the lab suggests, our lab was interested in understanding how we can mimic the natural organization and functionality of vascular networks in vitro in the lab. However, as we see in this video, cells in general and these vascular networks are very dynamic in nature. As you can see in this video, the MSC spheroid is trying to sense its microenvironment, and in this process, it tends to migrate away from its initial position to somewhere else within the scaffold. However, the similar behavior, vascular networks also pursue the similar behavior, and then they tend to lose their functionality over time. For this purpose, we were able to identify two key factors which can have the major influence on vascular network formation, being the fluid flow and biochemical cues, that is the growth factors. And within my PhD, I was focusing on developing tools that can spatiotemporally control growth factor presentation within a 3D scaffold. For this purpose, I went ahead and used aptamers, which are nothing but DNA-based oligonucleotide sequences that can form 3D conformations and bind to any target biomolecule with high specificity and affinity. This kind of reaction is reversible in nature, and when presented with a complementary sequence that have a higher affinity with an aptamer, it tends to hybridize with each other, releasing the biomolecule on demand. Similarly, this kind of system gives us an opportunity to mimic complex processes such as angiogenesis within our labs, where we have control over the presentation of multiple growth factors within the same system when we provide growth when we can release their growth factors at different time points during our experiments. With this basic idea, I was able to develop aptamer functionalized hydrogels within my PhD, which were able to, sorry, it's going very fast, which were able to sequester growth factors from the cultural medium within 1 hour of its incubation. Not only that, these hydrogels were also able to release the growth factor from themselves when presented with a complementary sequence. As you can see in this graph, we were able to get nice growth factor release of VGF, which is vascular endothelial growth factor, a pro-angiogenic growth factor, at day 5 and day 10 from these hydrogels when we presented with a complementary sequence. This was happened only within the acridite functionalized samples, where we have covalently cross-linked our aptamers within the polymeric backbone of the biomaterial, in contrast to JELMA and our control aptamer, which is just physically diffused within the hydrogel. Having confirmed the 3D presentation of these growth factors within our hydrogel systems, we were next interested to identify if this kind of presentation can have actual control on the cellular behavior. For this, we went ahead and developed these micro patterns where we patterned our aptamer functionalized hydrogel next to plain Gelma hydrogels and loaded them with VGF for just 1 hour. And we cultured them in the presence of mesenchymal stromal cells and endothelial cells together. And we were very happy to see that the endothelial cells were able to form nice vascular networks, which were not only cells trying to clutch on with each other, but are actually trying to luminize themselves and have functional filopodias and laminopodias within a 3D biomaterial scaffold. Having said that, we were also interested to upscale our technology to be biofabrication friendly so that we can have more control over the specificity of a scaffold. And then we went ahead and developed these bioinks, which are aptamer functionalized bioinks that can be used to 3D print simple constructs like lines. Here I have shown these are simple aptamer based hydrogel based lines next to the plain Gelma lines. And we can also upscale these designs to complex vascular tree like structures. Here, I would like to hold your attention to this beautiful picture where it highlights an interface within this bioprinted construct, where we have aptamer region and Gelma region. Of course, we have incubated these samples within our culture medium, which is loaded with VGF for 1 hour. And we were very happy to see that these endothelial cells could actually sense the localized position of VGF, and they were able to form these beautiful vascular networks confined only within an aptamer printed region, which is a very good thing to see within these samples. Here, I have also included one of like a direct application of this kind of technology, which I developed during my PhD, for which we were also got awarded this proof of concept grant from ERC, where we show these aptamer functionalized hydrogels as bioactive sealants, where they can not only seal active wound at the human tissue, but they were also able to recruit or sequester growth factors from the surrounding tissue fluid. This way we can create a growth factor localized zone within the tissue itself, and which will able to stimulate vascularization at the wound site. This is a proposed proposal. This is just the proposed idea, and we are working towards it. So during my PhD, I was able to identify the importance of biomolecule presentation and how this is helpful in defining the cellular behavior within a 3D biomaterial. And next, in future, I'm also interested to include ECM dynamics using dynamic hydrogels within these scaffolds, and then see the cellular behavior all together in combination of 3D presentation of growth factor as well as the ECM dynamics. Finally, in conclusion, I would like to reiterate what I have already showed to all of you, that aptamers provide a dynamic control, not only spatially, but also temporally over biomolecule presentation. In 3D scaffolds, this technology is biofabrication friendly, and where we can use it not only for delivering growth factors or biomolecules, but also sequestering them from the target site or site of interest. Finally, aptamers in a combination with dynamic hydrogel provide us a very good platform for mimicking the native ECM complexity for different kind of experiments, not only restricted to tissue engineering and regeneration, but also can be explored in mechanobiology or furthermore. With this, finally, I would like to thank my mentors and acknowledge all the funding sources. And finally, thank you all for your attention, and I'll be happy to answer your questions or even talk for any future collaborative ideas in the breakout room. Yeah, thank you.
Thank you, Deepti. Any questions for Deepti? Okay, we'll then move
we have one question there.
Oh.
Thank you. That was very impressive. What is the technical resolution that you can achieve?
Technical resolution of?
Yeah, for the vasculature that you can print.
Technical resolution of the vascular network, what we observe within these biomaterials.
Yeah.
So the lumen, what I showed, it's ranging from 2 micron in diameter, and these are like self-organized lumens. So this is like some primitive form of microvasculature. Yeah.
So how long does it take to print such a network?
Yeah. Yes. Thank you.
Now, how long,
Sorry.
How long does it take to print like a 3D network from like with 2 micrometer resolution up to like hundreds of microns and centimeter, maybe millimeter scale?
So the resolution of the 3D printed construct, each line is actually 400 micron in width and in height as well, 400 micron. And the networks which are formed are within these within this 3D printed construct. What I was saying, 2 micron is the lumen diameter. Yeah.
And how much time does it take to print one?
Yes. So the printing of the construct, it's actually depending on the complexity of the construct. Since we were doing just the simple lines, it was within 5 minutes. Yeah. Yeah. But we cultured them for 10 days. Yeah. Thanks. Any other questions?
Thanks so much. So I was wondering if you could tell us about the mechanism of the aptamer binding to specific proteins, and how do you achieve specificity so that other growth factors don't just get soaked up?
Yeah, so actually this is like a well-defined technology called SELEX. So this is the method of selecting your aptamer sequence so that it can have a higher specificity for a target biomolecule. So based on that technique itself, we try to deduct other sequences of DNA which will not bind to any other kind of biomolecule, and then you find a sequence which will be highly specific for your target biomolecule. So this kind of sequence, this sequence in particular, which I have used, is already defined in literature and very well studied. But so far, people are interested in studying in pharmaceutical range of ideas with these kind of aptamers and biosensing. And this is the first time when we were able to show the cellular impact. You know, we can use this technology to even for tissue engineering and controlling the cellular behavior. So that is the kind of core. But these aptamers are very well studied in biosensing area of biomedical research. Yeah.
Wonderful. Thanks.
Thank you.
Okay, let's thank Deepti again for that beautiful talk. Next up is Niroshan Anandasivam from UC Berkeley, where he's a PhD student.
Hi, everyone. It's really a pleasure to join you all. I'm joining you all from Durfee Aldo Cassian's lab at UC Berkeley. My name is Niroshan, and today I'm really excited to share my work with you all about how engaging adhesion receptors at the T cell surface can influence T cell activation. And the primary motivation for this work lies in the idea that a very important step of the way that we make adoptive T cell therapies, which are an up and coming class of therapies mainly used to treat cancer these days, is the T cell expansion step. And so this is a step in which selected or engineered T cells are expanded to clinically or therapeutically relevant numbers. And the current status quo in industry for expanding T cells ex vivo is what's called the Dynabead. This is a very rigid plastic polystyrene bead that's coated with what immunologists consider the 2 main activation signals for T cells. These are antibodies against CD3 and CD28. However, in reality, in vivo, at the immune synapse between dendritic cells and T cells and lymph node, the environment is way different than this very rigid environment with only 2 signals to activate T cells. And so this led me to ask the question, what kinds of cues are we missing from the way that we activate T cells ex vivo? And I point to 2 examples of cues that may be important to incorporate to improve our methods. The first being the idea that the matrix environment that T cells encounter in vivo and the lymph node is way softer than this gigapascal stiffness rigid bead. And additionally, there are plenty more receptor ligand interactions that I'll touch on that are important at the immune synapse between T cells and dendritic cells. So just to go a little bit further, several groups, including the Burkhardt group at UPenn and Claire Hivrose's group, have shown that upon maturation, dendritic cells stiffen, and dendritic cells are the activating cell type for T cells. Separately, on a larger scale, one group from UCLA, the Butte group, has shown that at the entire scale of the lymph node stiffens and softens over the period of viral infection. And even more specifically, some groups like the CAM group have shown that T cell activation is actually very pointedly responsive to matrix stiffness, and that you can modulate the level of T cell activation by changing the stiffness of the matrix that T cells are activated on. And so I'm bringing in 2 co-stimulatory adhesion receptors into the play here. These are CD2 and LFA1 at the T cell surface. And these 2 proteins are implicated in force transduction through their tethering to the actin cytoskeleton at the membrane. And through their role as adhesion receptors and integrins, I hypothesize that their roles in co-stimulation are likely responsive to the mechanical environment, specifically matrix stiffness. And so this leads me to the project that I'm presenting at the symposium today. in which I'm investigating how engagement of adhesion receptors can influence T cell activation in conjunction with matrix stiffness. And so this is kind of all the work that I'm presenting on my poster, but for this short talk, I'll be focusing on the most interesting part, which is looking at how engagement of these adhesion receptors by antibodies can vary the activation that we see in T cells. And for this work, this is a general method that I've used. I developed a PEGDA strep-tavidin system for immobilizing biotinolated ligand at varying stiffnesses. I incubate them with T cells and read out two main metrics of early T cell activation, which are the expression of early activation marker CD69 at the T cell surface, as well as the secretion of activatory cytokine IL-2 measured by ELISAs. And so my work initially shows that upon added engagement of LFA1, in addition to the classic anti-CD3, anti-CD28 control, we see a mechanosensitive increase in T cell activation past 2 kilopascal. However, this is pretty similar to what we see with the classic Dynamide-like control that's currently used. However, interestingly, upon added engagement of CD2 instead, we see an abrogation of this mechanical threshold for expression of this early activation marker CD69, which is interesting to see. Looking at a different metric, secretion of this activatory cytokine IL-2, we have a pretty interesting result where at stiffnesses higher than 2 kilopascal, engaging these adhesion receptors actually can increase the level of cytokine that is secreted by these T cells, which both of these pieces of data show that we can tune the level of activation by incorporating engagement of these adhesion receptors at the T cell surface during activation. One of the next questions I wanted to answer is how engagement of these receptors by antibodies might vary from lower affinity natural ligands that may also have a different target binding site. And these are the in vivo natural ligands for these adhesion receptors. In short, the answer is that I found is that there is not much of a difference. And this might be sensitive to a lot of other factors, but we can just talk more about that on my poster if you're interested. Just for the purposes of this short talk, I'll be focusing, or I'll save that for the poster. And so just the main takeaway that I want to share with you all today is that engagement of these adhesion receptors at the T cell surface really do have an effect on early T cell activation. That we can think about how we can incorporate these different cues into the way that we activate T cells and the environment that they're exposed to during our methods for ex vivo T cell activation and expansion, hopefully to improve the way that we make T cell immunotherapies in the future. Some future directions from this work that I'm looking into are characterizing later stage activation outcomes of these T cells, like phenotype polarization, as well as looking at some signaling pathways that might be propagating the effects we see on T cell activation. And so with that, I'd like to thank you all for listening. I'd like to thank my PI, Derfik and our lab for their support. Also, I want to give a special shout out to Dr. Abriata Williams-Jones for her work at the CEMB Institute and for making the postdoc preview day happen. She's been doing some really great and amazing work here at the center, and I'm excited to see what the impact of her work is in the future. So thank you all, and I'm open to any questions you may have.
Questions for Niroshan?
Thank you for the nice talk. I was just wondering if you have a clue about the number of cells that will adhere in the presence of LFA1 and or CD2, because the polyacrylamide gel is not really sticky for T cells. So did you look at the attachment or the number of cells that attach in the different conditions?
Yeah. So first, I guess the system that I'm using is not polyacrylamide, it's PEGDA, polyethylene glycol diacrylate. So it's a little different. I've worked a little bit with polyacrylamide. I've noticed that it is less sticky, and I've seen that with the scaffold that I use, there is a lot more adhesion in this PEGDA scaffold compared to polyacrylamide. And I do see a lot of adhesion, and it's pretty similar across groups, although it may be a little bit more that in the conditions with adhesion receptors versus the control that doesn't have those. Yeah.
Okay, so let's thank Niroshan again for a beautiful talk, and as well as all of our speakers from this session. Lunch will be served in the atrium right outside the auditorium. Thank you all for attending.
All right, hello everyone. Welcome back now to our third and final presentation session for the day. going to kick things off with Matt Lang, who's a professor of chemical and biomolecular engineering and molecular physiology and biophysics at Vanderbilt. And he'll be telling us about parsing mechanosensing through alpha, beta, gamma, delta, and CAR T receptor constructs. Matt.
Thank you. Want to thank the organizers for the invitation. It's nice to see old friends and make new friends. And also nice to see the Mechanome alive and well. So I'm going to tell you about the T cell receptor. This is the alpha beta T cell receptor. It's a heterodimer, and it's kind of like an antibody. It has these variable domains and it has these constant domains, but it senses this yellow guy here, which is like a 9 or 10 amino acid peptide. Some of you are very, very familiar with this. It's kind of like a hot dog in a bun. The bun is a molecule known as MHC, which is on our antigen presenting cells. And we want to sense peptides that are diseased or mutants and things like that. This is how our bodies protect themselves. So I've kind of got a story, a logic story, but I want to show you, I like starting off with this movie I got from my longtime collaborator, Ellis Reinhertz, and it's T cells that are scanning these antigen presenting cells. And so this is a very physical process. There's a lot of shearing interactions that are going on here. Let's see if I can advance. Okay, the job of the T cell, a really good T cell, even though it has so-called micromolar affinity, a really good T cell can see 10 molecules on an antigen presenting cell. We call these digital T cells. They're able to detect and to kill them. Hopefully this thing will work. And I want to just sort of show pictorially, those red guys are the foreign peptides that might be decorating a disease cell. That is amongst a sea of self-peptides that the T cell receptor has to ignore. So it has this sifting through a haystack problem that it has to undergo, where it tests every strand of hay and determines if it's self or non-self. So I wanted to kind of like at least seed you with some of the assays that we do. And the biophysical assays that we do, most of them are surrounding optical tweezers. And so there's this one assay called SCAR, single cell activation requirements, where what we do is we take a bead that we coat with peptide MHC. So the bead's in blue, peptide MHC is yellow, and we coat it at different densities. We trap that bead and we place it against a T cell. I'll see if I can get the movie to play. So we trap the bead, we do a set of calibration procedures so we can very precisely track the bead and apply specific forces on it. And then we use the piezo stage, which holds the T cell, and we apply a shearing or a normal force against the T cell. Meanwhile, we have the T cell impregnated with a calcium sensitive dye, and we can see calcium increases, which is indicative of the T cell activating. So we're trying to recreate this interaction. And if I go and show you the next slide, what you're going to see are a series of beads, some with different interfacial concentrations at the bead cell interface. And we can get all the way down to a single molecule, which doesn't trigger, but we can trigger reliably with 2 molecules if we have the right amount of force. With 29 molecules, we also need force. We can go to saturating with thousands of molecules. We want to see what are the physical and chemical requirements for activating the T cell. And if I play the movie, you can see that this cell with enough force gets activated at 18 piconewtons. But if I only put 12 piconewtons across that many molecules, it won't activate. With two molecules at the interface and 11 piconewtons, I can activate that cell. But if I only have 6 piconewtons, I don't activate that cell. And so we can go ahead and score cells that get activated here versus cells that are at the baseline in terms of their initial fluorescence. So that's one of our assays. Another assay is where we actually do a single molecule measurement on a single cell. So we take a cell and we trap a bead, but between the bead and the cell, there's a DNA rope. So there's about 1,000, there's a micron length of DNA, 3,500 base pairs. And on the end of that, we have an antibiotin Fab that binds peptide MHC. And we present that single peptide MHC to the surface of the T cell and pull back. And we can make a single molecule measurement on that interaction, the lifetime as a function of force, and look for things like conformational changes. And this is sort of the workflow of it. This is work that Evan Kirkpatrick is doing in my lab. And this is sort of what we get. So we pull to a certain force, and then there's a lot of compliance in the connectivity. So you have creep on the baseline, but you see this conformational change, and then you see a rupture. One of the things that we can get with this is a catch bond profile by mapping the different force that we pull to and what the lifetimes are. And typically we see this sweet spot at 15 piconewtons. It's indicative of a good T cell peptide interaction. There's another cleaner assay that we can do that we call single molecule. And here what we do is we work, if we can work with purified components, we can put them directly on the surface and then have a very well-defined load connecting pathway to make this same kind of measurement. And this was originally developed by Debendu Das, who was a great postdoc from my lab. And we ramped the system with the black dotted line. Then we have a pre-transitioned well. We see a transition going from green to blue, and then we see a final rupture. And then this is an actual trace, and you can see this transition. And then these are catch bond profiles between peptides that are specific for the T cell and peptides that aren't. The blue one, it isn't, and you see a slip bond profile. But again, you see this sort of sweet spot at 15 piconewtons. But a couple things I want to point out. One is that there's a conformational change that's massive. It's on the order of 10 nanometers. That's almost the same size as the complex. It's a large percentage of the complex. And then most of the assays that we're doing, if we're doing a force-free assay, we're here at no force where we don't have any sensitivity difference between the different peptides that we want to see. So we need to find ways of doing assays under load, specifically at 15 piconewtons where things are happening. Just to try to drive home this point, if I parse the amount of time pre-transition versus post-transition, the catch bond is actually in the post-transition dwell state, the extended state of the T cell receptor. So we think this is the relative strong binding state, which only occurs under load. If we think about the amount of work associated with that, I've got 15 piconewtons times 10 nanometers, that's 150 piconewton nanometers. That's 37 kT worth of energy associated with that. That's the same amount of energy as 2 ATP molecules. That's a lot of energy that's there to drive mechanosensing every time this thing is reading a molecule. And we think that, and in the sweet spot, this thing hops back and forth and causes signaling. So where is this conformational change? Well, let's compare some different constructs. Let's look at the alpha beta T cell receptor versus gamma delta. So let me show you a little bit about what they are. Gamma delta T cell receptor is shown here. It's found in barrier tissue, skin, places where there's a lot of signal that's associated with this. One of the things that I want to point out is there's not as much, there's not as much stuff between the constant domains and the variable domains. There's more connectivity between the constant domains and the variable domains of the alpha beta T cell receptor. My pointer isn't working. Okay, but what we did was we compared a straight up gamma delta cell to a gamma delta cell where we took the constant domain of alpha beta and sutured it in there. We call it the chimera. That's the one in the middle. And then we compared that to the alpha beta cell. And what we found was that what I'm showing in green is the normal catch bond for the alpha beta. We were able to recreate the catch bond profile in the chimera, but the gamma delta itself is way down here. So we were able to see sort of a gain of function with the chimera by suturing that in there. And then also we can do this not only on purified components on the glass, but on the single molecule, single cell. We also see the catch bond, whereas we see a slip bond in the gamma delta system. And then if we take a look at conformational transition, we saw the conformational transition reliably in the chimera, which is the red curve. The purple curve is the gamma delta. We only saw one transition in the whole study on the gamma delta, and that was out at 30 piconewtons. So the thing that is causing a conformational change seems to be located in the constant domain of the alpha beta T cell receptor, the thing that we sutured in there. So that was a major clue. Now let's look at the alpha beta T cell receptor versus a CAR T cell. And this is a study that was in collaboration with Nick Gasconi at the National University of Singapore. And the nice thing about these CARs, let me show you, the CAR is a strategy where you kind of take a conventional antibody and you attach it to T cell machinery. But the CAR that he had bound MHC. So it was binding the same target that our T cell receptor was binding, okay? So here I've got the alpha beta T cell receptor, but here I have an antibody where they can go through more rounds of affinity maturation and really get strong binding to these things. And we did this study both at the single molecule and on the cell level. So with purified components down here and on a cell. And this was done mostly by Debendu Das in my lab. And so what we found was kind of interesting. I'll just walk you through these studies. The T cell response is the green curve. And so it's kind of down at low lifetime, but you can still see a catch bond between 10 and 20 piconewtons right there. The CAR showed a catch bond, but at a much higher force, up there around 25 piconewtons. That was a bit of a surprise to us. So we also worked with the conventional CAR, a CD19 CAR, and that showed a slip bond profile shown here in black. Okay, so one of the things about the CARs is that right here at the sweet spot of where you're engaging the mechanosensing machinery, the lifetimes are all the same, but you have a lot of lifetime at higher force, and here you have a lot of lifetime at low force, but it's not a good impedance match to what you really want to do with engaging this machinery. The other thing that we saw when we looked for the probability of seeing the conformational change, most of the time we saw the conformational change 78, almost 80% of the time in the T cell receptor, but it was down around 12 to 15% for the CARs. And so there we had replaced this constant domain of alpha beta with an antibody that doesn't have a transition. So that logically points to the constant domain of alpha beta as being where the transition is occurring. So that was another clue. And I think we did some more studies, but I'm gonna skip through this. This was just recapitulating things with purified components. Now, another thing that I wanted to note that's part of this logic is we compared the pre-T cell receptor with the alpha beta T cell receptor. Now, the pre-T cell receptor, it's kind of half of the T cell. So you have beta, but instead of alpha, you have this thing called PT alpha. And this is just a comparison of the two. The pre-T cell receptor has a mechanosensor and it sees peptides. And what we found in that is that it hops back and forth. It hops very well, just like the T cell receptor. Here's some hopping in the T cell receptor. In the previous slide was hopping in the pre-T cell receptor. And so if we go and analyze the hopping, the forward and backwards hopping, the equilibrium force between these two, the pre-T cell receptor in pink and the alpha beta in blue, we get the same equilibrium force, the same critical force right here, around 13, 14 piconewtons. So it's telling us that it has a similar activation barrier between the two. So we think that the activation barrier is not only in the constant domain, but the constant domain of beta based on this part of the challenge. But let me just show you hopping. With this technique, we can kind of very precisely tune the force between these systems right at the critical force. And this particular one had a critical force that we scanned from 15.6 to piconewtons to 16.5. And we're able to change the equilibrium, just like with the DNA hairpin, of hopping back and forth between these states. And we can go ahead and use techniques that were developed by others to analyze the probability distributions and get activation barriers for these different things. Okay, so there's another part of the story that I want to sort of give you, kind of end with what we think is going on, but there's another part of the story that I want to encourage you to take a look at, is the alpha transmembrane domain. There's two states of that. One is an L conformation and the other one is an extended conformation. And we think this is where the energy is going, to toggle this from an L state to an extended state, which destabilizes this nice organization of the transmembrane helices and leads to a dissociation and exposure of the ITM domains, which are sequestered. So we think that this is where this system goes, and we even have some measurements on some mutants that destabilize that appear to show increased activation. So these are mutants with only 2 molecules and force that are leading to activation. So how do we think? I want to leave some time for questions, but we think it's in beta. And what is our working model? Well, I encourage you to take a look at this paper by Wanmuk Wong, another longtime collaborator of mine. And what we think is that under no load, all the extra stuff between the constant domains and the variable domains slightly destabilizes the proper interface between peptide MHC and the variable domains. But under load, that gets organized and stabilized. And one of the things that Wanmuk studied was just the variable domains, no alpha, and that had the best interface between those two. And so if we have a conformational change down here that just gets rid of this stuff and allows for really good matching between the variable domains and peptide MHC, you have a really strong extended state, strong binding in the extended state. So we think that force organizes that interface and allows it to read the peptide. And what we think is going on in the system, I've got the antigen presenting cell up top. We think it needs to be coupled to cytoskeletal machinery, which will depend on the compliance of the environment that the T cells are found in. We've got the alpha beta T cell receptor. The T cell itself has to be connected to machinery. We've even seen some motility, some myosin-based motility, and you get a shearing force between these two interfaces that pulls on this structure and reaches 15 piconewtons. And then what you get is a following of the motor to keep it in that sweet spot where it can just hop back and forth and eventually toggle the transmembrane domain where you get initial signaling. So that's our working model. And I want to acknowledge just a wonderful collaboration with Ellis Reinhertz over the years. He's been very patient with me being so clueless about immunology, and it's such a daunting area to kind of get into. But we've also taught him a lot about single molecule biophysics. And I've worked for many years with Wanmuk Wong, so the teams there, and then all the people from my lab, especially the initial development with Debendu Das and at the single molecule level, and Fang Yinian with the single cell study and our funding from the NIH. So I'm open to questions and thank you for your attention.
All right, questions from the audience. Already got one.
Yeah. Yeah. We think that it's going to be in the same range. When the cells are crawling over top of surfaces, they're exerting a tremendous amount of force. There's a lot of force between the two, but we think that it's probably going to need to be at a level where it's exerting 15 piconewtons on the actual molecule itself. But we think that that's in a perfect range from, the forces that cells can be shown to exert on their surfaces. Yeah, it'll be a shearing force, which can recapitulate itself as tension in this system. We've shown that it tends to like shearing better than other directions, but we can also trigger with a normal force. We and others, there's been some nice work from the Chengzhou lab with the bioforce probe, where they have more of a normal force geometry. It does have some shear components in it, but they can get activation there. But we think that shearing is the ideal direction, and that makes sense to me with the way that the cytoskeletal machinery will be organized for a crawling cell. Yeah, yeah.
Yep. Thank you for the beautiful work. I have so many questions. Just to start with, what do you think about then how the differences in the alpha beta and the gamma delta T cells translate into signaling and kinetic proofreading in particular?
Well, I think that it's a different, I think of gamma delta cells as having plentiful ligand during a stress response. And so they don't need to be digital. I think that alpha beta T cells can go both ways. Sometimes they'll see plentiful ligand and they'll be fine, but sometimes that ligand isn't really plentiful. And so we want the very best digital T cells. I do think that there are other mechanisms that will be enhancing this interaction, especially if you can bring the leaflets together in some of the accessory molecules. So I think that those things just enhance the signal. This isn't the only aspect of the signaling. Yeah, thanks for your question. Yeah.
Great talk, very cool. So I was wondering if you looked at the distribution of attachment durations at the different forces. You're reporting a lifetime. So for example, at 15 piconewtons versus 5 piconewtons, are those both exponentially distributed, or could you learn something about the unbinding mechanism?
Yeah.
Looking at
Yeah, we are starting to look at those distributions. It's tough because you've got a bin. It's tough to get these measurements, but we think it's exponential distributed within those. But we think that there might be something special at 15 piconewtons where it's hopping, that it can reset itself. And so that's the one that I'm really interested in, is that hopping distribution non-exponential or just sort of very different from what we see on the edges of the catch bond.
Hi, great talk. So I have a question about, I guess, so there's been reports that you can elicit like low levels of T cell activation with dimeric but not monomeric anti-TCR antibody, which is a different situation than what you show. But do you have any thoughts about how the kinds of force thresholds that you've discovered may be applied to this context of like an antibody?
So I'm wondering if it's if the dimeric is what it's doing is cross-linking two separate sets of machinery on the same cell and allowing you to pull through that bridge. We work with monomeric, but one of the things that I'll point out is it was subtle, but in that initial SCAR assay, we tried triggering very hardly with the single molecule, and we couldn't do it. So I think this second one is needed for organizing the interface. So we need two. I don't think we can do it with one unless it's a really strong binder and we're sitting at 15 piconewtons. So this guy here just has one, and it has sufficient force, but we don't think that the machinery underneath is organized the way it needs to be, the way that the motor can follow and keep it in that sweet spot for activation. That second molecule kind of anchors that railroad down properly. So I think that's part of the story of why you might need two and not one.
Thank you.
Thanks. Great talk. So I have a couple of questions. The first is, I'm curious to know if you think the force thresholds are similar across different memory T cell subtypes, for naive versus central versus activated memory T cells. Do you expect similar thresholds?
I think they are similar at the single molecule level. We have seen some differences in naive T cells versus other ones. I forget what it was. Maybe it was whether we could trigger it with normal versus, because there's a density difference of the T cell receptor in those cells. The naive ones are small but mighty. And so there's more density there that I think played into this activation story. But the actual barrier, we think, is the same in the two.
Okay.
Yeah.
Great. Now the second one relates to CAR T. So a lot of our traditional CAR Ts exhibit a slip-on behavior, right? So I'm curious to know, I guess conceptually, your thought about how it go about enhancing CAR T therapy by maybe making it more catch-on.
Yeah, thank you for that. I think that part of the problem with some of the CAR Ts is they're not engaging a lot of the machinery that we like. So we're missing that alpha transmembrane domain. We're missing that 15 piconewtons hopping back and forth that we can shake things up. At 15 piconewtons, where I don't know if you remember the CAR T slide, but there wasn't even though there was a lot of lifetime at no force, there wasn't necessarily much more lifetime. Let me see if I can get to it. At 15 piconewtons, where you're engaging the mechanotransduction machinery right here in the middle, but we get strong binding at no load for the black curve, which is a conventional CAR T, CD19 CAR T. So I think there's a lot of promise where the CAR T can, I would like to see that move closer to engaging the native T cell machinery at the native forces, try to impedance match it mechanically with these mechanisms.
Maybe I can ask a question. So Matt, great talk. So I just want to have a question at the little bit higher level, right? So I would imagine, the two cells are exerting maybe nanonewtons level of force integrated. So I was just wondering how that nanonewton is broken up into.
Yeah, well, the nanonewtons
and the cooperativity.
Yeah, how it's broken up into.
So nano is 10 to the 9, and
pico,
so there are, I guess there are 1,000 receptors, right?
Yeah, and I think that it is grabbing and reading all the self, or a lot of the self. It's like a little bloodhound that's scanning all the self. So you're going to need much higher levels of force that the cells are exerting relative to each other, such that when that is divided by the number of receptors, you're in the sweet spot of 15 piconewtons, 30 piconewtons, and things like that. So I think a lot of times when we're talking single molecule, we're talking about individual interactions, but that nanonewton level of force is spread across an area that's got many molecules. And so you need that, we call that the grid. This guy plugs into the grid. That's the energy source, the cells crawling over each other, where each of these molecules only engages like a single interaction, a subcomponent of that energy and force. So I like the fact that cells can exert hundreds or nanonewtons worth of, hundreds of piconewtons and nanonewtons worth of force on their substrates. Thank you.
All right, thanks. Let us thank Professor Lang again. All right, our next speaker is Alexia Kaye. She's a research associate in the Oaks Lab at Loyola University of Chicago, and she'll be telling us today how T cells use integrin-dependent adhesion to migrate in complex environments.
So hi everyone, my name is Alexia Kaye. I'm a postdoc in the laboratory of Dr. Patrick Oaks, and I really want to take this opportunity to thank the organizer for this opportunity. So today I'm going to talk to you about how T cells switch a migration mode to maneuver complex environments. So the main focus of my project is whenever CD4 T cells are activated by antigen-presenting cells and then start to migrate in the rest of the body toward the inflammation site. As they do so, you can imagine that they will migrate in many complex environments that are composed of collagen fibers, fibronectin, chemokines, and other cells. So as they need to maneuver all of those new physical cues, they will mostly be migrating in what we call amoeboidal migration, so mostly in confinement. So in comparison to mesenchymal migration, which has been really well studied, and we know how those cells interact with the substrate, creating focal adhesion that are composed of many layers of proteins, how amoeboidal cells are interacting with the substrate is still poorly understood. So one of the main papers that has studied that is this paper from Michael Sick's lab in 2008 that has studied dendritic cells in collagen gel. And they have found that when you remove integrin, cells are able to migrate just fine. So here you have the cell tracks and the speed are not affected by removing integrins. But when you do a similar experiment, this time in vivo, so this is an inflamed ear of a mouse, when you track T cells, what you see is that when you block integrins, cells are no longer able to migrate. So the difference between the two of integrin dependent or independent suggested to us that maybe how the environment is organized may be affecting their migration mode. So to dissect that, we started to just try and see how the confinement and the ECM was affecting cell migration. So first of all, we looked at ICAM, which is the strongest adhesion protein you have on blood vessel, as you may know. So cells are bonding to ICAM without confinement and are migrating just fine. But when we instead let the cell migrate without confinement on fibronectin, which is the major ECM in the rest of the body, what you're going to see is that they're not actually migrating anywhere. They're mostly creating Brownian movement and just wiggling around. But as soon as you confine the cells, what you see is that you recapitulate what you had on ICAM. So they start migrating just fine when they're in confinement on fibronectin. And this is the tracking. I think it's a bit more visual to see out of the tracking that you recapitulate what you had with ICAM. So like I said, when you look at the velocity and the displacement, there is not many, there is a slight difference with fibronectin, but they are mostly in the same range of velocity and displacement. But what is also really interesting is if you start comparing those results with substrate that block the integrin-based adhesion, so using PLL or PLL-PEG by example. So if the cells are not able to make integrin-based adhesion, what you see is that they are migrating less fast and less far. But again, studying cells between two flat surfaces is really far from what you have in the body, right? So what we decided to do is to confine the cells between two really soft gels. So they are really more in a snug environment. And what you see is the effect is even more drastic. So when you have no coating at all, so the cells are not able to create any integrin-based focal adhesions, what you see is that they're not able to migrate, the velocity and the displacement is not there, and they are better on fibronectin and even better on ICAM. And why is that? Is because they need to be able to grab their environment to lift the gel or make some space and migrate in between. And that's what you see in this example here. So you have the cell, so it's going to loop again, but when the cell is at the beginning, you have a space in between both gel, and as the cell leaves, the space is closed up. Okay, so, but now that we know that they react differently to the ECM, are the cells actually binding to fibronectin? So to test that, what we did here is we have micropatterning. So we have region of fibronectin here in green, and in darker, it's PLL-PEG region. And the cells are just floating on it. And what you're going to see is that when they are touching the fibronectin region, they are slowing down. And just you can imagine that a bit like when the cells are slowing down on ICAM, right? They are slowing down when they're hitting the fibronectin region. And if you're not convinced by that movie, this denser movie, I think it's even more telling. You really see all those cells floating, and they're really slowing down when they hit the region of fibronectin. So now that we know, again, like I said, they react to ECM, is ECM itself sufficient to induce migration? So to test that, we did again what I did earlier, the migration experiment, but this time I removed the serum from the media because in FBS, of course, you have some type of ECM. So what we see now is that on PLL or Timoxa, so the integrin-independent substrate, cells are no longer able to migrate. And as you reintroduce ECM, the migration is proportional to the amount of ECM on the substrate. Here on the movie, it's a bit more convincing maybe to see it by yourself. So you have the cell on Pimoxa with FBS, they're able to migrate, but once you remove FBS, they are not moving, versus when you have still a bit of movement here, but really little, versus when you have fibronectin with FBS or without, cells are able to migrate. Okay, so now that we know, like I said, they really clearly respond to ECM, they must be binding to it by using focal adhesion, right? So that's what we wanted to see. So we first use ICAM. Like I said, this is the strongest bond, so we were expecting to see focal adhesion with ICAM. We use vinculin, which is one really important protein that is part of focal adhesion. And what you see is that as the cell migrates, you have those really beautiful punctae that is really typical of focal adhesion forming under the cell as it migrates. And what you see is that when you go on fibronectin, you have the same type of focal adhesions. But what is interesting is if I look the cells, but this time on PLL or PLL peg, you have the cell jiggling a bit, but they are not actually making any focal adhesions. So if I bring back that movie again, like I said, they are slowing down on those micropatterning region. So is that switch between integrin independent and dependent fast? So that's what we wanted to see. So we zoomed in on those micropatterning regions. So here you have PLL peg, and here you have fibronectin. And what you're going to see is that as the cell hit the fibronectin region, it's going to instantly switch, and you're going to see behind some retraction tail. So it hits here, and you have some retraction tail left behind. As it leaves, the retraction tails are broken. So now that we know that they are able to form focal adhesion, are they making forces using those focal adhesions? So to test that, we use traction force microscopy. If you're not familiar with this technique, it's a polychromate gel that contains fluorescent beads, and as the cell are creating forces toward the substrate, it displays the beads, and we can measure the forces by the displacement. And in our lab, what we do is double-sided TFM, so we have gel on the bottom and on the top of the cell. So what we get is a result like, so here what you're gonna see is the cell migrate, and as it migrates, you're gonna see the gel pulling on, the cell pulling on the gel. So here you see, you see the gel is being displaced by the bead, by the cell. And here in yellow, it shows the forces that the cell is exerting toward the substrate. And what is really interesting is that I'm looking again here at vinculin, so the focal adhesion protein, and what you see is that those forces clearly colocalize with where the vinculin is. And you see the directionality of those forces are really going toward the center, which is really typical of traction. But what is also interesting is that on that specific cell, on the other side of the gel, there was less focal adhesions. There was a couple of them in yellow, but you also had a region that had no focal adhesion, and you could see instead forces around the cell. And when you look at those, what you see this time is the beads are being pushed outside of the cell. So that tells us that whenever you have integrin-based forces, it's going to go toward the center, the cell is pulling, but in the same cell, she can also generate integrin-independent forces, which this time she's going to be pulling outside the gel, like I said, to lift the gel outside of the way. So with that, I would like to summarize with my conclusion. So what we found is that when they are in confinement, cells can use both integrin-independent or integrin-dependent forces. When they're using integrin-dependent forces, they're going to pull on the substrate, versus integrin-independent, they're going to push away the substrate. And they can switch between both to whatever is making their life easier. So with that, I would like to thank my lab and our collaborator, specifically Mathieu Piel Lab, who helped us with the confiner, and all of the P1 group who helped us with cultivating the T cells. And here, just for the pleasure, I made a little macro pattern of Chicago skyline because it's a beautiful city and that's where we are. So I'm happy to take your questions.
Thank you. Questions?
Well, thanks very much. Thanks for flying out here to Philadelphia for this talk. So you saw a very interesting phenomenon whereby confinement leads to migration. Is it the integrin dependent or integrin independent forces that lead to this response? Or do you have a sense of how the mechanosensing occurs for that?
Can you repeat the beginning of the question?
Is the effect of confinement on migration, is that related to sensing of the ECM by integrin-dependent forces or by integrin-independent forces?
I think it would depend on the level of confinement, because I think at a certain level of confinement, it would be more maybe the nucleus who would detect that the cells are confined. In this specific setup, the cells are in 5-micron confinement, so it's mostly the height of the cell, so the nucleus is not touched. So I think in my opinion, it would be the integrin that would be sensing, because they have twice the amount of substrate that they would have without confinement. So I think it would be integrin based.
Very interesting talk. You only looked at the Th1 like subtype. Do you expect this to be conserved among all type of like the subtype of T cells, or is this particular to the Th1?
That's a really good question. In fact, the reason we chose Th1 is that it has been shown by our collaborator Deb that after the activation from naive to Th1 and Th2, there is an upregulation of integrin alpha V beta 3, which is the main integrin that binds with fibronectin. So that's why we chose them. So I don't expect to recapitulate that with naive T cell, and I would expect this result to be even stronger with Th2. But that's going to be the follow-up of my project. Thank you.
Hi, really great talk. I'm wondering if, from like a bigger picture perspective, could you provide more context about like what you think these findings relate to in terms of T cells in vivo, like in terms of thinking about where ICAM is expressed, where RGD is expressed, how this story might relate to what T cells experience in our bodies?
Yeah. So how we see it is that whenever a T cell is not in the blood vessel and not in the lymph node, the biggest ECM is going to be either collagen or fibronectin. So they're going to need to be able to migrate in those type of environments. And they're going to be in really tight spaces or really loose spaces. And what I think is that whenever there is, by example, if the space is really too tight for them, I think they're going to switch to integrin independent, in which it wouldn't be efficient to use integrin base. Or if in the other way around, if the nucleus is not touched and they have just enough confinement, like 5-micron or in that area, then they could switch to integrin base to help them to kind of like Tarzan and from fibers to fibers. And then when they go to something more loose, then they could go back to swimming amoeboidal. So I think they would really switch regarding how the environment is confined, how dense it is, and the composition. But like I said, there's mostly fibronectin or collagen, so we do expect them to at least use integrin in those regions.
Well, we have time for one more question if there's anything else.
Yeah, great talk. So, so the forces, in your case, stresses, they are in the 100 Pascal range, right? So, I mean, you didn't show this, but in the beginning, you motivated it with aligning collagen and, movement in collagen. Yeah. So T cells, I don't think can align collagen fibers, right? I mean, the forces are probably not big enough to compare to, say, a fibroblast or,
to pull on collagen fibers?
Yeah, and align them when they move. So I'm just wondering how they move in 3D as opposed
to... I definitely think that they can, because there is some movies that we saw from Michael Six. Yes, on Michael Six published some, I think it was quite recently on T cells, some movies of them migrating in collagen. And you definitely see as a cell leave behind, you're going to see some fibers detached from the cell, and in the front, some are being pulled. It's just that since it has always been assumed that those were not making any integrin-based adhesion, they were mostly being assumed of the cell is pushing away those fibers by internal pushing forces. But what we suggest is that in fact, we think it's a combination of both.
Okay, thanks.
All right, let us thank Dr. Kaye again. All right, our next speaker is Professor Miley Mulatto, studying molecular biology of plant microbe interactions at UC Davis, and she will be talking to us about molecular mechanisms underlying stomatal immunity.
All right, thank you. Good afternoon, everyone. Thank you so much to organizers for inviting me to come here. This is quite a treat for me because I'm a plant person and I've been learning a lot about other systems and how the work that I do can be applied to other types of research. And so this is really amazing to be here. So thanks to organizer. So I'm a plant biologist and I studied plant immune responses against microbes. And in particularly, I focus on a very special type of cell, of a plant cell, that is the guard cell. So if you can see here on the background of my slide, you can see a very nice pair of guard cells here that forms the stomatopore.
And that is the focus of my research, to see how this stomatopore can actually control plant diseases. So okay. So what is the motivation of for this research? First, we are trying to understand how the plant can stop microbial infection of their internal tissues. So that's the motivation. If I'm showing here some bacterial cells labeled with GFP, and if these are successful pathogens, they have to enter the plant, the leaf, through pores, like the stomatopore. So you can see here in this move, bacteria has to enter through these pores. And then when they do, they actually can survive in the intercellular space of the leaf. And if they are successful pathogens, they will multiply. And if they multiply, increase their population, we get diseases of plants. And this is very detrimental, as you may see. You can imagine that it's going to be very detrimental in many ways. So we want to stop that. How we do that? There's something that's called apoplastic immunity. And it means that plants can mount an immune response against these microbes and stop their population. They'll control the population. And then we can have here a healthy plant. Now, this is basic plant pathology. So it's a very typical cartoon for a plant pathologist. But what I want to show you is that I have studied how we can actually stop that penetration phase. And now we have something that's called stomatoimmunity. So it's a physical force. So if you close the pore, microbes cannot get in anymore, and then you can have healthy plants again. So that is the context of this story. And I'll focus, although I do some work on this area here, today I'm going to focus only on the stomatoimmunity, because this is actually related to the symposium, which is the physical force of keeping microbes out. So this is the stomata team. Over the years, there are huge faces here. I didn't realize how big this would be. So over the years, I had wonderful graduate students and postdocs. So the ones in red frames here already left my lab, but the ones in green, they're still working on here. So this being a work of a lot of people in the lab, because it's not very easy to study guard cells, as some of you in the audience might know, like Charlie. So it's kind of very complicated, but here I simplified the state of the stomatoimmunity story in just one cartoon over here. But this is all papers from my lab, but there's a lot more papers that contribute to this story. So we're going to focus here on what we are doing, what we have done to understand how stomata can close and keep the microbes out. It turns out that is part of the innate immune response of the plant. So when microbes land on the plant, the leaf surface, it can be perceived by receptors at the membrane. So one of their famous one is called FLS2. And this FLS2 receptor here can sense conserved molecules on the microbe. That's a flagellum. So most of the plant pathogens and also human pathogens, they have this flagellum that can be used to be, the plant recognize, human cells also recognize those through receptors in the membrane. When this recognition happens, it triggers a series of signaling molecules and a signaling cascade. This is not necessarily in this order. I just put there because that's are the components that my lab and my previous, when I was a postdoc, actually we discovered these components that will lead to stomatal closure. It's not necessary in this order. But what I want to try to show you here or bring your attention to is that we have a MAP kinase cascade, and there are some plant hormones that are involved in the response. So salicylic acid is a plant hormone. Also, you take that in your aspirin, right? And this salicylic acid is a signal to close the stomatopore. And this is something that we discovered a few years back. And receptors in PR1 for salicylic acid is also involved. ABA is another plant hormone that's been known for a long time that induced stomatoclosure. But it has not been known that it was also important for stomatoclosure in response to microbes. So, and that I want to show the actual picture. So this cartoon comes from a lot of papers, but this is what we see. If this is a leaf surface of tomato, so these are beautiful tomato leaves. It has some nice pigments here. Once it's exposed to microbes, it triggers this pattern-triggered immunity, or PTI, and then you see the stomatopores all closed. So it's a very fast response. It goes into minutes, and within an hour, most of the guard cells or stomatopore are closed in the on the leaf surface. So it's a very fast immune responses. So, but to understand this immune response, I need to disturb the system. So my lab has been focusing on ways to disturb that stomatoimmunity so we can understand what are the signaling pathways that happens in the guard cell that leads to the stomatoclosure. So one way to do it is to change the air, the humidity, the relative humidity, because high humidity actually induces stomato opening. So the first question we asked was, can we modulate stomato-immunity by changing the humidity of the air? Does it matter? Because the guard cells will sense so many different stimuli. So the answer is that actually, if you put plants under high humidity, stomata does not close anymore. So it is affected, impairs stomato-immunity. So high humidity is detrimental for immune responses. And actually, this is a bacterium pathogen that normally does not cause any disease on the plant. So it is under 60% relative humidity, but under high humidity, it enters the plant and starts causing disease. So there's direct evidence that the relative humidity is very important for this. So the question was, what happens in the guard cell? So we looked at the salicylic acid signaling pathway and see what's happening in the guard cell. Is it affected during high humidity situations? So we looked at gene expression. Those PR1 and PR2 are markers of the salicylic acid signaling pathway. And you can see here the expression of these two genes are very minimum under high humidity. So this shows, and actually, if we give salicylic acid to the plant under high humidity, it doesn't close the stomatopore. But at lower humidity, if increasing salicylic acid exposure, you see this nice decline in stomatoaperture. So the signaling pathway is actually impaired under high humidity. Well, it is very well known in the plant field that salicylic acid works antagonistically with another hormone that's called jasmonic acid. So we looked what happened to jasmonic acid in that cell. So just to give a brief overview of how salicylic acid is made in the plant. So the biosynthesis is a complicated synthesis, but I want to highlight here some genes that are markers for the production of jasmonic acid conjugated with isoleucine, which is the active hormone. And then after biosynthesis, there's some signaling pathway with some negative feedback loops here. And then you have expression of jasmonic acid genes. So again, we looked at the expression of OPR3, which is leading to production of jasmonic acid isoleucine. And you can see here that when plants are exposed to high humidity levels, as early as 15 minutes after exposure, you start to see an upregulation of biosynthesis of jasmonic acid. And this is in the guard cell. All this work was done with the guard cell, isolated guard cells. And if we have a plant mutant that cannot respond to jasmonic acid, so we don't see any difference. So that mutant shows here that the signaling and perception of the signaling pathway or perception of the hormone is required for that modulation of the pathway. And this is true for all the other genes that we looked. So now we have, within the guard cell, we show this antagonistic interaction between salicylic acid and jasmonic acid that leads to stomata opening under high humidity. So let's switch gears a little bit here and come back to the microbes, why this is important. So I just showed you that we have this beautiful PTI here about 2 hours after inoculation with a pathogen. But then a successful pathogen like Pseudomonas syringae, it produces a toxin that's called chromatin. And within 2 hours, this chromatin can actually open the guard cells again. And you can see here on the microscopic scale, and this is the whole plant scale. These are thermal images that indicates overall stomatal opening and closure. So cooler plants, the stomata are mostly open, and hotter plants, the stomata is mostly closed. So we can see this in 2 different ways. And now the question is, what's this chromatin? What does it do? How come so in 2 hours after exposure, the stomata comes back to the previous state? So then bacteria can get in. So the first question was that, is chromatin actually produced on the leaf surface? Because this is a phytotoxin that is actually induced in the pathogen when it's exposed to the plant. So we can show here is that about 4 hours after exposure, the bacterial cells start to glow, which is an indication of biosynthesis of chromatin. This is a reporter cell. We express the promoter of the reporter gene with driving GFP expression, and the cells start to glow at the same time of the stomatal opening. So a direct correlation there. So yes, chromatin can be induced, and even later, like 24 hours later, we have a lot of cells are glowing. So yes, chromatin is induced on the leaf surface and is opening the guard cell at the right time. So what is this chromatin? It turns out that chromatin, this phytotoxin produced by Pseudomonas syringae, is a molecular mimic of the plant hormone J-isoleucine. So it turns out that the bacteria, it is hijacking a signaling pathway of the plant to open the guard cell, to open the stomatopore, change the guard cell volume. So we started looking at, and this is the evidence for this. We use the mutants of the bacteria. So this is the chromatin producing bacteria, chromatin defective bacteria. And then you see about 3 hours later, the stomata is mostly open for the chromatin producing bacteria. But for the defective mutant, it cannot reopen the guard cell. So the question is, what does chromatin do? So how the guard cells perceive this? So on a related project in the lab, we were trying to understand how the JA signaling pathway works. And again, it's been a lot of collaborations, great papers. This was discovered back in 2007, a back-to-back Nature paper. We actually discovered how chromatin and J-isoleucine can be perceived by the plant. And this was done in other cell types, not just whole other cells in the leaf, not guard cells. So back in 2007. So when you have in situations when we have low J-isoleucine or low chromatin levels, what's happening, there's a repressor complex sitting on the JA responsive genes promoter. So this is the transcription factors, and this is the JAS proteins. They all form this repressor complex and repress the response of JA genes. But when you have high concentrations of the hormone or high concentration of chromatin here, what happens, it acts as a molecular glue, bringing the co-receptor COI1 and JAS together and releases the repression of the JA responsive genes. And the way it releases is that this JAS gene is actually degraded through the proteasome pathway. And this only happens when you have the ligand, which is JA-isoleucine or chromatin. Now, JAS, it is a very important component of this repressor complex because it is dictating the on and off state of the JA signaling pathway. So we looked at the JAS proteins and thought, well, maybe we should study this one and see how, which JAS proteins would be important for stomatal responses. It turns out we have one receptor for JA-isoleucine and chromatin. We have one hormone, and we have in Arabidopsis, which is our model plant, we have about 13 co-receptors JAS. And all this JAS is important for the specificity of the JA response. The way it does it, JAS family of proteins, it has these conserved domains, the N-terminus, the ZIM, and JAS domains. And these domains are important for interactions with transcription factors and other types of proteins. So depending where or with which partner the JAS is working with, we have different responses. So let's say we have the mix here, mix bind to the JAS domain, and then we have defense responses. And then JA hormone is important for so many things in plants. It does so many different things in development, in defense, freezing tolerance, senescence. So these JAS proteins will give different specificities depending where it's expressed and what are the partners of these proteins. So that was the basis of this story. So how can I connect this with my stomatal immunity story? And we looked at our favorite is JAS4. So JAS is the fourth member of that family. And we looked at if this JAS4 can actually bind to different partners of the J pathway. So yes, JAS4 binds. This is an East2 hybrid assay. So the blue color indicates interaction between the proteins. So JAS4 binds to Ninja mix, but it requires the ZIM domain to bind to Ninja. It requires the JAS domain to bind to the transcription factors. But to bind to the COI1, the receptor, it needs chromatin, it needs the ligand. So we showed here, it needs the ligand to bind the COI1, and it's required, the JAS domain is required because now we have no interaction here when the JAS domain is missing the mutant. And this one here is just the control showing that all these strains are actually expressing the intended protein. And now we have here that JAS4 actually is degraded when in the presence of chromatin. So that's the pictures that are more important here. So we have, when you expose guard cells to chromatin, the signal for JAS4 is gone. So it is degraded in the presence of chromatin and requires the proteasome pathway. MG132 is the inhibitor of the proteasome pathway. So we placed JAS4 in the JA pathway. Now, we thought, okay, what's next? We're going to develop mutants of the JAS4 protein. So we developed plants that cannot produce JAS4 proteins and looked at their guard cell. So is that important for guard cell response? So this assay here, it shows our favorite assay, which is measuring stomatal aperture width in this control. So the mutants here already has a smaller aperture in the native state. But when we have DC3000, which is our chromatin-producing bacteria, all the plants, they close their stomata in response of the bacteria. But here, look at the 4 hours later, our mutants are not able to open the guard cells anymore. So that places JAS4 in the signaling pathway for stomata immunity. And JAS4 mutants cannot respond to the chromatin producing bacteria. So we had a mutant plant, but where are these expressed? Where is JAS4 expressed? So we developed plants that are expressing a GUS marker with the promoter of JAS4 and looked at the whole seedling. We see there's some nice blue GUS expressing here, but when we looked at the leaves, JAS4 is expressed mostly in the guard cells on the leaf surface. So that it's timely, it's in the right place, in the right time to be important for stomata immunity. And here we also expressed the JAS4 plants under the control of a strong promoter. And you see it is in the nucleus of the guard cells. So also it's important to be in the nucleus because it's a transcription repressor. So now putting all this data together, we have a model where, like this simplified model, where we have the salicylic acid pathway leading to stomatal closure, and this be in the moderate relative humidity, or some microbes that will do that. But virulent pathogens and high humidity, what we have, the salicylic acid pathway, it is stopped by the activation of this JA signaling pathway, and we place these components here that induce for stomatal opening. So it is a simple model, but we are trying to build this story and see the timing that it is happening that is relevant for the stomatal immune responses. So that's all that I have, and I would like to thank you for your attention. These are my current lab, but a lot of people before them actually contribute to this research. Our funding agencies and our current collaborators are very grateful for all of this. Thank you for your attention.
Great questions.
Yeah.
Thank you. So I'm curious about the size of pathogens and whether that matters. You know, thinking back to the earlier talks on confinement of cells, does how tightly the stomatal pore close matter in terms of whether bacteria or other pathogens of certain sizes can get through that narrow channel?
Right. So that's a very interesting question. Well, it does matter because if you think about a pathogen that is a 0.5 micrometer in length, and then a guard cell pore, you know, a stomatal pore that can be as wide as 10, depending on the plant. Arabidopsis is pretty small. It doesn't matter, but it is like, how can I say? It is dosage dependent, right? So if it closes, if the pore is smaller, fewer bacteria can get in. And you'll never have 100% closure unless the immune response is very strong. And that is also transient. It will not stay closed forever because once the immune response is fading away, it will open again. However, these pathogens, when they land on the plant, they land randomly. So if you have a small closure, and then fewer pathogens can get in. And this has a big implication for downstream, like disease development, because disease development, it is a function of the population size, right, of the pathogen. So more pathogens, more disease. So it won't control 100%, but it can decrease dramatically for bacteria. But for fungus, it might be 100% because fungus is big. It's bigger, as big as this guard cell, the stomatopore. So for fungus, it could be 100%.
It was a nice talk. So you talked about the stomatal closure as a potential mechanism to stop bacterial or microbial infection. My question is, how would that affect different stomatal parameters like conductance, transpiration, respiration, and in terms photosynthesis, and how would that affect the crop productivity in general?
Yeah, that's a very interesting question as well. I've been asking that myself for a long time. It's not a simple answer. Yes, this pore is very important for many things. Is the lungs, the mouth of the plant, it's gas exchange, so it's important to be open for photosynthesis. However, this is again, so this is a transient response. Once the microbes die on the surface, because if they cannot get in, they will die, they dehydrate on the surface, they cannot get in, so plants can go back to their normal lives. So and also another thing to consider is that the signaling pathway to close the stomatospore is a different pathway that is triggered by microbe or is triggered by different environmental stimuli, right? The sensing is different. So if we think about to improve plants to have a stronger biotic stresses response and not abiotic stress response, it's possible to do because these pathways, they have some overlap, but there are some uniqueness that we can modulate.
I also have another question in continuation with what you talked about fungus, that this should be effective against fungus, but then how would you address the problem with those fungi that, I mean, you know, like the fungi like vesicular, arbuscular, mycorrhizae, those are beneficial fungi, but they have a different pathway of infecting a plant. And there are several fungi that follow another route and affect other organs. So is there any, like, do you have, like, have you thought about any preventive mechanism in that sense?
Well, for mycorrhiza is a root pathogen, right? So what can happen if it's infected in the roots, not necessarily is going to have effect on the leaf surface. So that's spatially different. But it has been shown that if we have some pathogens, root pathogen that can trigger the stomato immune response in the leaf systemically. So kind of priming the plant to be more immune to secondary infections by bacterial pathogens or foliar pathogens. That has been shown. But again, it is a transient response, and the plant may have a lower photosynthesis at the beginning. So you will stop photosynthesis for a little bit, but then long term, it doesn't matter so much. It will recover. Right?
Thank you.
So, there's another.
So it's a fascinating result that some of the pathways that are involved in protecting the plant against high temperatures get in the way of pathogen resistance. What about pathways for protecting the plant against low temperature? Do those also get in the way?
Low temperature.
Yeah, anthocyanins, for example.
Right. Yeah. So the low temperature, what happens to, we still can see stomato immune responses in varying temperatures. So the temperature does not affect the immune response of the stomata. We did that some time ago and showed that it doesn't matter the temperature, you still have the immune responses. Now, humidity is the major environmental factor for this response to happen. So is that more or less your question?
Time for one more question if we've got it. All right. Vivek.
So has the role of the plant nucleus in any of this? Again, I don't come from the plant field, as you can tell. So how much is known about the role of the nucleus? In
the nucleus on this? So all of this, it's very interesting you ask, because a lot of this signaling pathway of the JA and salicylic acid, it is a nuclear response. Even for salicylic acid, the receptor is actually, it moves from the cytoplasm to the nucleus when you have high salicylic acid. So it is mostly a nuclear response.
So how does this information go from the outside to the?
Oh, it's like a signaling cascade, phosphorylation cascade to the MAPK cascade. There's also calcium, extracellular calcium. Yeah, all the normal signaling molecules. Yeah.
So there isn't real force, but it's rather signaling that's in.
It is signaling. The force is more like the cell wall constraint, which is Charlie Anderson's domain. That's a force, because this is a growth and shrinking of a cell every day, like the somato movement. So that is the force that is very important for this function, because cell wall of the guard cells is a little bit different than the cell walls of other parts of the plant because they have to shrink and swell all the time. Yeah.
Thanks.
All right,
All right,
let's
thank
thank
you.
Professor Milano again. All right, so we've got a break scheduled for the next 15 minutes or so. So you're all welcome to move about until then, but be back here for our next speaker at 3 p.m. All right, anyway, for our next speaker, we'll welcome Professor Marco Frice. He's an associate professor and Rosalind Franklin Kennedy Trust Research Fellow at the Kennedy Institute of Rheumatology at the University of Oxford. And he'll be talking about the mechanobiological control of T cell activation.
Yeah, thank you very much. We were just talking with Claire that we don't have enough mechanobiology symposia in Europe, so I'm very excited to be here. Thank you. So I'm based now between the Rosalind Franklin Institute and the Kennedy Institute. The Rosalind Franklin Institute is the new National Institute for Biophysics in the UK, and the Kennedy Institute is in the heart of the medical science division in Oxford. And I guess what the COVID-19 pandemic has all reminded us of is that the immune system must really be understood in its entirety. And I mean, from the organism scale, the tissue scale, to the cell, and ultimately to single receptors, as we just have heard in the previous talk, when a TCR engages, for instance, with an MHC complex. But in addition to the length scales, there's an associated time scale. And most importantly, there's also a force scale. And I can't stress this enough that in contrast to all other physical forces, mechanical forces are really abundant across all the scales. For instance, T cells experience very different mechanical settings when they interact, for instance, with healthy tissue or when they're interacting with a cancer tumor. And I thought to start by sharing some data from colleague Tal Anon, who is a professor in the Kennedy Institute. And they're really world experts in imaging the interactions of T cells and B cells in the living organisms, like here in the mouse, deep within the spleen. And I find these images absolutely breathtaking. If you look very carefully, for instance, at how the T cells, here shown in red, interacting with B cells in a living organism. But as a microscopist, if you look very carefully at, for instance, the T cells, you can observe that they're jumping to the field of view and even vanishing from the focal plane, suggesting perhaps that the temporal resolution isn't sufficient. Or if you more carefully look at the B cell classes, they're suddenly breaking up into individual cells, suggesting that perhaps the spatial resolution isn't sufficient. And we believe that light sheet microscopy will transform the way how we can understand these processes in 2D and in 3D. And this shows data from our close collaborator, Eric Betzig, who's one of the Nobel laureates in 2014 for a super resolution. And here, in combination with adaptive optics, we can really see now at unprecedented resolution how lymphocytes deep in tissue interacts with its environment. But on the other hand, the resolution is also sufficient to very carefully study the interactions between the T cell, as you can see on the right-hand side shown in red, with an antigen-presenting cell. And as a lab, we are very interested in understanding the processes that lead to the immunological silence, which is one of the most important steps during the adaptive immune response. So we now know that the T cell receptor that is expressed in the membrane of the T cell, as you've heard before, and there goes a sequence of events, so forms it from nanoclusters and microclusters, and then it moves in a directed manner from the periphery of the interface into the center, ultimately forming the supramolecular activation cluster. And this is a consequence of TCR triggering, when it's thought that possibly a single TCR PMHC interaction could induce the formation of the image synapse. And this has been originally shown by Michael Dustin, who kind of discovered the image synapse, was a scientific director at the Kennedy Institute. And here they used turf microscopy to image how single TCRs move at the periphery towards the center, as highlighted before. And with lattice light sheet microscopy, we can now actually study these processes in three-dimensional space. And what we can also do is we have built together with Eric Wetzig the TurfSyn microscope, which allows us now really at unprecedented resolution to visualize the actin structures at the interface when the image synapse forms. And I would like to highlight here, it's really like the combination of high spatial and high temporal resolution. If you would image at lower resolution, you would average over some of the features. And you can really appreciate how individual fibers at the interface between the lamellipodium to the lamellum are organized in this quite mechanically complex processes. And this is important because these are processes are ultimately responsible for organizing the signaling molecule at the Nanoscale synapse. And so I thought for today, I'd tell you a story about our work of how we believe that mechanics underpins the formation of the Nanoscale synapse. And then I will try to also introduce a tool that we have developed that might be helpful for your own research. So before I start, I would like to define what I mean in the following with resting and activated cells. So when we originally got engaged in the question, the common picture was that I want to expose our T cells to a less inert surface. So typically the glass surface were coated with polylysin. But in fact, what we find is that polylysin is highly charged. And in single molecule imaging, as you can see on the left-hand side, you see that the TCR is actually highly clustered compared, for instance, when you expose and activate cells on agarose. And the same you find in supervision STAT microscopy, the T cell is strongly activating. So we came up with this hydrogel technology where we basically hold the T cell in suspension. And you can, with that, achieve a phenotype similar to what you see in cell culture. They're basically in 3D, as you see on the slide image, and constantly generating these actin ruffles. So in the following, when I refer to activating cells, I mean those cells who are exposed to some kind of surface. It might be an antibody or an activating peptide, or those cells who are resting in the gel. And as you know, there's a myriad of methods that are now available to characterize the dynamics of molecules and cells, but there are very few techniques that are available to systematically characterize the forces at these very sensitive scales, as it has been shown before. And our methods of choice, or one of the methods of choice in the lab, is traction force microscopy, and the goal is to measure the forces at single receptor sensitivity on the cellular cell and on the monolayer scale, and ultimately on the tissue scale. And as it has been introduced before, just briefly, the idea is you have elastic substrate, you have fiducial marker beads underneath the cell. And when the cell now exposes forces, it displaces the beads that can be imaged. And because we have knowledge of the mechanical properties of the gel, we can solve the inverse problem and calculate the forces that the cells are generating. And traditionally, this has been performed with diffraction limited confocal or wide field microscopy. And because it's diffraction limited, it minimizes the resolution, so the density of molecules that you can suspend within the gel. But then what you can do is, for example, we combined it with STAT microscopy, and with that, because the increased localization accuracy, you can increase the density of beads beneath the gel, with that increase the sensitivity. So over the last couple of years, we have just done now 3D structural animation microscopy. And last year, we published 2 papers we combined with TurfSim microscopy, as I showed you in the beginning, and with astigmatism. And this has really transformed the sensitivity of the method. So if you compare, for instance, here with confocal microscopy, we could improve it up to 100-fold in the temporal scale. And if you compare, for instance, the axial resolution in diffraction limited mode, it's about 800 nanometers, but we could bring it down to 10 or 20 nanometer sensitivity. So we can basically measure now in single TCRs binding MHC complex on the scale of the whole cell. You may ask, how can you form a turf field on top of a gel? So this can be achieved by basically matching the reflective index of your gel with the glass. And with that, you can basically extend the turf field to the top layer of the gel at the interface with the fluid. And in fact, you don't need to do any changes to the optics. You can basically just match reflective indices. And just to show you a little bit of data, on the left-hand side, you see the super-resolved T cell in TurfSim modality and the beads. And on the right-hand side, you see the corresponding diffraction-limited wide field modality. So you can appreciate already by eye the significantly increased sensitivity of the method. In terms of astigmatism, you actually need to introduce a cylindrical lens in the optical light path. And then you can very carefully basically calibrate the reshaping of the points of reflection depending on where you are in the focal plane, and you can calibrate that. And effectively, you can achieve up to 10 nanometer sensitivity. So this enables us now, the movies are screwed up, this enables us now on the whole level of a cell in 2D and in 3D, calculate the forces underneath the cell and laterally and axially when the cell is activated. So the other advantage of this method is that we can mimic the surface of the antigen-presenting cell. So we can step by step reconstitute the surface of the ligands that are presented by the antigen-presenting cell. And in the following, I will show you data in the absence of any kind of integrants because we are interested in the forces that are generated only due to the TCR. And we will do this with a transgenic TCR system, with a 1G4 TCR system, or with an activating antibody. And in both cases, you can see on the top layer that the cells are reliably spreading and activating. We see this very characteristic load fail events underneath the lamellipodium. So whenever a TCR cluster binds, it gets displaced and then goes elastically back to its original position. But we don't see these forces in the center where the Mischke synapse forms. And you can convince yourself that these forces are indeed generated by actin retrograde flow. So if you block actin retrograde flow with just plequinonide, you basically just go back to noise. And so this method enables us now to understand the relationship of actin retrograde flow in the lamellipodium, as you saw in the TurfSim data, and the TCR coupling when it binds to a peptide or an antibody on the single receptor level. And we can actually change the electrostatic force between the TCR and MHC complex by exchanging the peptide that is presented to the cell. And we performed these experiments now for 4 different peptides. So for a weakly binding peptide 4D, a strongly binding peptide 9V, and the most commonly used antibodies OKT3 and UCHD1 to activate T cells. And we made a very surprising observation, which we didn't understand in the beginning. In fact, we found that the peak force was the same across all the conditions. So if we expected that if you have a weakly binding peptide, so the force is weak. If you have a strongly binding peptide, then the force is strong. So we really didn't understand that in the beginning. So we came up with 2 assays. Once we just went back to classical confocal microscopy, and we just photo bleached the lamellipodium, basically marked it, and we tracked the photo bleach spot throughout the cell and calculated the actin retrograde flow. But we also went back to our super TurfSim image to use it in optical flow analysis and can basically track the turnover dynamics. And what we found in both conditions consistently that the velocity adapted to the peptide affinity. So for weakly binding peptide 4D, you had the highest velocity, and for strongly binding peptide 9V, you had a slower velocity. So what does it mean? So that means that it's not like the classical perspective that TCR signaling basically induces cytoskeleton rearrangement, but possibly there's mechanical feedback between TCR signaling and antigen recognition. So we wanted to understand better how the TCR is coupled to the membrane, to the cortex. So we set up a dual-color set super resolution imaging, and just in panel C, you can see that in both channels, we get about 50 nanometers resolution. And now we can basically measure the density of the actin network at the membrane. And in both cases, and we found a bimodal distribution. So in some areas, it was very close to the membrane. In other areas, it was further away. We could actually measure the density. In some areas, we found that the distribution was almost like exponentially distributed, as you can see in B. And in other areas, it had a clear Gaussian peak further away from the membrane, suggesting that in some areas of the cell, the actin could be very close. In other areas, it could be very, very much away from the membrane. So we wanted to understand that further and wanted to push these technologies further. And the next question that we asked him, so what does it mean in terms of turnover dynamics? What type of filaments do we have in the actin cortex underneath the membrane? And for that, we turn to fluorescence recovery after photobleaching experiments. You probably all have heard about it, but just briefly, you label your molecule of interest, and then you locally photobleach it. And because of the intrinsic dynamics of your molecule of interest, you see a recovery. Then you can apply a mathematical model of it and can make conclusions about the underlying dynamics. So I present this data in this logarithmic acceleration plots, which basically shows you then, this is just the second derivative. So the first one just shows you the number of processes and the relative abundance. And what we found in T cells was that one process, you can see here in red, was dominant on short time scales, and one dominant process was dominant on long time scales, suggesting that there are 2 types of filament populations in the actin cortex. So what we did, we combined this now with cytoskeleton perturbations. So like cytokinesin D, for instance, specifically targets the barbed ends of actin filaments, for instance, when you have ARPT3 mediated filaments. And indeed, when we change the concentration, as you can see on the left-hand side, you can actually control the turnover dynamics, suggesting that those filaments who are short and dominant on short time scales are actually turnover faster. And then the only nucleate that is actually known that can compete in terms of KD for binding of barbed ends with cytokeratin D are formines. And indeed, when you use a broad-spectrum formine inhibitor, you can control the second slope. So this suggested now with this type of simple experiments that there are two types of filaments in the actin cortex, short ARPTA3 mediated and long formine mediated filaments. And we have shown this now in different cells. It seems to be like a shared structure across many different cells, just the relative abundance and the filament length and turnover dynamic change. So how can we push that further and actually can measure the filament length? And I don't have time to go into the mathematics, but what we did is we combined fluorescence recovery photo bleaching with single molecule imaging. And then we came up with a framework to derive basically the filament length distribution of the different filament types. So what we found is that if you compare resting cells with increasing activation strength from the left to the right, that the ARPTA3 filaments became actually shorter, and that the formine-mediated filaments became longer. And this was particularly true at the immunological synapse. And this is really exciting because it's consistent with what I showed you before, that the turnover velocity changed. Right? So in fact, what happened, or depending on the peptide affinity, the filament length changes. It basically changes its structure in response to the affinity of the peptide itself. So what are the consequences of that? One of the consequences is that the mechanics of the cell changes. So you can do computer simulations where you're comparing short filaments as shown here on the y-axis compared to long filaments, as you can see on the x-axis. And if you make effectively longer filaments, that means you move from the blue into the red regions from the left to the right. So what the T-cell is effectively achieving by adapting its filament length is makes a stiffer and flatter interface, basically mediate the organization of the signaling molecules at the interface of the synapse. And this is very exciting because what we could show in another study is that dendritic cells, or the antigen-presenting cells, they are forming actually protosomes in the periphery of the immunological synapse. So protosomes are spring-like structures that are constantly measuring their mechanical properties. And this really shows now a very quantitative picture of this type of imaging that I showed you in the beginning that we can do with lens light sheet microscopy. This basically means now that the antigen-presenting cell is constantly measuring the mechanical properties of the T cells. And the T cells, once it engages with the peptide, it basically changes on the nanoscale structure its organization to basically mediate interpretation of the antigen itself. So this is super exciting. And how can we move that forward and what can we learn from that? Ultimately, we want to understand how naive cells are maturing into effector and effector memory cells. So what we do more recently is we moved away from these flat gels, and we're making now these spherical mechanosensors. And as you can appreciate in the movie, it's like they basically have the same mechanical properties as the flat gels, but we have fiducial marker beads as we have them in the flat gels suspended within the 3D structure. And we can run this either with, as you can see here, appreciate on the top, we can just image it with a light sheet microscopy, but we also can run it as a high throughput kind of assay in low magnification. And what we do now more recently, we're actually using this as an activation platform to compete with Tenure or Dyna beads that are broadly used in almost every lab in clinical studies. And what we find is that by tuning the mechanical properties of these beads and the nature of the interaction with beads of the cell, we can actually change the proliferation of the rate of the cells, but we can also control what type of cells we are generating, right? Is it as memory cells or is it as an effector cell? And this is super exciting, obviously, for CAR T-cell therapy in the direction that we're moving at the moment. What is also very exciting is that we actually want to understand the process, the underlying mechanism when the cell engages initially with the target and all the way becoming a memory cell. So what are actually the processes? Does it make a difference when I engage 10 times the same peptide or different peptides? And for that, we developed now a new toolkit that I just very briefly wanted to introduce. And the challenge is really that we know that the immune response system is anything else than linear. It's highly nonlinear. And there is the smallest number of cells in the ensemble, for instance, just within T cells, that are playing important roles, like, for instance, Treg cells or memory cells. And one of the question is, or one of the standard method that has moved in the field is flow cytometry. And as we know, we have a large ensemble of cells. We can present this ensemble as just a histogram. And this is usually characterized ideally by a single characteristic value. And when we see changes in these distributions, possibly immeasurable, it has huge effects on rare events, right? You shift the cluster gradient a little bit, but what in the tail, it has a huge effect. So how can we actually develop methods that can pick up these changes, for instance, in the memory population? So what we came up with is a new type of light sheet. So with a little bit of a trick, we are forming a super extended light sheet that is 5 × 5 millimeter, but it's only 25 micrometer thick, so a single cell layer thick. And I don't have time to show you all of the details, but the power of the system is that we have 2 orders of magnitude less photobleaching. So we can comfortably do 10,000 to 100,000 images without significant photobleaching. And this just to give you the idea, in the middle, you see the field of view of a classic confocal microscope, when you have like a 10x kind of objective. This is our field of view. And if you think about rare events now, probably has a single event in your normal microscope, which you just wouldn't take it serious. But in our kind of setup, you would have now hundreds of these events, right? And you can follow these cells over extended periods of time. Just to give an idea here on the right-hand side, it's a very simple setup. You have the fiber coming in, you have then a cylindrical lens and an objective that forms the light sheet. And then I don't really have time for going into the details, but we also have a microfluidic device that allows us to image up to a week and constantly basically following these cells over time. This is the kind of quality of data that you can see on the left-hand side, which is competing with confocal microscopy, so we're getting really high contrast. But we can also expand this now, for instance, to cytotoxicity. So we can basically get very reliably image now how 100,000 cells interacting with targets and follow basically through all the way when they become a memory cell. We have developed algorithms that basically automatically segment the cells and follow them over periods of time. And we have also algorithms that basically can quantify the length of interaction between the target cells and the T cells. And we're observing some really interesting processes. For instance, the T cell comes in the very beginning, touches the cell, walks away, touches many other cells, comes back to the original, and then kills. Right? So there's like this really unexpected processes you can see in the imaging. We have also built a tool that we're preparing at the moment. So basically GUI that has all these software segmentations and classifications pre-implemented, so you can basically just load your data. And and if you get automatically all the analysis. And I'm already running out of time, and I'm moving then on and say thank you for my team and all our collaborators across the world. Thank you very much.
All right, questions?
Maybe I can start. Amazing talk. So, so have you measured the forces that things like proteasomes and invadopodia exert? Because you have a really cool attraction force system that's pretty sensitive.
Yeah, so most of our work has been on the T cell side, except the part where we discovered, I guess, the proteasomes. But I guess, yeah, these are the kind of questions that we want to ask now. And ideally, I guess, you're totally right. Ideally, we want to measure the forces in both cells, and not only the cell, I guess, on the surface. I guess one of the challenges that there are almost no tool to actually measure forces within cells. Or that I guess the tools that we have are Fred kind of type sensors, but they only give you the amplitude, right? They don't give you any directionality. So there's a lack of tools.
And the K-offs that you showed in one of the slides, those were in the tens of seconds range. Is that right? The K-off, the off rates.
Yeah. So the 9V peptide is about, I think, 3 seconds, so 0.33. And
so that means it should be quite sensitive to viscoelasticity, right? Because a lot of the materials, they relax on tens, of scale of seconds.
Yes, so this is a really, really good question that I worry a lot about. Maybe for everyone else is that cells have elastic regime and a viscoelastic regime. And I guess the elastic regime is time independent. Basically, if you displace the cell, you basically just see a linear response. But in the viscoelastic, there's a characteristic time scale associated to it. And if you put force on a viscoelastic substrate, it basically just dissipates. And I find this very interesting question because I think there was just a paper last year in Nature Physics from Joseph Case, and they did viscoelastic measurements of tumors. And what they find is that the breast cancer tumor actually downregulates its fluidity. So it actually suppresses its viscoelastic properties, but it's then only an elastic regime. And this is very interesting because CTLs are quite weak immune cells, for instance, compared to B cells. So you can think about if T cells want to invade a tumor, usually it's a very good strategy to just use the viscoelastic properties of the tissue, because if you push hard enough, you can just crawl through because everything just relaxes, right? But the problem if you have a purely elastic system, if you make it stiff enough, you just can't enter anymore because it's just too hard.
I was really interested in your FRAP on the surfaces of these cells. How much mobility is there on the surface of the cell, and how important is that to function?
I unfortunately can't go backwards, so I'm moving forward. I think mobility is super important, and the movie that I didn't show you, but maybe I have the second now, I can do that. So what we're doing now is we're moving everything to 3D and tissues. And one of the efforts that we have been doing is combining light sheet microscopy with fluorescence recovery after photobleaching. So now you can actually image now. You can do dynamic measurements in 3D. So you can appreciate, for instance, here on the top that rather than having a 2D FRAP region, you have a 3D, and you can basically very nicely appreciate how it recovers. And just to give you an idea, here on the left-hand side, we basically we're tracking features of the membrane and of the cortex. And we here, for instance, discovered, if you thought about it, the classical picture is actin-reticular flow goes at the interface towards the center to push the signaling molecules. But in fact, not only at the basal plane, but also as apical plane. If you think about it, it makes sense, right? The whole lamellary protein is floating. But what this data show you is it's actually floating all the way up. Right, so all the way up to the cell. So that now I don't understand anymore how TCRs are actually transported to the interface, because everything is transported to the apical phase of the cell, right? And what's happening there? It's just like interchanged and it's reorganizing. So yeah, so it's highly dynamic in the flow dimension, the diffusion dynamics, and obviously also in the binding and unbinding kind of space.
So when FRAP was invented, this was in the laboratory of Elliot Elson and Watt Webb. They actually had the exact opposite problem. They were trying to get to the surface, and they'd already invented fluorescence correlation spectroscopy to actually get at the inside and figure out what was happening on the inside. Can you do FCS at this length scale inside the cell and actually get the fusion constants and diffusion directions and answer that question? Is that possible?
We work on that. Yeah, we have a couple of work in that direction where you can get diffusion, subdiffusion. We even have a paper now we can get oligomerization dynamics from FCS. But there's basically, I guess, one limitation of that is that you need to scan, right? Or
you could do it at focal volumes.
So. Yeah. But then you only have to. But I mean, ideally, in the light sheet world, I want to have 3D, 4D kind of surface, right? Where I get context from the cell. This is the beauty of this kind of approach, right? You can track your molecule, but you have context of the cell topology. In spectroscopy and FCS, you basically have your volume again. You don't have the context anymore. So it's something that we're doing now. We're actually using the lattice light sheet to doing FCS. So you basically can cross-correlate the whole volume rather than the PSF.
Very cool. Yeah. Thank you.
Any other questions? Yeah, for one or two more.
Do you see any? Very nice talk. Do you see any motors or stepping or anything interesting like that connecting to the T cell receptor?
So we haven't looked at that specifically. we have started trying to understand what actually is responsible for the termination of the synapse. Is it a purely biophysical mechanism or is it a signaling response? And we have a theoretical model that suggests that probably just the anisotropy in the motors is basically determining the characteristic kinetics of the termination of the synapse. Yeah. So it basically, the anisotropy in the actin network is responsible of contracting the actin network. And something that you saw in the turf sim images is you have these dark areas. You have this very beautiful flowing actin and you have this dark area. So these are actually tangential waves that move along the lamellipodium because the actin mass is so compressed that it starts moving in 3D. And this is probably the areas where you form your TCR clusters that are transported then to the center. And then compressing your actin until a critical situation, and then you probably can detach from the actin network. How they're directly communicating with the TCR, we haven't worked on that.
All right, if there's no other questions at this point, let's thank our speaker again. Right, next up, we have our final speaker for this session, Karen Shu, who is a PhD student here at the University of Pennsylvania in the Burdick and Mock labs. And she'll be talking to us today about microphase separation and viscoelasticity and how they mediate meniscus cell migration in hyaluronic acid hydrogels.
Thank you. Okay, awesome. So we all know that hydrogels have been used extensively for mechanobiologic studies. For example, polyacrylamide is a very well-known single network that can be used to tune the stiffness of an extracellular environment. If we add a second network through perhaps putting a polymer within a cross-linked first network, we can create semi-IPNs. If we actually take that polymer, that second network, and start to cross-link that within the first network, that creates an IPN. And by adding multiple different polymers together, we have the opportunity to introduce chemical versatility into our systems, which can be really important when we're trying to mimic complex environments for cell material studies. Some examples of IPNs that have been used in the past in order to explore cell material interactions include this study that was performed on the left, where they take gelma and hyaluronic acid. They create a dynamic hydrogel and explore how this material can be used for skin tissue engineering purposes. On the right here is probably a diagram that some of you are very familiar with that consists of an IPN that has alginate and reconstituted basement membrane. This first study was used to explore how independently tuning ECM stiffness and ligand density influenced the transition to a malignant phenotype. But of course, this material has been used extensively since then in order to continue other cell material studies. The material that I'm using is an IPN that consists of a covalent and a physical network. The covalent network is a gelatin-based material that is cross-linked with transglutaminase, which essentially introduces a temporal covalent cross-linking mechanism. We also introduce a physical network that consists of adamantane and cyclodextrin, both independently conjugated onto a hyaluronic acid backbone. The adamantane and the cyclodextrin form an extremely strong physical interaction, and I will refer to them as the guest host network from here on. So we were particularly interested in how the physical network changed our material properties as well as how the cells interacted with our material. So we take these experimental groups, the first one being 0% physical network, i.e. just our gelatin covalent network, and then a 1% and 3% physical network within our IPN, where I have a consistent amount of my gelatin covalent network throughout all of these materials. We first wanted to explore how the macroscale rheologic properties of our material change with altering the amount of our physical network. And so we did some studies here where we can see that the stiffness of our material is maintained across these different physical network concentrations, but that the tan delta, which is a measure of energy dissipation, increases with increasing guest host concentration. Tan delta is also used as a marker of viscoelasticity, which we've heard that word tossed around a lot today. But this essentially tells us that by changing the amount of our guest host network in our IPN, we can change the viscoelasticity of our material. Recognizing that our IPN consists of multiple different networks, we wanted to explore how these different networks were interacting with each other. And so then we labeled our material, specifically our covalent gelatin material, fluorescently. And we can see here that, as expected in our control case, the gelatin is seen homogeneously throughout our material. But when we add our physical network with the 1% and 3% cases, we can see that our material starts to phase separate into what I'll call the gelatin-rich domains that are labeled in green, and the gelatin-poor domains which don't have that label. We think that this phase separation might be occurring because we're mixing two relatively viscous solutions together, and as soon as the guest and the host find each other, it kind of stops the ability of the network to continue mixing. So in order to try to understand this further, we wanted to quantify the structural heterogeneity that we were seeing. So we took these corresponding fluorescent intensity profiles, and we looked at the coefficient of variation across these different profiles. We can see here that the presence of our guest host leads to increased structural heterogeneity in our IPN. We then wanted to understand if our material was also mechanically heterogeneous, and so we performed atomic force microscopy of 100 by 100 micron force maps. We can see here again that the heterogeneity is increased in this particular spatial region for our materials with the presence of our guest host network. So recognizing that we have understood now the structural and mechanical heterogeneities that were occurring at this micron scale level, we wanted to explore how this influenced cell migration. I am a member of the orthopedic research department here, and so we are particularly interested in studying dense connected tissues. And so my organ/tissue of interest is the meniscus. We took meniscus cells, formed spheroids, and we embedded them within our different IPNs. What you'll see here in this time-lapse image, or time-lapse video, is that the cells are in blue. Again, the gelatin is labeled in green, and we're seeing that the cells crawl out rapidly in our material with higher guest host concentration. What's particularly interesting about these videos and being able to label the gelatin-rich component is that we're seeing that the cells like to crawl along the domains that are gelatin-rich, but they never actually go within those regions. This suggests to us that the heterogeneities that we are creating microinterfaces for these cells to crawl along. So we wanted to quantify this. We looked at our cell outgrowth in all of our different experimental groups on 1, 2, and 3 days. We can see that across this time span, there really isn't that much outgrowth in our gelatin-only case, but that there is pretty extensive rapid migration when we add the guest host. We were also curious to see how cells deposited matrix, so we used non-canonical amino acid labeling in order to look at nascent protein deposition. We can see that as our cells are crawling into the extracellular environments, they're depositing matrix. We then wanted to explore if the cell migration trends that we were seeing in vitro were also seen ex vivo, so we took meniscus explants and we placed them on top of our different experimental groups. You can see here with these 3D reconstructions that in our 0% case, when the cells crawl out of the explant, they stay along the surface, the interface of the material, but they don't crawl through the hydrogel. Whereas when we have our physical network present, they are found within the hydrogel itself. And we can see this quantitatively when we look at the maximal cell infiltration as well as the depth where we see our cells. We also wanted to explore this in vivo. So we took meniscus explants. We then injected our different experimental groups into a defect and then placed them, we implanted them subcutaneously in the back of rats. We can see here in my preliminary data that there's a pretty high density of cells found within our experimental groups with higher guest host concentrations. We can again bin the distance within the defect space, and you can see here that as we saw in our previous studies that there is an increased cell infiltration found with our materials that have the higher guest host concentrations. So we were excited to see that we could induce rapid cell migration in vitro, ex vivo, and in vivo, but we really wanted to understand what features of the cell material interaction were promoting this rapid cell migration. The first question we had was, well, if our covalent network consists of gelatin, which can be degraded, how much of a role is that degradation playing in the cell migration trends that we're seeing? So we took a broad spectrum matrix metalloproteinase inhibitor and we treated all of our experimental groups with this. And we can see that over 3 days, in our 3% group in particular, that there's a decrease in the amount of cell migration that we're seeing. But it isn't as much as the 0% case. So this really suggests to us that while degradation plays some sort of role in the trends that we're seeing here with migration, it's not all attributable to the degradation. We then wanted to explore the role of integrin-based adhesion in the migration trends that we were seeing. So we took specifically our 3% group, and we treated with soluble RGD. We can see here that the cell outgrowth is significantly decreased, suggesting that, in fact, integrin-based adhesion is important for the migration that we're seeing here. So recognizing that I showed you, and I'm trying to convince you that these microinterfaces are important for the rapid cell migration, we wanted to look at, well, what features of this interface are important for what we're seeing here. We decided to create the simplest sort of interface that we could, which was a layer-by-layer approach, where we took gelatin, created a substrate for our spheroids, we put the spheroids on top, and then we put some sort of other material on top, essentially creating an artificial interface. So when we just put our gelatin and our spheroids on top, we see that in one day, the cells are crawling out very rapidly onto this gelatin interface. We can see when we put gelatin on top that there isn't cell outgrowth. I'll point out here that this gelatin, once we put that on top of our spheroids, it's capable of cross-linking with that first gelatin. So essentially, in this particular group, we're creating a fully encapsulated spheroid that is akin to our 0% experimental group that I showed you previously. We then took our physical network and put that on top. We see that our cells are crawling out pretty rapidly throughout. And then we were interested to see, well, our guest host does consist of a hyaluronic acid backbone and the guest host chemistries. What if we just put an inert, non-adhesive material on top? We took different whey percents of agarose. We placed them on top. We still saw that the cells were crawling out. This is preliminary data, but this really starts to suggest to us that the mere presence of an interface is enough to be able to promote the cell migration that we were seeing. So recognizing that I've shown you, so I guess you guys are getting to see the results already, but recognizing that I've shown you that my material is both viscoelastic and has these microphase separations, we were really curious to understand which of them might be playing a bigger role in the trends that we were seeing regarding the cell migration. So we created these new materials. This first one is what I call a uniform material. It consists of every single thing that was in my 3% group, but this time the guest host moieties are soluble factors, which means that we can mix our material completely before we conjugate the soluble factors to the HA polymer backbone. This essentially means that our material is homogeneous and viscoelastic because we have the guest host moiety still present. And when we put our spheroids within these gels, we see that they're not infiltrating into their extracellular environments, which is my uniform material case right here. When we try to look towards developing a material that is minimally viscoelastic, but potentially heterogeneous, we decided to go for a gelatin-based material. So this is essentially our control 0% case, where we've also added in these fragmented agarose particles. What we can see here is that our material is heterogeneous based off of these particles, and that when we put spheroids in, that they infiltrate within this material, despite the fact that it's minimally viscoelastic. So over here with my quantitative data, you can see that the 0% and 3% data is what I've shown you previously. The uniform material represents a homogeneous viscoelastic material, and our agarose interfaces material represents a minimally viscoelastic but heterogeneous material. This really suggests to us that the microinterfaces that we're seeing here are necessary and sufficient for my particular system to support cell outgrowth. So hopefully I've shown you that the IPNs that we have created are both viscoelastic and have microphase separations, but that we are most interested in the microphase separations as a means to create microinterfaces to support rapid cell migration. This is really exciting for us because we think that the identification of 3D interfaces represents a potential new biophysical cue that can be engineered into tissue-engineered constructs when we try to repair defects in dense connective tissues such as the meniscus. And with that, I'd like to thank my labs and our collaborating labs, and specifically I wanted to thank Brendan Steckel and the Plushko labs. They have done incredible work on this project that unfortunately didn't make it into this PowerPoint. I'd also like to thank our funding sources, and my poster is 26, so if I don't get to specific questions, I'm happy to talk about them later.
Thank you. Questions from the audience?
Hey, Karen, that was really great. I was wondering if, as you watch cells crawl between these interfaces, do they leave openings? And do cells kind of then come follow behind? Does it leave sort of open tracks and highways so that one cell's generating a bunch of energy and then it's much easier for more cells to migrate?
That's a really good question. So we are starting to do studies where we're labeling all components of the material, which will give us the opportunity to look at paths that are created. That being said, I think that the guest host network, the gelatin pore network, is very malleable. And so I think that if a cell were to crawl through, the material would end up flowing back into place. But we are planning on doing those studies, so stay tuned.
Becky, go ahead.
Karen, really nice work. What is the length scale of these interfaces? Have you tried, going up and down to see what the limits are?
Yeah, so when I measure the heterogeneities, they're on the scale of 5 to 20 microns, which is very helpful for, it's on the same scale as cells. In terms of trying to tune the heterogeneity, what I have found is that my 1% case has qualitatively bigger gelatin-rich domains, which I think could be playing a role as to why we're seeing increased migration in our 3% case versus our 1% case, but definitely not something that we have quantitatively looked at yet, but we can, and so we probably will.
Vivek, you had a question?
Yeah, so following up on Becky's question, in your images, what is the size scale of these domains?
Yeah, 5 to 20 microns. That's what I was referring to.
But then you had a case without the size scale, real interfaces, right, between two.
Yeah, exactly.
So why are the cells? What do you think the cells are going along the interface as opposed to, inward?
My prediction that I have not necessarily I haven't started doing experiments for. I think that the interfaces present a path to these cells, and I think that for the cells, it's easier to crawl through that path and sort of maybe degrade a little bit and crawl through an interface that already exists as opposed to trying to degrade its way through. I'm sure that we would see degradation if we looked at longer timescales, and my in vivo data actually supports that so far, but because we're looking at very, very short timescales, that path is just easier for the cells to crawl to, I think.
Are the cells any different after they start moving along these paths? Do you have any way of knowing this? So if you were to somehow stop one of these cells, would it just keep barreling on through, or would it change its mind? Do you see special persistence out of any cells, or once they start moving?
I think that the time-lapse videos would suggest that they just keep moving. They just, once they're out of the spheroid, they just keep moving around. I have started doing some studies on exploring genetic expression of the different cells within the spheroids versus when they leave, and I can tell you that data that I saw yesterday suggests that potentially cells that have crawled out of the spheroids like to express collagen 1 to a greater extent than cells that are within the spheroids, which suggests that there might be some sort of differential genetic regulation that is occurring once they have migrated from the spheroid. Does that answer your question? Okay.
Great talk. And I might have missed this, but I'm curious to know if you can comment on the porosities. So could the gels just be more porous, and the cells taking advantage of that?
Yeah, that's a good question. So going back to me starting to label the different components, I have demonstrated through labeling the guest host hyaluronic acid-based components, which I didn't show in this presentation, that it is full of material throughout the entire three-dimensional bulk hydrogel. So it's possible that the cells are pushing material throughout, but I don't think that there is naturally a presence of pores that the cells are crawling through.
Okay, so you don't think that at the interfaces
I don't think there are pores.
Okay. And then also, I know this will not be an IPN, but I'm curious to know what happens if you take these different, I guess, materials and then cross-link those together. Do you expect them to behave more like the gelatin uniform system?
You're saying if I were to cross-link the hyaluronic acid to the gelatin?
Correct.
I think that it would behave more like the uniform system. I think that the presence of these micro interfaces are creating these paths, and I think that if I were to cross-link them together, I would be, even though the phase separation might still be occurring, I think that I would be cross-linking and sort of blocking these paths for the cells to crawl out. That would be my prediction if I were to do that study.
Thanks.
All right, if there's no other questions, let's thank Karen again. And thanks again to all our speakers for a wonderful session. That concludes our presentation sessions for today. The poster session will start right outside here in a couple minutes at 4, and we'll also have a reception, so please join us out there.
All right, good morning and welcome back. So it was a great session yesterday. So we covered a lot of ground, molecules to cells to tissue, and also a number of different tools. So I want to start today by thanking the team that put the symposium together and identified speakers. And so they did a really fantastic job. So thanks to them. And also to our staff, Annie, as a speaker's note, she's been in touch with you, really taking good care of you. So as well as Abri. So with that, look forward to today's session. So Eileen Zhang, a graduate student, in Suchin's lab, will moderate this session.
Good morning again, welcome. It is my honor to introduce our first speaker today, Dr. Kate Miroshnikova. Dr. Miroshnikova is a Statman Tenure-Track investigator and acting section chief of nuclear mechanotransduction and cell fate dynamics in the Laboratory of Molecular Biology at the National Institute of Diabetes and Digestive and Kidney Diseases for the NIH. The title for her presentation is Nuclear Mechanotransduction and Stem Cell Fate Regulation. Please.
Hi, thank you so much. Let me see if I can share my screen. All right. And so hide the controls. Okay. You can see my screen, correct?
Yes.
Perfect. Well, anyway, thank you so much. It is really a pleasure to be a part of the symposium. I'm really sorry I couldn't join in person, but anyway, really great to be here. And my long-term interest has been to understand mechanical forces as sources of cellular stress, as well as instructional cues. As probably this does not need introduction to this audience, but tissue cells and organelles undergo a tremendous amount of deformation throughout our everyday life and during development. And in particular, nucleus, which is the largest and stiffest organelle, is also perturbed essentially daily. And really what I'm trying to understand is how does this organelle actually manage mechanical stress? And especially this is important because nuclear structure and nuclear functions are directly linked, as is really clearly indicated in case of cancer, where the nucleus is abnormally shaped, and also genetic diseases such as lipopathies, where destruction of nuclear architecture really has profound effect on the organismal function. And the biological questions that my lab aims to understand is really how do cells integrate information from their biophysical environment as well as biochemical environment to make the right decisions at the right time with respect to their fate. With respect to biochemical inputs, we know that they operate at the level of modulated transcriptional programs that drive cell fate transitions, whether it's or maintenance. But of course, even here, the situation is not so simple in vivo, where cells actually receive a multitude of inputs at the same time, both biochemical and biophysical, and also have to operate at the level of noise. So how do actually cells know what is actually signal and what is noise? And with that, I would like to start to tell you a story of how we think about cellular decision-making, and I will start talking about epidermis, which is the outer layer of the skin, and epidermal stem cells. Now, this layer begins during development with a single layer of stem cells. During development, at some point, by mechanisms that are completely unknown, some cells will undergo a differentiation, and they will move upwards in the layers of the skin. The higher they are, the more differentiated. And essentially, what is very interesting in this process is that size is not determined at birth. So these cells, the pink basal stem cells at the bottom, have to constantly decide during every division whether they actually going to remain as stem cells instead of renew or differentiate and move upwards in the layers. And this is very clear that this information cannot be genetically encoded at birth of the cells, because the tissue is constantly growing to accommodate the growing and changing needs of the organism. And of course, it also is an outermost surface of the organism. And so it has to maintain quite a bit of repair and generation capacity. And so how do actually cells know actually what their fate should be at each division? And so on population level, we know from the labs of Phil Jones, Sabir Blampang, and many others that during development, there's a shift in the balance, total balance, where differentiations are favored during postnatal growth. And in adulthood, situations a bit more balanced, where differentiation and proliferation are really 50-50. Now, so how do cells actually, this is kind of nice to know population dynamics, but how do individual cells at all times know what they need to do? And we got inspired with our collaboration with Cedric Blampang, where he was interested in understanding how do individual clones growing within the skin actually decide the direction of their growth. And what we found in collaboration with Cedric is that actually growth of clones is oriented relative to the local tension generated by collagen fibers underneath this clone. And this really gave us an idea that perhaps local tissue mechanics could be really the cues that direct and specify cell fate behavior. And to try to understand it, we started with a simple model system. We can actually take this skin epidermal stem cells and grow them in vitro. And we can induce the differentiation and stratification simply by adding calcium into the system. And then we actually did exactly just that. And we began to image this epidermal stem cell. And what we noticed that there's, as we began the differentiation process, there's a distinct transition where this tissue turned from a fluid-like state with really coherent motion to a solid-like jammed state. And this was exactly the time point of in this movie, about 24 hours, where the rates of delamination was highly increased. And this was despite constant mitosis rate. And we actually saw that in vivo, the system exists in a completely jammed state. So this gives us an idea that perhaps this lateral compression could be a sufficient cue to really trigger differentiation in the system. And so we designed an experiment where we plated confluent monolayers of epidermal stem cells and we stretch on stretchable membranes, and we stretch the membranes to 150 degrees, 150% of the area. We allowed the cells to form again a monolayer there, and then we abruptly released and released the substrate, therefore generating a transient jamming and transient lateral compression. And then we performed qPCR for really known differentiation genes in the system. And I would like to draw your attention to this red bars in this graph. We were able to show that this lateral compression per se was able to induce this increase in differentiation potential. And again, if we did the same experiment, and this is this blue and gray bars, the monolayers that were not confluent, to really eliminate any buckling effects of the substrate as a cause, we didn't see this. It really was literally lateral compression that was driving this differentiation. And furthermore, if you actually combine differentiation, combine actually lateral compression with cell elongation, the differential is even more potentiated. And we did this by utilizing square versus circular patterns. We know about the virtue of having really stress anisotropy on the squares, you induce this cell shape anisotropy. Now, this was really interesting, and another student in the Wickstrom lab did actually the opposite experiment, where he was not laterally compressing this epidermal stem cells, but he was rather did the opposite experiment of stretching, which stretch is, of course, a really relevant mechanical perturbation in the skin. And what we saw was actually quite remarkable. There was a really global downregulation of transcriptions, and you can see in this RNA sequencing plot. And when we looked at the type of genes that were actually a type of rather programs that are being mediated, and we saw that enrichment or HTK27 silencing mark, and this is a repressive mark on the chromatin deposited by polycomb repressive complex 2. We can really clearly see this transcriptional repression and increase in this repressive mark by immunofluorescence Here at the top, we see the control where stretch increases this mark. In the parallel, there's a decrease in RNA pol 2, the phosphorylated active form. And so where this actually change is happening, we know that actually this is really specific to lineage specification genes. We performed chip qPCR to show this. So essentially, mechanical force in this increase in tension here is really driving global transcriptional repression. We see this happening across the entire genome, yet there's a specific induction of HEK27 silencing mark on differentiation genes. And so collectively, these two studies really demonstrate that mechanical forces really change the way that cells interpret biochemical cues, whereas mechanical confinement and lateral confinement and elongation really triggers differentiation, and the opposite force, where actually the state of high tension triggers the opposite reaction, where there is suppression of differentiation under tension, despite the presence of a really strong biochemical cue. And of course, we know the mechanisms by which, some of the mechanisms by which this is operating. In the case of the stretch, there's actually a formation of a protective ring of actin, perinuclear actin ring around the nucleus, and that depletes G-actin from the nucleus, and G-actin is a critical cofactor for RNA pol 2. Thus, the loss of this cofactor leads to transcriptional repression, and it is really this transcriptional repression that drives the deposition of HEK27 silencing mark. Now, all of these changes that I discussed so far are really happening on the orders of 12 to 24 hours. More recently, we became really interested in understanding really the immediate effects of mechanical forces on the nucleus and cellular state. And to make a long story short, again, this story is published, what we see is actually on the level of minutes, there's a global decrease in another type of heterochromatin mark. It's a really tightly bound peripheral HEK9 trimethyl heterochromatin. Essentially, there's a tremendous downregulation with mechanical perturbation of this particular chromatin, with a few regions in the genome where it's upregulated. And if we look at the regions in the genome where this regulation is occurring, it really seems to be preferentially occurring at the non-coding genes. And indeed, if we take all of the chromosomes and normalize them for their length and just look from beginning to end and look at the where the downregulation is occurring, you can see that the downregulation actually occurring at chromosome ends and subtelomeric region. So this really gave us an idea that this downregulation is not occurring to modify transcription. And indeed, data I'm not showing here, but if we correlate the transcriptional changes with the changes in this HEK9 trimethyl occupancy, there's absolutely no correlation. So what is actually the role of this loss of heterochromatin? Well, the answer is mechanical in nature. What we see is that at the same time, when you lose this heterochromatin, we see the wrinkling of the nuclei. And this coincides with a softening of the chromatin and also a decrease in its mobility. And we measure this by look at mean square displacement over lifetime. So essentially, mechanical forces lead to transient softening, wrinkling of the nucleus, and increase in its mobility. And these are not just things that actually happen; they have really important biological function. If we pretend this softening by overexpressing an enzyme that keeps depositing this marked HEK9, and we stretch these cells, what we see now is that unlike the control conditions where the cells actually cope with mechanical stress and never accumulate any DNA damage, we see that there's an emergence of gamma H2AC and other markers of DNA damage. So again, now the system actually begins to experience mechanical damage, mechanical stress. So our working hypothesis is that this reduced chromatin, this reduced nuclear stiffness and chromatin mobility is really required to protect the genome from mechanical stress under high force. And so what are the mechanisms that actually, by which the nucleus actually sends the deformation? And that this is really the work in the field by us and many others. And we know that it all comes down to the deformation of the nuclear envelope. And the magnitude of the deformation is really determined by the steady state amount of chromatin and lamins in the system. So there's quite a bit of cell type specificity in this response. For instance, cancer cells do not have this mechano response that they trigger. And the downstream effects tend to be mediated by calcium signaling of the entities that are actually residing on the ER, and which is contingent with the outer nuclear membrane. And long term, this chromatin mechano response is generally shut off in physiology, and this kind of force sensing and distribution is taken over by tissue level forces inside a skeleton, which is of course not the case in chronic diseases of the nucleus, let's say such as laminopathies, where the similar phenotype to this mechanical perturbation is really chronic, and that has really detrimental effects on function. Okay, so practically, it is really the formation of the nuclear envelope that really triggers this mechano response. And to kind of go back, so this is now published work. So what are we doing with this knowledge now? Well, my really interest is to understand sulfate transition, understand human development. And so what are the physiological relevant deformations during sulfate transitions that we might want to study? Now, we were inspired by the work of Kevin Charlotte, where he took, where using mouse embryonic stem cells, and he was driving them towards exit from pluripotency. In the case of the mice, they exist in naive state. What he noticed was quite striking, that there is a really, as the cells transition from naive state, they really transition in shape from this ball-like high tan state to a really flattened pancake phenotype. And my interest really is to understand human disease, human development and disease. And so we took human-induced pluripotent stem cells and we differentiate them into either of the three germ layers. And what we saw, there was actually indeed, similarly to the mice, really one unifying feature is there was this flattening that was happening in all of the biochemically differentiated conditions that into all three lineages. And this was really happening exactly as the cells exited pluripotency, in this case prime pluripotency, before they started committed to their terminal states. And so having the deformation in mind, the type of questions that were really open, that remain the key open questions in the field, really trying to understand mechanical forces induced changes theoretic, are they reversible, is there cell type specificity, or they're more global? And something that's also interesting to me is to try to understand the concept of memory. So essentially, if there is this deformation to the nuclei in cells, is there mechanical memory that the cells retain, and does it have any impact on cell fate decisions? And so knowing kind of the questions in mind, and also having this, having the deformation in mind, we utilized a system, a compression system using a negative pressure that originally developed by Mathieu Piel that we adapted. We took iPS colonies and essentially deformed them to the level that we saw during our differentiation experiment. And just to kind of come back to this heterochromatin status, we saw that there was a similar conserved heterochromatin response with nuclear deformation, where we saw a decrease in both the facultative and this constitutive heterochromatin. But we wanted to, of course, understand the system better, and we began imaging, and we were compressing, and something really striking stood out to us. And this was really that there was a transient, we noted a really transient loss in volume during our deformation. And this, of course, gave us immediately a clue to look at hypertonic stress, which is a very similar deformation with respect to actually changing the shape of the nucleus. And of course, it's known that it will drive water out of the nucleus and thereby decrease the volume of the nucleus. But what was really interesting is that this deformation was really happening at differential tension of the nuclear envelope. Compression really made the nucleus really taut, stretched all the ER reserves that we had, and the hypersmortic shock did the opposite, where the nucleus were really much smaller and much more wrinkled. So now this gives us really a beautiful system with which to decompose two aspects of deformation, which is this osmotic effects and this tensional effects. And you can actually see this also better here with the light compression. You can see there's wrinkling at the level of the hypersmortic shock and really stretching in the case of compression. Of course, we can measure this using membrane tension probes, but for the purpose of time, I won't show. And so what we think, and of course, we really wanted to then begin to understand what is actually the difference and what are the similarities between these two systems. We knew from literature that hypersmortic shock drives transcriptional repression, which is really reminiscent of what we have discovered in epidermal stem cells previously. And at the level of transcriptional repression, these two perturbations, hypersmortic shock and this high tension deformation, lead to transcriptional repression. And I think the reason for this is that this deformation drives nuclear volume loss, and this is actually what is driving the repression at the level of transcription. But they're really different. There are really differences at the level of activity. And now I'm just showing you one. We have performed a number of sequencing and analysis, but YAP is one of the, probably needs no introduction here as well, is a mechanosensitive transcription factor, which is generally present in the salt and pepper-like pattern in pluripotent stem cell colonies. And this salt and pepper is actually really important for maintenance of pluripotency in the system and many others, such as intestine. Compression really increases YAP activity and changes the nucleus to cytoplasmic ratio, whereas hyperesthetic shock does completely the opposite. YAP is gone completely from the nucleus. And if you look at by K27 acetylation cut and tags, we look at enhancers of some of the downstream targets of YAP, such as CTGF, we also see the same phenotype. So essentially, there's an upregulation of YAP programs, which are totally lost in hyperesthetic shock. Now, I'm happy to discuss more programs that we noted that are different and similar, but I wanted to come back to this, to the concept of memory and decision making, which I referred to before. We designed a number of experiments, and one of which I really want to share with you today. So essentially took these IPS colonies and we deformed them either using compression or hyperesthetic shock. But we did this so here in two different contexts. One, we performed the experiment in the context of sulfate maintenance, meaning that we compressed them or hyperesthetically shocked them in pluripotency medium. So this is a media that they generally exist in. Or we did the same experiment, but in the context of a transition where we actually switched them to the medium that pushed them biochemically towards the default differentiation state, which is ectodermal fate. And then we came back to these colonies 24 hours later, and in this case, I'm showing RNA sequencing, I think that the data came out really beautiful and informative. And I would like to focus your attention here on the simple PCA plot, principal component analysis plot. All the dots to the left here were performed under sulfate maintenance condition medium, and all the shifted dots to the right are the conditions of differentiation sulfate transition medium. So therefore, this principal component 1 really describes cell trait transition axis. And similarly, these conditions at the bottom are really non-compressed colonies, and all of the 4 conditions at the top are the deformed colonies. So this is really the deformation axis. But what was really interesting and that really emerged from this analysis is that the mechanotransduction that is sensitive to nuclear envelope tension was only evident in the context of the pluripotent media. If we force the cells to essentially biochemically force them to undergo a fate transition, we saw that this nuclear envelope tension sensation was gone. And what dominated was really the osmotic effects. And what was even more interesting is that if we just look at the signature of differentiation, we see that there is an upregulation of transcriptional programs that induce, that involve nuclear volume regulation and osmotic effects. So this really implies that perhaps many, or if not all, sulfate transitions perhaps involve changes in nuclear volume and osmotics. And so I just like to come back to the kind of the context, despite being a mechanosensitive transcription factor. If we now look at this YAP activity in the context on the left is a maintenance, so sulfate maintenance condition, we see this upregulation again, which is gone in hyperesthetic shock. We see absolutely no upregulation of YAP mechanosensitive programs in the context of transition. So that's really saying that deformation really changes in the context of a remodeled genome. And with that, kind of the summary of what I'm kind of showing you is that in the effect of mechanical forces on chromatin, on sulfate transition, and kind of the state of the cells really depends on the epigenetic landscape, where in the case of this sulfate maintenance experiment, we really see that transient mechanoresponse dominates. And it really prevents this transition of cell state. Essentially, mechanical memory and nuclear membrane tension dominate in promoting this pluripotent state, which is totally different in a biochemical differentiation signal, where we see that the osmotic shock dominates, and data have not shown you by chip sequencing. We know that the reason that this biochemical switch is occurring is via mobilization of this PRC2 complex, which deposits HEK27 trimethyl. And so taken together, we think that this deformation really changes interpretation of biochemical signals to alter cellular identity. I'm really interested in building a signature of nuclear volume changes and tension from our RNA sequencing and chip sequencing data to try to understand this in development. And again, there's kind of this temporal evolution of the putting together the whole talk. We think that this immediate effect of mechanical forces has to do with the loss of nuclear volume, which drives chromatin decondensation and transcriptional repression. On a longer term, we see that depending on the biochemical context, there's a domination of either mechanoresponse and activation of key mechanosensitive genes, or really transcriptional repression and enhancement of HK20 reprogramming programs. And of course, this is whenever I present, I always get this whether or not we're interested in more physiological transitions. This is something as an outlook, a work in progress. We're collaborating with clinicians in Helsinki, where they're able to actually grow human embryos and follow their nuclear shapes and their kind of cellular architecture during development from 2 to 4 to 8 state to more relative and hatching blastocyst. And so we're in the process of collecting many of these image data sets, and we are segmenting and really trying to figure out what are the physiologically relevant deformations that we might want to induce and then actually perform and redo our analysis. So again, we are super interested in physiology and hope to apply this to more relevant system. Oh, hi, I am back. I apologize.
Great.
I think I was able to finish my talk and just didn't acknowledge my people, but apologies for that.
Hi, this is Ken Zarrett. Can you hear me?
Yep, can hear you.
Okay, I'm at UPenn. I really enjoyed your talk, and
thank you.
I was waiting till the end when you finally talked about nuclear deformation and continuity with the endoplasmic reticulum, and the ER is perhaps a source for membrane components to come in and out of, or something like that. And it's a really interesting concept. And are there any perturbance of ER structure that might suppress some of the activities that you're seeing?
Right, that's a really great question. And we're actually building tools to try to figure out, we see, of course, a signature of ER stress. So membrane tension is one of the things that we're super interested in, but we also are picking up ER stress signatures, and we're building tools to try to identify architecturally what could be actually happening there. Are we causing any sort of disruption? And also, of course, looking at lipids, right? If we talk about nuclear membrane, kind of the lipid is what actually is giving a structure. And so beautiful questions. We're looking into it, but I don't have much insight to provide.
Yeah, this is Vivek Shenoy. Great talk. So I had a couple of points to clarify. So whether you're compressing the nucleus or hyperosmotic shock, it's going to lose volume. Hyper, it might lose more, but it's losing volume nonetheless. When you push something, there's tension on the membrane, but the volume. So I'm trying to understand sort of this volume loss, and you said volume loss, transcriptional loss, and then there was also membrane components. So I'm trying to clarify.
Yep. So we think that transcriptional repression is actually the common mechanism between these two perturbations, and it drives, this is actually what partially or largely drives the global transcriptional repression. The specific mechanisms that we see are really the mechanosensitive mechanisms, such as MIG program, YAP programs, are really only occurring at the high tension state, but not with hyperosmotic. So we're really trying to decouple the effect of the osmotic shocks, so the volume loss, versus the tension of the nuclear envelope, which kind of facilitates the mechanoresponse and activates specific mechanical, essentially, programs.
So when you compress the nucleus, you don't see a volume loss? You do, right?
We do, yeah. And this is why we wanted, so that's why we wanted to really decouple the effect of the osmotic, kind of the volume loss versus the tension. This is why the hyperosmotic shock, you have the similar, it's a bit more, the volume loss, but it's very similar. Let's say it's 10% difference. But it happens with really wrinkled membrane, which has really low tension. And this is something that we have measured with the flipper probes.
So is the transcription repression, you're seeing sort of downregulation of transcription. Is that because there is more heterochromatin in compressed nuclei, or is it because it's there's euchromatin, but the transcription is not working?
Yeah. So there's actually less heterochromatin. So there's a loss of heterochromatin with mechanical perturbation, both hyperosmotic shock and compression. We think that transcriptional repression is essentially when you have this like really drastic loss of volume. Just like in hyperosmotic shock, there's really a stress response that's triggered. So there's a global reduction. So I think it's a cellular way because, right, in the nucleus, loss of volume doesn't just mean volume of water. It means that all the transcription factors are now, all the molecules and the wrong concentration, let's say. And so it's just a cellular, it's a way of the cell to essentially to deal with the stress, to pause everything it's doing, to be able to sufficiently kind of address the issue at hand, right, the stress. So we see specific upregulation of certain programs on the background of global transcriptional repression. And that's probably done to efficiently facilitate this kind of recovery or response to stress.
Hi, this is Song Lee from UCLA. Okay, great work.
Hi, Song.
Yeah,
thank you.
Yeah, I have a question regarding this osmotic effect. So this actually is much more complex than a simple difference in the concentration of molecules and also mechanical deformation of nuclear laminar. And it seems there's something common between the tension and osmotic effect. For example, you do see the wrinkling of this nuclear laminar, and that may have caused some detachment between the chromatin, heterochromatin especially, and nuclear laminar, right? So on the other hand, the osmotic stress can cause so many things. That, for example, you mentioned the chemical effects and even the phase separation in many other reorganization of chromatin. So I'm just wondering whether the osmotic stress itself will be a kind of a more combination of many different things that very hard to compare with the mechanical effect alone.
Right. No, I completely agree. And if you have any suggestions of how to decrease nuclear membrane tension with deformation, would be super. It's just, of course, it's a proxy. And it's not, let's say, perfect, but when we saw this loss, essentially when we saw this loss of volume with the compression, we just wanted to figure out a system where we could see the same volume loss, but without this tension. And with respect to the lamina, we see that what is really interesting in the case of compression, we see that there's within 5 minutes already, there's an increase in lamina phosphorylation, and clearly the lamina is being disassembled. And of course, now this has been recently published. And this doesn't seem to be the case in hyperosmotic shock. There, it seems to be really like collapse of the kind of the nucleus and cytoskeleton, which I think the compression is really remodeling the nuclear, the lamina chromatin interface a lot more than does this kind of collapse. But yeah, I would be super happy to chat more on this if you have better suggestions on how to kind of do this study. And so we're really hoping to collaborate with Ilya Leventhal to do some lipid analysis of the nuclear membrane to see if we can figure out differences, and that could be a way to modulate this tension without this osmotic effects, which of course have so many pleiotropic effects, as you said.
Great, thank you so much, Kate.
Thanks. It was
okay,
really a
now
pleasure.
I'm going to. Thank you. Now I'm going to introduce our second speaker today, which is Franklin Staback Rodriguez. Franklin is a PhD student in the Burkhardt Lab from University of Pennsylvania and Children's Hospital of Philadelphia. The title for his presentation is Cytoskeletal Regulation of Differential T-cell Chemotactic Responses.
All right. Hello, everybody. Can everybody hear me? My voice good? All right. First of all, thanks to the organizers for giving me a chance to both give a poster and a talk. Had a lot of fun meeting a couple of you and getting to know you and talking about my work. So, have a high voice anyway. All right, cool. Yes, awesome. So I'm going to talk about cytoskeletal regulation in T cells and in chemotaxis. And in my lab, essentially, we like to take a long view of studying T cell migration. We think of immune cells as just functional cells and cells whose function is not fully described, and especially in the case of migration, because understanding migration is influential to immune response. And we see it in cases where, such as Wiskott-Aldrich syndrome or X-MAID. These are cases where not proper regulation of the cytoskeletal mechanisms of T cells can impact patient pathology and impact these patients and lead to disease. And also, these diseases sort of help us understand the importance of this key regulation of these proteins. We also think that further understanding of migratory mechanisms of cells and immune cell function can help therapeutic strategies, for example, CAR T cell strategies, by further understanding how cells migrate. And so my thesis work is on mechanisms of naive T cell chemotaxis. So basically, we have that naive T cells are cells that have not been activated yet. These are cells that do have antibody. And so this is called a mechanism of surveillance because these cells travel the whole body. They go through different compartments, the blood, the lymph, and spend quite a bit of time in lymph nodes and spleen. And essentially what these cells are doing is they are looking for antigen. If antigen is there, antigen that activates them, then they will be at that spot where they become activated, and then they can proliferate, differentiate, etc. So whilst they're not activated, they are still doing this surveillance where they are traveling and being guided by chemoattractants in and out of compartments. And so some of the signals that have really been shown to regulate this function of naive T cells, we have the receptor S1PR1, which is the egress signal. Without S1PR1, the cells do not egress, do not leave the lymph nodes. Whereas CCR7 is the signal of not only bringing the cells in, but also keeping the cells inside these compartments, exploring them, searching for cognate antigen. So if we look at a model, a schematic of the lymph node, we'll have naive T cells expressing sphingosine 1 phosphate receptor 1, binding S1P, and so this allows them to egress, and cells expressing CCR7 bind CCL19, and this keeps them inside the lymph node, keeps them exploring for about 2 days, and then we know that the cells at one point egress. And so what really began my thesis work was work from former grad student of the lab, Tanner Robertson, who decided to really just understand what the migration of these cells look like. Let me see if I can play this. Yeah, awesome. So what you can see is that when cells are responding to CXCL12 or CCL19, they do more of a lamellipodial type of motility. So they begin membrane ruffling, they begin sort of moving forward. And this is a 2D experiment. They were not confined in any way. They're just lightly adhered, and we put chemoattractant on top of them. And this was not surprising. Other cells use these modes of motility, and it's been pretty understood in a rack-dependent manner. But what was very interesting was when the cells were responding to S1P, because we don't really know how these cells egress, what are the mechanisms, and what the tissue that the cell passes through looks like. But what Tanner importantly showed was that the cells have a different mechanism of motility. So after a couple of minutes of ruffling, they began spontaneously blebbing, or not spontaneously blebbing, but directionally blebbing in the front of the cell for a couple of minutes, and then the cell stopped. And so this was very interesting because bleb-based motility had not been shown in T cells as a form of motility, rather just the cells dying. And so, okay. And so this began my thesis work, understanding the mechanisms and the migration. So the first thing is, based on literature, what do we think is being activated downstream of these receptors? And so I hypothesized that these cells were activating Rho A in response to PS1PR1, since Rho A is involved in blebbing and myosin contractility, which we also looked at in that paper. And CCL19 via CCR7 activates RAC1 in a well-known actin polymerization mechanism that leads to lamellipodia. Now, why this became a very interesting thing is because other labs in the sphingosine 1 phosphate field have typically shown that, in tumor cell lines and endothelial cell lines and HeLa cells, et cetera, these cells typically activate RAC1 in response to sphingosine 1 phosphate receptor 1. So the first question that I had to ask was then what GTPases are being specifically activated in response to S1P or CCL19. For this, I used GELISA assays. Essentially, these are ELISAs, but the capture is a capture protein that is an effector protein that only binds activated GTPases. And so we use this to get a colorimetric readout of GTPase activation. So the first assay, the RAC1 activation assay. Throughout this time point, what I saw was that S1P was not selectively activating RAC. while CCL19 was. So it seemed to go with one part of the story. We think that the cells are not preferentially activating RAC when they're responding to S1P. What was also very interesting was seeing that in response to Rho-A, they were preferentially activating Rho, which seems to correlate with the blood-based motility and the myosin contractility that we have observed, where CCL19 was not. And to validate these experiments, I also did looked at differential activation of PAC. PAC is downstream of the RAC pathway, it becomes phosphorylated. And so I just essentially used it as a marker of differential activation of the RAC pathway. And you can see it here that CCL19 is preferentially phosphorylating PAC, while CCL19 is not. And so since now I'm starting to describe this Rho-A S1PR1 pathway in T cells, I also came across literature where there's been a role for S1PR4 also described. It is not really well understood in the sphingosine-1 phosphate field, but it has been shown that you need sphingosine-1 phosphate 4 to migrate to S1P as well as 1. So I decided to look at that, but with my model of July's experiments and using selective inhibitors for 1 and 4. So the data shows that when inhibiting 1 and 4, and both 1 and 4, we take away Rho-A activation. So it seems to be dependent of both these receptors. I don't yet understand why that is, but it seems very interesting and seems to correlate with the fact that if the cells are not responding through both receptors, they seem to not be able to migrate. And I wanted to also test that as well in my system. And so I did just a transwell assay using both receptors and showed the same thing. Through a transwell assay, both receptor inhibitors took away migration to S1P. And so now, again, since most of this pathway is well understood and well characterized, I wanted to look at the mechanisms downstream of Rho-A in our system. And this work was also started by Tanner Robertson, and he showed that S1P was differentially activating myosin light chain phosphorylation while CCL19 was not. So I'm going to skip through the steps a little bit, but essentially I looked at the downstream effectors according to the literature. So downstream of Rho-A, I looked at ROCK. I also looked at downstream of PLC and calmodulin, myosin light chain kinase. So essentially, both events that could lead to myosin light chain phosphorylation. And skipping over a little bit because of time, essentially what I found is that ROCK inhibition takes away myosin light chain phosphorylation, as well as myosin light chain kinase inhibition and PLC inhibition. So showing that there is a two-pronged pathway where we inhibit the phosphatase and we also activate the kinase. And this is what results in myosin light chain phosphorylation. And so overall, what I showed you mechanistically, S1P preferentially activates Rho-A and leads to myosin light chain phosphorylation, and CCL19 preferentially activates RAC1 and PAC. And so we know that these mechanisms seem to lead to a blood-based motility in 2D, which had not been shown before. And in 2D for CCL19, we see lamellipodial motility. So it suggests that there is a migratory versatility of the cells, but rather, the signaling is different. So now why do we care? So why do cells need these different mechanisms in vivo? And if we think about what's going on in the lymph node, we think about the differences in responses. We think that the S1P, S1PR1 pathway seems to be a quicker response than CCL19, CCR7. And we know that these cells have to get throughout a tissue that we don't really understand. What I'm trying to use, and this is why I'm at the Mechanobiology Symposium, is I'm starting to work with microfluidics. We talk plenty about microchannels, and so this is work I'm doing with Guilherme and Nader, which you guys met and talked to yesterday. So first of all, I'm right now focusing on putting naive T cells in microchannels and trying to understand the different circumstances in which the cells can or can't migrate, and then make S1P and CCL19 specific questions, such as, when the cells are stuck, can we get them unstuck? And we'll eventually pursue assays such as collagen chamber assays, where we're hoping to also understand chemotaxis in response to these two chemoattractants. And a little bit on the design of the microchannels. So essentially, for the diameter of the cells, these cells are about 5 microns in diameter. So if we think that the channel is broadly 5 by 7, they are not confined at all. But when they get to the constriction sites, we modify the size of the constrictions that the cells have to go through, so we can really try to understand where they can't and can't go through. So this is by 2 microns, they just can't or you'll see like 1 or 2 cells that just got pushed in. But by 2.5 and 3, we see like the cells have an easier time migrating. And so as you can see this right here. So this is a cell getting just stuck in a 2-micron constriction. Here's is in 3 microns. This is actually the slowest part of this whole point of view, because in the other ones, we just couldn't see the cells. They were moving so fast, they looked like they were jumping. So yeah, so now I hope to use this technology to make S1P and CCL19 specific questions, try to understand where the cells can or can't migrate, and just, interestingly, understand naive T cell behavior in these environments. And so I'd like to, thank my lab. My lab's awesome. Jan is wonderful, and Guilherme is also awesome. And my funding sources, HHMI Gilliam and NIH. So.
Have time for 1, maybe 2 questions.
Hi, that was a lovely talk. I was just wondering, like, what are the size constrictions of like the vessel leaving the lymph nodes, and how does that directly correlate to the size of your like microfluidic device?
The size constriction when at leaving the lymph node, that's what we don't really know. So in vitro, these tissues have not been properly. People have tried, and are trying to understand what this tissue looks like, but essentially, once they mash it and try to remake it, they don't have the same properties. So we don't know if the cells are passing in between tissues, if they're passing through a pore, if they're not confined at all and they're just directional. We don't know.
Yeah, I have a very quick question, maybe a naive one, but when
that a pun?
Thinking about the blood-based movement, do you see that over time the cells start dying, or is the viability maintained?
Sorry.
When the cells move, in terms of the blood-based movement, do they die eventually, or is the viability maintained? What's the viability of that kind of when the cells start moving like that versus?
Yeah, I mean they don't die after doing this blebbing. They just become rounded up and stop. They stop doing things. Actually, so one of the things that I've been asked about, and I could play around, for another PhD, is adding different chemoattractants and understanding cells after responding to S1P, S1PR1, that we interpret that their receptor has become endocytosed and their pathway is now regulated. If we add CCL19, what do the cells do? Do the cells now are able to migrate, which probably, so yeah, they don't die in this. They just, migrate for a little bit and then start blebbing, and then they become rounded up. So there seems to be a mechanism that moves the cells, and then they have like a smaller form of migration, which could be, behavior necessary to get out of that specific environment that we don't fully understand as immunologists.
So in the lamellipodial case, you have adhesions with the substrate, and that can pull and push. So in the bleb case, how's the cell attached to what's underneath, and how does it pull and move?
How is it attached? I mean, so if I didn't explain it, the cells were adhered with VCAM. And so they are binding to a ligand, and so that if we don't give them a ligand, they would not migrate. So actually, that's one of the secrets with me working on the microchannels is, and why it's taking me like a couple of months to work with them, is trying to understand what ligand conditions the cells need in order to start becoming active inside the channels, because otherwise they don't go in, not even with PLL PEG. Yeah.
Okay, thank you, Franklin.
Thank you.
For our final speaker today, we have Dr. Lance Kam. Dr. Kam is a professor of biomedical engineering and professor of medical sciences from Columbia University. The title for his presentation is T-cell sensing of mechanotopography.
All right, good morning, everyone. Thank you to the organizers for putting this together. And thank you to everybody who's been sharing their ideas, their data, and interpretations over the past day. It's been fantastic discussions, and I am truly hoping that we can talk more about it to help me figure out this thing this mechanotopography idea that I'm going to be talking about today. The starting point for this really is cells. From movies that we've seen throughout the meeting here, and movies like this from the Betsy Laboratory, they show cells as kind of a real strategic level of organization for figuring out how life works. We often call it the fundamental unit of life, right? What we're trying to do is figure out the rules on which make these things work. We heard various variations of this today and various aspects of it. But what I want to talk about is the interface between these cells. It's really a dynamic kind of interaction where there's forces being applied by both sides of this interface. And they're making things happen to both of the partner cells in this thing. It's kind of what inspired us to do a lot of things. And we entered this realm admittedly kind of innocently. We took T cells, put them on substrates made out of PDMS, standard Silgard 184 stuff that everybody works with. And for what I'll be talking about today, these are all coated with anti-CD3 and anti-CD28 antibodies, species-specific ones for the most part, leaving out ICAM and the adhesive molecules. We want to find out what does it do to these. So we started with primary human cells and found that cells could sense the stiffness of the substrate. In the shorter term, a couple of hours, on an hour's time scale, we found that IL-2 secretion by these cells increased, what could be modulated by changing the stiffness of the surface. Quite surprisingly, if this is the stiffest surface and this is the softest, the activation of the cell as measured by IL-2 increased on the softer surface. This continued further on to long-term expansion of the cells. After 3 days, we take the cells off of the surface and let them grow in culture. We found we could actually change how many cells we get in one round of expansion from these over a couple of weeks. Here is the stiff surface. We get a normal growth curve like this. Going to the soft one, we can get about 4 more doublings out of these, an order of magnitude more cells just by stimulating the surface with the softer elastomer. Let's see, where did we go with that? Well, okay, we applied this to immunotherapy. Yes, a daunting topic to present at Penn Medicine, but I'll talk about where we are in this thing over here. What we took on was re-engineering these dynabeads that everybody uses to activate the T cells. One of the challenges with these is that it works great if you're activating cells from healthy humans. It works great for cells from a number of diseases where the T cells can activate and actually expand. But for other ones, like chronic cancers in particular, quite often the cells are exhausted and can't expand too well. Examples down here. Here is healthy donors versus 3 CLL patients. These are the growth curves. You can see for the healthy ones, this is the normal growth curve. Some of the CLL donors, no problem, you can expand them fine. But other ones, we see these deficiencies. We only get like 2 doublings out of these cells. And this is the challenge in the system. What we did is it took the idea of mechanosensing, applied it, and replaced those dynabeads. The ultimate platform we came up with was the elastomer matrix, fibers that contain PDMS inside of them, decorate them with the anti-CD3, anti-CD28, and the cells attached to these things and get activated sort of in this kind of a context. Fibers that are about the same size of the cell as opposed to the flat PDMS surface we originally started with. And what we saw is by using a softer elastomer, we could actually get a lot more expansion of, well, not just healthy cells, we could actually rescue expansion of the cells from the CLL donors. So in this graph over here, the dynabeads are the red bars for 4 different donors, not the same ones as here, but 4 different donors. This is the maximum number of doublings. Particularly for D2 and D4, you can see that using a soft mesh versus a stiff mesh rescue these to give you usable numbers of doublings to get the cells to grow. So this is kind of the clinical application that we're looking at to use this in. But what I want to talk about today is, how the heck are the cells doing these things? We'd love to know what are they responding to. And so what we did was the next logical step. We first wanted to measure what the forces being generated by the T cells on these surfaces would be. And so for various reasons, you've heard about the use of the gels and some beautiful microscopy and measurement yesterday. Well, we went the other route. We used the micropillar arrays developed by Chris Chen's group to do this. The idea being that when you put cells on top of these pillars, they're going to deflect the pillars, and much like the beads, but kind of independent of each other, you could measure, you could estimate the force being applied to it. For humans, for resting human T cells, we put them on there, and they would come down, they would spread. There'd be a short time of transient kind of deflections of these things. Finally, the cell got its act together and decided to kind of pull centripetally on the pillars, very much like in the traction force microscopy mode that we're familiar with. It could generate forces sort of in tens to maybe 100 piconewtons on these pillars. However, T cells are weird. We found that they also love to do another kind of interaction, illustrated up here. They would extend processes between the pillars and reach down deep into the spaces in between. These pillars are 1 micron diameter, spaced 2 microns center to center, and about 6 microns deep. They're relatively floppy because these forces are pretty low compared to the cells you normally use this for. We tried patterning just the top of the pattern, the pillars with the anti-CD28. The cells would still reach down to reach the bottom of this system. To look at it a little bit more closely, we had switched to pre-activated mouse cells for these panels over here. Over the course of half an hour, what would happen? The cells will attach to the surface, start extending processes down between these, and this kind of illustrates the embedding that happens. What you see in green is staining for the cell membrane at certain time points, but really a lot of other structures became embedded within the system. The microtubules would localize about halfway down the pillars. This picture over here, the nucleus in green, the nucleus would embed various parts of itself deep into the pillars as well. So something's going on there. This kind of reflects the different modes of motility that are being described today, but these cells really like these surfaces. And maybe there's something we can do with that, given the ideas that are being discussed today. Do the cells care about these things? This is what I want to get at. Forward. We wanted to find out, we looked at a couple of T cell responses as function of the pillar dimensions. And this is work with Susie Jin. We started with this 6 micron tall uniform pillars here that have a spring constant of 0.77 nanonewtons per micron. This is the cantilever equation that's used to describe it. Apply a force to the top of the pillar, and this is going to be the spring constant of the system. The shortest function we looked at was centralization of the microtubule organizing center. So what we have in this graph over here is how far from the center, 0, as a fraction of the edge of the cell, do you find the mTOQ in each of these different cells. And on our standard arrays of 6 micron tall pillars, 1, 10, and 30 minutes, if you make a measure of this, we find that even though the microtubules were embedded halfway down into the pillars, they moved to the center pretty quick. Now compare this to another type of pillar, 3 micron tall ones that have a spring constant about order of magnitude higher. Oddly, that centralization got delayed on these different pillars. And kind of counterintuitive, you would think that having smaller barriers would give you less resistance to the thing, but it seemed to slow it down. So something strange going on inside there. We tried one other geometry as well, some tapered pillars here that are going to have the same height as the 6 micron tall pillars, but are going to have the same spring constant. Again, the stiffness measured at the top of the pillar, they're going to have the same spring constant as these shorter 3 micron ones over here. And we found that these behave quite like the 3 micron tall pillars. So what we have is, if centralization is a central function of how these cells behave and function, what we saw is the softer pillars, the lower cantilever one, gave better function. Now, moving to longer term functions in this, NF-kappa B, nuclear localization, no effect based on geometry. The odd one over here, IFN gamma secretion by these cells, what we see is that there is a response with the stiffer pillars over here are giving a higher response of IF and gamma than the softer pillars. Kind of the reverse of what we saw in terms of the short-term MTOC localization. So we're kind of getting differential regulation of these functions. I should point out, this one is much more in keeping with mouse cells. We had done this on polyacrylamide gels with mouse cells and found that for those cells, as opposed to the human cells, a stiffer surface gave a stronger activation. So it kind of matches this result over here. So kind of where this is pointing to is that structuring of a material, if you're trying to develop a biomaterial ways to modify it, structuring it so that we don't just have the bulk modulus or something, but we have this kind of structural system going on over here to give measures of mechanical resistance, we can perhaps differentially regulate the different functions of how the cells are going to interact with this system. How this happens, we're not quite sure. The pillars are really embedded deep into the cells. How are they measuring this this spring constant? That's what I'd love to get some insight into because it's something we haven't figured out yet. I wanted to show one more piece of this thing. So, these pillars, we change the spring constant, and we're kind of giving this as the parameter that's being changed, but certainly the geometry is changing as well. How do you get away from that? We have one more set of experiments I want to talk about, starting with these 6U pillars that are made of the Silgard 184. Another way of modulating that stiffness that we approach was to use a softer PDMS, mix the 184 with a Silgard 527, which is like a fourth in terms of spring constant or modulus. We mixed it together at a point where we reduced the bulk modulus of the elastomer by a factor of 2. So this hard one over here is going to be a 184. This can be the softer one over here. And mouse cells, naive cells now, to try to capture the initial interactions of the cells with these ones. This is work by Chirag Sachar. We found that that on the standard ones, the hard ones, the T cells came down in extended processes right to the bottom of the pillars, kind of like we saw in the last one. Change the stiffness in this manner. The cells did not enter the pillars. They stayed on top of these things. Maybe some processes being developed inside here, but they certainly didn't reach down to the bottom of these things. One criticism came back on this is that, well, you're still changing the chemistry of this thing, right? You're mixing two different elastomers together, even though they're the same series, both by Dow Corning, you're changing the elastomer formulation. So the last thing we did in this to try to show that really it is the structural stiffness of this, we modified the pillar system to include magnetic beams essentially inside each of these. We filled the pillars with magnetic nanoparticles that together they can give you a magnetic moment that you can induce into each pillar. Now, this has been used before to make artificial ocilli, and people would apply magnetic forces in this direction and make them move and dance around and swirl. In our case here, we're applying the magnetic field vertically, and what that's going to do is going to make that magnetic moment try to align with the system. So if you have the field off, you have the mechanical stiffness of that pillar remaining. But when you apply the field, what you're going to have is another restoring force that essentially makes it look a lot stiffer. That's going to be kind of added to what you have going on in the pillar over here. With that approach, we can change the apparent stiffness of these things. Soft PDMS, hard PDMS, these are the spring constants there. These are the ones with the magnetic particles inside of the pillars. We kind of estimate those. And then we measured these over here, would be the magnetic particles with the field of about 0.4 Tesla that is applied to the field going in this direction. What we found is that the cells can respond to that. So these are the deflections where negative numbers are contracting, kind of like this example here, and positive numbers on top are going to be pushing them apart, like on the harder pillars. And what we saw essentially is like as we go from softer to harder across these pillars, using the magnetic field to manipulate things now, we can induce that transition from where they're sitting on top of the pillars to now make them penetrate into the system. So it goes beyond just the deflection of the pillars. We found IL-2 secretion by these cells followed it as well. The stiffer you go, the higher the IL-2 secretion from these cells, much like we expect for the mouse cells. This graph over here just does measure the depth. They really are going down into going down into the pillars, kind of illustrated in this function over here. So this is kind of the core thing that we're using to define mechanotopography, and we love to have discussions on how to sense these things. In the last couple of minutes here, I want to touch on a few other things we've been working on, more in the spirit of the mechanical memory that's being talked about here. In a different direction, moving away from the short-term sensing, we are also looking at, well, no, actually, let me put this into here real quick. We are also looking at the counterpart of this whole system. Instead of having pillars on the surface, we made a system where we got PDMS with little pits in it instead. These are intended to allow development of processes by these cells, which across that spectrum of kind of structures, we're calling these ILPs, invadosome-like processes. Essentially, we're allowing the cell to be able to put a process down inside of that hole and develop it. They're first described in terms of allowing cells to probe what's going on in the antigen-presenting cell over here, but we've been replicating this in the PDMS surface. We take mouse cells, put them on top of the surface. In this case, the nucleus doesn't go into the wells, but certainly actin processes are being elaborated into these wells over here. And if you segment them off, we can measure some of the morphology of these different pieces over here. What's interesting about this is IL-2 secretion and IF and gamma. They both follow the same kind of pattern. We get a certain amount on the flat planar PDMS surface, but on surfaces that have 1 micron pits that have 4 micron depth, we get an increase in cytokine secretion. It seems to be a biphasic response, going up to deeper pillars of deeper pits of 6 micron depth. We still have an enhancement over the planar surfaces, but it's not as high as on the 4 micron depth now. So we're trying to figure out, why do we have this kind of biphasic response? The ability to extend these processes seems to help, but there is a point at which you start to have diminishing returns or even a negative effect from it. The intriguing part about this, in fixed images of cells on these surfaces, if you measure the length of these processes that are extending into there, the average length is only about a 1 micron. Certainly there are much longer ones that go into each of these, but they don't really reach the full depth of the pit. So somehow they are sensing how deep the pit is without really touching the bottom of it. It's something that we're still trying to figure out. Some live microscopy things we saw today or yesterday would be wonderful to apply to this kind of thing. But ideas on why we can sense this would be great. Okay, so with that in place, I want to talk about just going a bit into the epigenetics of how things behave. We're also looking at induction of regulatory T cells as a function of stiffness on these surfaces. I'm going to speed up a little bit because I am running out of time a little bit here. Essentially using the same kind of system, looking at induction of regulatory T cells from conventional cells on polyacrylamide gels, and these are human cells. We found by looking at 3-day induction of these cells as a fraction of FOXP3 expression, an increase in the number of FOXP3 positive cells as a function of stiffness. Stiffer surfaces under the conditions we set for this gave a higher induction of these systems. Perhaps more interest to this group over here. By doing this at the 3-day time point, we could capture cells kind of along their progression from being a conventional T cell to turning into regulatory T cell. We did single cell transcriptome analysis on this, beautiful UMAPs, these kind of things. We can catch these populations kind of still connected in some sense, right? So what we did is we did a pseudo-time analysis across this, or trajectory analysis, and it saw that a number of genes, particularly those, well, this shows you all the genes in that process, but a number of these, particularly those involved in metabolism, final changes of oxidative phosphorylation, these would increase as the cells went from a conventional cell to a regulatory T cell. But if you separate out the two populations in terms of those stimulated on the hard versus the soft polyacrylamide gel, we found a difference in the pattern, in that the transcription of genes involved in these last final stages of energy usage was higher on the ones that are hard versus soft. So we're not just changing the number of cells that get induced to be regulatory T cells. We're kind of changing the degree of the functionality, perhaps through different differentiation state in these systems. Going a little bit beyond the transcription of this thing, we went back to PDMS as a more durable system for trying to induce these T cells. And found some very strange things about this thing. So these over here, we want to look at longer term inductions, not just 3 days, we're going out to 15 days, but on substrates of 3 different stiffnesses, 17, about 20 kilopascal up to 2000. So a wide range of stiffness on these things. With the PDMS at this higher range of stiffness, we find that induction of the regulatory T cells, both in terms of percentage of FOXP3 and the cell counts that we get, these are better on the soft surface rather than on the hard one. Kind of mimicking our production kind of work on this thing, but now going into induction of the Tregs. They seem to be more stable over time. I don't have the data on here, but they also are more functional. These are the ones made on the softer surface compared to the stiffest one, have a higher suppressive function on conventional cell proliferation. What's going on inside there? We did methylation analysis on these systems. This is just a differential analysis over here. Stiffer on this side, softer on this one over here. We found that there are a number of different genes that have differential methylation in these systems, suggesting we're getting different kind of cells. Again, not just a number of cells, but they're functionally different from each other. Looking a little bit different, closer into some of these, like the FOXP3 itself. The induced Tregs look very similar, very different from the normal natural Tregs. But within this range over here, we're seeing an effect of the stiffest surface versus the softest ones. Surprisingly, the ones induced on the hardest surface are more like the natural Tregs than the ones on the softer ones. So there seems to be a balance here. We're getting a signature that looks better on the stiff surface, but we're getting more cells on the soft surface. I think putting these two together, we can try to find conditions that balance these two outputs and improve Treg induction. So I'm out of time here. I'm gonna skip the last piece of this. We were coming back to looking at how can we understand the difference in expansion from different patients. We have a clustering method to do this, but I think I'll save that for discussions after this. So in closing, I'd like to thank, well, first of all, my group that did all the work in this, our funding sources, and also thank you for your attention today, and I'd be happy to answer any questions.
Okay. Yeah, okay. Hey, fantastic talk. I have 2 questions. One, have you put naive T cells on top of the pillars to send to, and tried to understand the behavior? And 2, can you use that system to surface coat these pillars with chemoattractant ligands like CCL19, CXCL21, et cetera?
I missed the first part. The naive cells and
naive cells instead of activated cell or activating cells with CD3, so just putting naive cells and
look at the longer term function. No, we haven't completed that part there. I mean, we had the IL-2 secretion from the naive cells on the pillars, on the softer pillars, but we didn't go back to the 3 different higher stiffnesses from there. Yeah, cytokines on there. This would be amazing to put on there. See how it affects it. There's got to be interaction between these. We know like CD28, we have the anti-CD28 on the pillars. We know that behaves just as well in solution as on the pillar. So that's not itself involved in mechanosensing. We'd love to see what happens with the cytokines. Does it behave differently in different zones?
Right. So like, for example, CCL21 is when the cells encounter it, surface bound, and that allows them to get into lymph nodes. So it's very,
exactly.
So thank you so much for a great talk. I'm curious about the distinction between those extensions and invaginations in the interface between the T cell and the APC cell. Are those involved in just increasing the surface area, you think, between the cells and therefore allowing for more interaction? Or do those extensions actually form sort of microdomains in the T cell that allow for, let's say, highly concentrated signal transduction reactions going on?
It's not clear. The original, a lot of that started by looking at electron microscopy pictures between the cells, and in that case, we also saw exclusion of the coats, the glycocalyx essentially, to allow better connection between the TCR and its target. So both of those, like to allow better connection and to kind of localize the signaling, this is how it's proposed for. But our results work whether you coat the inside of the channels or not. And that's why we're not sure exactly where it ends up.
Yeah, that was going to be a follow-up question, is if you put the coating only in the channels versus on the surface, do you see a distinction in activation or the behavior of the cells?
Haven't figured out how to do that one yet. We know how to put it on top and not the bottom by stamping. But I guess you could stamp a passive eating thing on first and then coat it, but we haven't tried that yet.
Thanks.
Yeah.
All right, thanks for your talk. I have a question that sort of follows on the same theme. So you pointed out that the micropit experiments were mimicking how the antigen presenting cell has pits that the T cell sticks down in. And so can you coat those micropits with antigens or components of the antigen presenting complex? And would you get, more robust or different structures or, more signaling centers or anything like that?
I think that's probably what we're gonna try to do Friday, I guess, when we get back. I don't know. I mean, thank you for the suggestion. Again, we knew how to put it on top but not the bottom. That'd be a great way to test whether that makes sense. Yeah, because that's where a lot of the motivation came from. It's from those pictures of the cells. But in that case, the T cell, we think, was probably very instrumental in making that structure happen, pushing into the other cell. And right, the idea over there is that a lot of the activation signal is going to be found in that structure.
I guess if you have the activation signal there in your model, you might get more profound results or something. I don't know.
Yeah, the weird thing is that the 6-micron pits gave a lower enhancement of the activation. So even providing more things along, if we assume that the pits were evenly coated, the 6-micron deep pits should have provided more activation signal overall. But we're getting less. We're getting a lower activation from those. But somewhere's in that mix could be activating another thing that counters the activation. Somewhere's in the mix.
Great talk here. You showed that the nucleus is also deformed with the micropillars. So do you think there's anything that changes the nucleus and the chromatin? Maybe.
That's what I'd love to find out. And, I mean, that's an area we're not too familiar with, but I'm looking forward to talking to you folks here on how to get a grip on that. Something's got to be going on inside there, right?
Hi, really great talk. Thank you. So in some of your earlier work, you were talking about those micropillar stiffness and depth, and you were giving metrics that were kind of a trade-off between cytokine secretion and the distribution of the MTOC. Could you elaborate more on the importance of that MTOC centralization, the speed of that in the context of T cell activation? Like what's the importance of looking at that?
There's more important. I should specify here. We're looking at the centralization of the MTOC. I mean, when the cell lands, it could be like kind of wherever it's facing, right? But pretty rapidly after you form the contact with surface or another cell, motors engage that move the MTOC to the middle of the cell. And that's believed to be involved for a cytotoxic T cell for delivering the vesicles to that target. For helper T cell, that would be for delivering vesicles of not to kill the other cell, but to inform it. So that's normally seen as kind of a key step along the way of activating and forming a functional interface with the surface. It hasn't really been studied so much, but on how the timing of that happens, but the relocation of that to the middle of the interface is believed to be a key step or a key indicator of the functionality. That was developed mainly in terms of like flat planar surfaces and things where there's no barriers or anything in them. So how indicative is that of a function that's behaving oppositely to the other ones that we had? We're not exactly sure, but we see that we are retarding the motion of this along the surface with the softer pillars.
Hi, thanks for your talk. Earlier, like at the beginning of your presentation, you were talking about how you could partially rescue T cell expansion when you put the cells in a matrix. Do you think you can kind of see a different response of activation, like on an organized matrix versus more of like a randomly aligned matrix?
Let's see, we tried aligning them versus randomly aligned. The pore sizes in these are about 50 microns across. We did not see an effect. The short of it is we did not see an effect of alignment versus random. We think it's because the cells are like they're like hanging on to a log. They see locally just the fiber rather than like other cells see the alignment between two fibers, just because the T cells are so small. Yeah. Right. Yeah, so I guess to reiterate the question for this microphone, you see a transition in mechanosensing with kind of a barrier around the 1 kilopascal range. Your surfaces had the adhesive molecule as well, right? Yeah. So I think the numbers may be a little bit different. We have just this anti-CD3 and the anti-CD28. The exhaustion measured in the long of response. Okay. We haven't looked at that directly. We certainly, in terms of the shorter term responses, activation, 4 hours cytokine secretion, we see a loss of it on our surfaces with the mouse cells, particularly. Below 5 kilopascals, we saw loss of adhesion as well as the mechanosensing of the cytokine secretion. Above that, we have strong adhesion and the modulation of it. Sorry, CD4, sorry. So there's definitely different regimes of what's going on. I think there's very, below some cutoff, you're talking about turning the cell on at all. And then above that, activation. Now, where does exhaustion fit into that? I'm not sure.
Great. Thank you, Dr. Kem.
All right, thank you.
So this concludes our session. Let's thank all our speakers again.
All right, so welcome all to the final session of the Mechano-Immunology Symposium. My name is Venkatesh, and I'm one of those few plant biomechanic people in CEMB, and I'm from Anderson Lab at Penn State. And it's my great pleasure to introduce the pre-penultimate speaker of our symposium, Dr. Adu-Berchie, who is a researcher at French National Center for Scientific Research, National Institute for Agriculture, Food and Environment. And he is gonna give his talk online. The floor is yours, Dr. Berchie.
Yeah, thank you very much. So thank you very much for the invitation. I'm very disappointed and very sad to get stuck in Toulouse because my flight was canceled on Sunday. But yeah, it is what it is. And so I'm going to talk about plant and most specifically connection between plant mechanoperception and the plant immune response. So the most common form of plant immunity is called the quantitative disease resistance. And it's characterized from a phenotypical point of view by a continuous distribution of phenotypes. So it means that plants are not fully resistant or fully susceptible to the disease. It's always in between. Almost every time in plant, the resistance is achieved by every living cells of the organism. And another specificity of this resistance is, from a molecular point of view, it's highly multigenic. So it involves the modulation of thousands of genes. In the case of Arabidopsis thaliana, there are more than 1/3 of the genome modulated to participate to this resistance. So more than 10,000 genes. And obviously, all these genes are not specific to plant immune response. They're also involved in a lot of different functions. And so over the year, we gain insight in the plant disease resistance. And in fact, we described it and we understand it like a non-covalent interaction between the molecule associated with the pathogen or the damage created by the pathogen with plant receptor and downstream signaling pathway, which at the end triggers the plant resistance. And it's perfectly okay. It leads to great breakthrough in understanding plant immunity. But in fact, it's not really sufficient to explain something, and especially to explain the spatio-temporal patterning of the plant immune response. And so the idea was that maybe it's not sufficient because you cannot reduce plant microbe and plant pathogen interaction to the chemical part of the energy, because obviously plant pathogen interaction involve also a lot of physical forces. The first reason is because the microbe need to punch into plant tissue to penetrate into their host, and all the microbes are not so subtle that Pseudomonas syringae we saw yesterday. You see here what happened with Sclerotinia sclerosorium that create an affection cushion to punch through plant tissue. And obviously, once into their host, microbe grows, which lead to the formation of geometric incompatibility, so the formation of mechanical stress and strain in plant tissue. So that's true for fungus, but it's also true for bacteria, because at some point, bacteria can form very dense biofilm, leading to the substantial modification of Tugger pressure. That's the hydrostatic skeleton of a plant. And at the moment, we have no clear idea if all these mechanical cues participate in the regulation of plant immunity. But what we already know is that in plant mechanoperception is a kind of super sense. It's involved in the regulation of a lot of processes. And plants are able to perceive mechanical cues coming from associated with the environment, such as the bending created by wind or the gravity leading to the regulation of growth. But they can also perceive internal mechanical signal, that's involved in the regulation of ontogenesis, morphogenesis, and also proprioception, so the ability of plant to perceive the distribution of their own weight in the gravity field. And in plant, mechanoperception is very tightly associated with two cell specificities. So the first one is a vacuole, so full of water, and obviously the water flux is driven by the osmotic pressure, and the cell wall. So cell wall is composed by bound polysaccharide and form a stiff material with Young modulus in the magnitude of 1 megapascal. And obviously, when the volume of water increases because of a gradient in osmotic pressure, that creates mechanical stress and strain in cell wall depending on cell shape. And mechanical stress and strain are actively monitored and perceived all along the life of the cell. And yeah, it's very interesting. Maybe you will correct me at the end of the talk, but in my knowledge, it's the only case I know in which a cue, the signal code for an intensity, so the magnitude of stress and strain, but also the direction, because stress and strain are tensorial by nature. And so to test a possible connection between the plant immune response and plant mechanoperception, we use the Arabidopsis thaliana, the model plant, and a favorite bad guy called Sclerotinia sclerosorium. So it's a necrotrophic fungus. So it means it's actively kill its host to grow, and is able to infect more than 400 species, and is very widespread in all the temperate area of the world. So it means that if you have a garden and you grow dicot in it, be sure that Sclerotinia is waiting for new vegetables. So we focus on what happened in the later phases of infection. So once the fungus was already growing in a plant tissue. And in fact, we need to basically monitor two things. So the first thing is obviously the mechanical cues. So in a case, we monitor the mechanical strain on the basis of image analysis. And the other things, obviously, is the response of cells. So to decipher if cells perceive these mechanical cues or not. And for that, in fact, we monitor the organization of plant cortical microtubule because in plants, cortical microtubule are very convenient anisotropic probe because in the absence of growth, they are organized into the direction of the principal strain or the principal stress. And we monitor both the mechanical cues and the plant cells response over 2 hours. So because we were very limited by the growth speed of fungus, so the growth speed of Sclerotinia scleroserium is about 1 millimeter per hour. So when we try to monitor on longer time, most of the time we monitor mostly dead cells. And so this is the kind of data we produce. So you see here on the micrograph, the epidermal cell, the jigsaw puzzle cell of Adu-Berchie staliana, and the filament are the cortical microtubule. And you see here on top left, a knifey of Sclerotinia scleroserium. And we compute for every visible cells the relative surface variation, so mechanical strain, and also the direction of the principal stretch. That's the blue segment of every cell. And as you see, we observed a limited relative surface variation in the magnitude of 1%. And you see that the principal strain patterning is a mix between radial and circumferential. And so we derived a very simple model to understand further what could happen. And we mimic a plant tissue here as a membrane under isotopic tension. And we mimic infection in 2 cell domains by releasing the internal tension. And so as you see, when the release of tension is limited, we observed strain patterning, which is a mix between radial and circumferential. And when we go further, we release more internal tension in the infected area. We observe a perfectly circumferential strain patterning. That's the pattern you can observe after laser ablation. So it means that during infection, there is next to the infection, there is the release of stress in the plant cell wall, leading to this limited variation of cell surface. And in fact, why it's limited? It's because it's achieved chemically. So a few years ago, we modeled the metabolism of Sclerotinia scleroserium, and we found that an important part of its metabolism is devoted to the biosynthesis of enzyme targeting the primary cell wall and the production of oxalic acid. So now the question is, are these limited mechanical cues are perceived by cell or not? And so we monitor the organization of the cortical microtubule. And as you see in the healthy cells in infected tissue, we observe that cortical microtubule were more organized into one main direction. And when we compare this direction to the direction provided by the infection-derived stretch, you see on the right that more and more cell exhibited microtubule reorganizing into the direction provided by this infection-derived stretch. So it means that this, despite limited, this infection-derived stretch associated with the necrosis created by the pathogen are perceived by cell, at least in the vicinity of infection. So as you know, plant tissue are more like solid-like structures because cells are bound together by the middle lamella, so they form a kind of continuum of cells. And yeah, as I said, they are under tension, especially epidermal tissue are under high tension. So you see here what happens when you cut a cotyledon leaf of Adu-Berchie staliana, so you release the tension that produce a very fast displacement in tissue. And so the idea was that this solid-like structure may favor the transmission of the fluctuation of mechanical stress in cell wall, maybe to inform distant and healthy cells of the presence of the pathogen. So I'm sure you will agree with me. So to be efficient, the transmission of the stress fluctuation of the mechanical cues must be faster than the growth of fungus. And to produce a 1 millimeter growth, we know that a fungus needs something like 1 hour, so 1,000 seconds. And so the mechanism could be involved in the transmission of stress fluctuation in cell wall could be based on the diffusion of enzyme or oxalic acid. But in this case, in the best case, in fact, the typical time is of the same magnitude than the growth speed of fungus. So it means that all the diffusion of chemical product, in fact, occurs in the same time that the growth of fungus. And so the other possibility is the reshuffling of internal stress caused by the necrosis. Necrosis is like all, so you modify the boundary condition, and so stress, you perturbate the force balance in a cell wall, and obviously this force, the force in cell wall, will change to get back to a new mechanical equilibrium. And this mechanism, in fact, is purely passive and involves the movement, the displacement of water. So the typical time is given by the pore-elastic time, and in fact, it would take less than 1 second. So it's definitively the best candidate to, in fact, transmit stress fluctuation in healthy cell far from the infection. And so we use a very simple model. We use we previously used, and so it's a membrane under tension, and we mimic infection by releasing the tension. And as you see, when we release the tension, there is a formation of an overstretched ring around the necrosis. And what is nice is that the width of the necrosis, the overstretched ring, does not depend on mechanical parameters such as the Young modulus or the internal tension. It's only depending on the relative size of the infection compared to the size of the leaf. And as you see here, in fact, the width of the overstretched ring could be quite important. So when the infection is 20% of the size of the leaf, the width of the overstretched ring is also 20% of the size of the leaf. And over time, almost every cell composing the leaf could be at some point in the overstretched ring. So our model is very simple, maybe simplistic, because plant tissue are also highly viscoelastics, and maybe maybe it can damp the stress fluctuation. So in fact, we try to observe what could happen by monitoring the reorganization of cortical microtubule. And as you see, next infection, we observe the very same cortical microtubule anisotropic patterning we observed previously. So more and more microtubule reorganize into the direction provided by the infection-derived stretch. And we look what happened 2.5 millimeter away from the infection. So it means next the border of the leaf. And as you see, there is the initiation of this cortical microtubule anisotropic patterning. So all the microtubule are not yet reorganized into the direction of the stretch, but they have started to reorganize. So we know that to kill a whole leaf of Arabidopsis thaliana, sclerotinia takes around 24 hours. And so it means that the cells in the overstretched ring can be informed of the presence of the infection something like several hours before the fungus reach these cells. And so this time it could be enough maybe to create, to implement immune barrier to face the growth of the fungus. And so that's we monitors, and so we perform a RNA-seq analysis, so in the overstretched ring around the necrosis, and we want to test if the transduction of gene is associated with the ability of cortical microtubule to reorganize into the direction provided by the infection-derived stretch. And for that, we use a mutant line called PFD61, which exhibits only one mutation in the prefolding 6 gene compared to Colombia. And this mutation, in fact, lead to an increased cortical microtubule dynamics. So microtubules are functional, they reorganize, but much more slower compared to Colombia. And as you see, most of genes are in the overstretching are expressed in the same way and independently of the cortical microtubule dynamics. But some of these genes appear depending on the dynamics of the cortical microtubules. And so, in fact, we look, we compute the number of genes in gene ontology associated with plant defense or plant immunity. And as you see, for every ontology, more genes associated with the immune response were modulated for plants with regular cortical microtubule dynamics. So obviously now we want to test if this gene modulated can participate to the immune response. And so for that, we basically monitor the growth speed of fungus. So you see here, we compute the speed of lesion development on Arabidopsis leaf. And for plants with different cortical microtubule dynamics, so plants with increased cortical microtubule dynamic and impaired cortical microtubule dynamics into different genetic background. And as you see, we observe that for plant with impaired cortical microtubule dynamics, we observed that plants were much more susceptible to the infection compared to plants with regular or higher cortical microtubule dynamics. And so this variation is in the amount of 30%, which is quite important, especially in the context of the quantitative disease resistance, because it's highly multigenic, and most of the time the effect of a gene or some gene are more in the magnitude of 1%. So it means the contribution of the overall resistance is quite important. Yeah, so to put it in a nutshell, the infection, in fact, consists in the partial release of cell wall tension caused by the fungal hydrolysis. And that leads to the formation of an overstretching around the necrosis. And this overstretching, in fact, are the pathogen-derived mechanical cues actively perceived by cells, and leading to the differential expression of gene associated with resistance through the cortical microtubule anisotropic patterning, and that this mechanism contributes a lot to the plant immune response to necrotrophic fungus. And so we call this layer of resistance, which obviously is not the only one, but maybe it's a new one to study. We call this layer of resistance MTI for mechanosignaling triggered immunity. To finish, I would like to thank people, collaborators working with me on MTI, but also on something we call sigmoimmunity. Sigmoimmunity, in fact, is the modulation of the MTI by external mechanical signaling. And so I just want to name a few of them. So Sylvain Raphael, Medicafif, Ophélie Léger, who did the experimental part of the work I show you today, and Frédéric Garcia. So thank you very much. Yes, I'm ready for the question.
Thank you so much for the talk.
Hi, this is Miley from UC Davis. Very nice talk. I just have a question about how this mechanosensing is actually associated with the damage-associated response. So because fungus, especially necrotrophic fungus, they release a lot of peptides, things from the cell wall, and this causes an immune response in the plant that is associated with damage. So I just wonder, what's your perception? How is it associated with mechano perception?
Yeah, it's a very good question, and it was very hard to tackle. So in the vicinity of affection, so just next the necrosis, it's almost impossible to think there is only mechanoperception, because on such a small distance, all damp and so on can diffuse, and so to trigger plant immune response, PTI, maybe ATI, and so on. So next to the infection, there is a mix between all the PTI, MTI, ATI, and so on. But far from the infection, that's where is the difference, because damp cannot, small molecule cannot diffuse quickly over a long distance. We are talking about millimeters, and so the idea is maybe for these cells, so 1 millimeter away from the infection, the MTI dominate all other form of perception. Was it clear?
Yes, I think that was clear. Hi, I'm Charlie Anderson from Penn State, also
hello.
a plant biologist. So I'm really interested in the timing of the microtubule response in relation to other potential signals that might be sort of physically mediated. You know, for instance, Simon Gilroy's lab and others have shown that there are calcium waves that propagate throughout the plant pretty rapidly in response to, you know, either wounding or even herbivory. So do you imagine that there is a wave of calcium that is transmitted across the leaf when the fungus starts to invade, and how does the timing of that relate to the timing of reorientation of microtubules?
Yeah. So yes, yeah, tough question. So first, we monitor calcium wave during infection, and in our results, we saw calcium wave only when the fungus punch plant tissue, and that's it. After that, we do not observe calcium waves. So first thing, and second thing, for me it is not clear connection between modification of calcium concentration and reorganization of a cortical microtubule. To answer maybe your question is some other signal which same typical time scale could also be involved, but it was not. I think that at least we know now that mechanical signals are involved, but we have not an idea about the big pictures. It's I think that we will do in our further work.
Thank you.
Are there any more questions? Or all right, if there are no more questions, please join me in thanking Adeline. And our next talk would be by Adu-Berchie from Wyss Institute at Harvard University. He's a research scientist there.
Okay, that point works. Yeah, so I'm excited to present some of the work we've been doing, where we've been trying to see how matrix viscoelasticity influences or modulates T cell phenotype. So adoptive T cell therapy, I think we've discussed quite a bit of it during this symposium. But kind of just to give you a brief overview, it involves taking T cells out of the blood, activating them, and after activating them, performing in vitro genetic manipulation, generating CAR T cells or TCR T cells that are able to recognize a specific antigen on tumors, allowing these cells to proliferate and then putting the T cells back into patients. And there has been a lot of interest in thinking about how to generate specific T cell phenotypes, because phenotype generally links to function. And so for a specific therapeutic application, how do we generate specific T cell phenotypes? Now, there's been a lot of work on modulating the activation and changing the cytokine milieu and others. But I wanted to take a different approach in the Muniz group to really think about how the environment of the T cells can modulate the phenotype. And this was motivated by studies. This is from Rafi Ahmed groups, where they take T cells, inject those T cells into mice, and then they're able to see that over time, the T cells that are in different tissues then end up having different phenotypes. We took that and we extended this analysis, and we were specifically interested in these three tumor types that are the single-cell RNA-seq data sets that have already been published. And we saw that T cells in blood, normal tissue, and tumor tend to be different. And these are pan T cells. But then we're also interested in T cells that have shared TCR clonotypes. And we saw that they are also different. And so we have a situation where T cells in different tissues are different, whether or not they have the same parent T cell. And of course, there are a lot of things that are different between blood, normal tissue, and tumor, but we're specifically interested in the mechanical environment. So we asked these questions. The first question is, what role does the tissue mechanical environment play in tuning T cell phenotype? Now, the second is, can we then generate functionally distinct T cells by mimicking key features of the tissue microenvironment? And so here we have, for instance, if you take T cells and then you put them in matrices of different mechanical properties, will they end up generating phenotypically distinct T cells? And within the context of cancer, will that lead to different therapeutic outcomes? So that is what we were trying to pursue here. So I want to take a step back and go through some quick definitions. We've had a lot of these being thrown around, stiffness and viscoelasticity. So stiffness here just describes a material's resistance to instantaneous deformation. So we can intuitively think about things being stiff or soft. But viscoelasticity really is a specific property of many tissues, all tissues, and also matrices, which have both a viscous component and an elastic component. And so it refers to materials' time-dependent mechanical response to applied stresses and strains. And so then imagine two matrices, one of them being covalently cross-linked and the other one non-covalently cross-linked. Imagine that those two matrices have the same stiffness, right? Now, in viscoelastic definitions, what we are trying to say here is that the matrices that are more covalently cross-linked will relax a lot slower when you apply a specific stress that reaches a particular strain. And so to hold it at that strain, the covalently cross-linked matrix will relax a lot slower than the non-covalently cross-linked matrix. And so the covalently cross-linked matrix will be called a slow relaxing matrix, and the non-covalently cross-linked one, a fast relaxing matrix. And so we believe that a system that allows for the independent tuning of matrix stiffness and viscoelasticity could be used to generate functionally distinct T cells. So that is what we pursued. So just to present what a material system is, so we have a collagen type 1 base system where we modify with norbornene. Now, norbornene reacts in a very specific way with a group called titrazine, click chemistry. And so we also know that collagen type 1 is able to polymerize and form a gel by itself without any need for covalent cross-linking. And so if we take T cells and we encapsulate them in these matrices, and we flow in methyltitrazine PEG methyltitrazine linkers, what we do get are very local cross-linking, which then allows for the tuning of viscoelasticity. And so the locally cross-linked matrices become a slow relaxing matrix, whilst the non-covalently cross-linked matrix becomes a fast relaxing matrix. One thing that we saw was very interesting was that we were able here to maintain the stiffness of these matrices, whether or not they are locally covalently cross-linked. And this, at least to our knowledge, is the first demonstration of doing this, since collagen generally tends to increase in stiffness when you cross-link them. We are then able to significantly change the viscoelastic properties of these matrices, with the fast relaxing matrix relaxing a lot faster than the slow relaxing matrix. Not only that, we are also able to maintain the collagen architecture. And so if we have, in the presence of cross-linking, we do not in any way see a change in the fiber thickness or the pore diameter as measured by cryo-SEM and SAG. So now we have a system where we can pretty much maintain at least for the factors we measured, we're able to maintain all the properties whilst changing only viscoelasticity. And so then if you see changes in T cell phenotype, you can correlate that to viscoelasticity and not any other thing. So we took T cells and we put T cells in matrices that are either soft or slow, soft or fast, soft or stiff, or slow and fast. And we did see that T cells in matrices show phenotypic differences that are driven more by viscoelasticity than by stiffness. And so if you look at CD62L and CD45RA, which are the canonical markers we use that show T cell memory phenotype. You do see here, for instance, that the soft fast and the stiff fast are more similar to each other than the soft fast and the soft slow. And there's a PCA plot that shows that at PC1, the stiff and soft slow group together. Now, I wanted to mention that stiffness also has a role, because if you compare the soft fast and the stiff fast, they are different, but most of the difference is driven by viscoelasticity and not stiffness. I wanted to further go ahead and see what is happening. And so we took T cells and put them in the matrices and performed single-cell RNA sequencing. And so here what I'm showing is UMAP density plots where the hot areas means more cells. And so you see that there are areas, the areas here that are hot in the fast relaxin are different from those areas in the slow relaxin. We then went ahead and we performed non-negative matrix factorization to be able to, in a non-specific fashion, be able to identify functional gene modules. And by doing that, we're able to find 2 functional gene modules upregulated in the fast relaxin, 4 in the slow relaxin. When we look at the genes that are specifically characterizing these models, we took those genes, performed GSEA, and so that most of the pathways that are upregulated tend to be T cell activation, TCR signaling, T cell killing, and others. Whilst one module showed up that was enriched in the fast relaxin matrix, which is leukocyte adhesion to endothelial cells. Now, how does this relate to in vivo? And so we took for instance, the slow relaxin matrix, the genes that make up the slow relaxin module, and we said, how is the expression of these genes in blood, normal tissue, and tumor in the tumor type that I show here, both for pan T cells and T cells with shared TCR. And you see that the levels of expression are different based on the tumor type, with the T cells in the blood being having the lowest expression, T cells in the tumor generally having the highest expression. Now, from there, I wanted to move on and look at function, because we know that T cells are phenotypically different. They have different transcriptomic profiles. How about function? Does this translate to function? And so we did 2 things. One, in vitro functional test, where we generated anti-CD19 CAR T cells after stimulating them with this three levels of stimulation, putting them in soft or stiff gels, fast or slow relaxing gels. And we see here that T cells in the slow relaxing gels killed better than T cells in the fast relaxing gel. And T cells in the, for instance, the stiff, slow relaxing gels killed the best. And so there is kind of this synergy between stiffness and viscoelasticity that could even lead to more killing. And importantly, this killing was accompanied by increases in several effector molecules like TNF-alpha, IL-2, and granzyme B. So these T cells have a marked phenotype. They're able to more quickly produce these effector factors and able to kill better. Now, what happens in vivo? So we took these T cells and we adoptively transferred them into mice that had already been given RIG tumor cells that express luciferase, and we did see that here as well, the T cells in the slow relaxing gels led to about 80% of the mice being able to clear the tumors, or the T cells in the fast relaxing gels, all the mice died. And so we have a situation where this phenotypic differences we are seeing leads to therapeutic outcomes, driven again only by changing viscoelasticity and nothing else. And so then, to summarize, we've engineered a collagen type 1 ECM model with independent tuning of matrix stiffness and viscoelasticity. We've demonstrated that mechanical properties in these tissues regulates T cell phenotype, and that viscoelasticity is a dominant driver. We've shown that this is consistent with T cell profiles that are obtained with mechanically distinct tissues. We have generated phenotypically and functionally distinct T cell populations by tuning ECM mechanical properties. And so I'd like to thank all the people that helped. Dave, my PI, worked really excellent PI who gave me the freedom to do this project. Newton, I worked really closely with a PhD student who is defending this afternoon. So congratulations to her in advance. Also want to highlight Kyle, who also was very helpful, especially thinking through the mechanics and how mechanical stimulus impact T cell phenotype and function. So I'll end here and take questions.
Great talk. I have two questions. One is, what about these ligands presented by collagen itself for cell adhesion? Do you observe some difference or effects on the T cells? And second question is, how many folds of T cell expansion can you obtain from this 2D scaffold in comparison to Dynabeads, for example, in 3D suspension?
That is a good question. I'll take your last question first. So these T cells were first activated with Dynabeads for 4 days before they were put in the gel. So I wanted to say that, so these T cells are T cells that have already been pre-activated. And so we are not changing the initial activation, I just changing the phenotype of the T cells after activation. Now these systems are 3D systems where the T cells are encapsulated. And so the expansion fold is not as high as if we left them in 2D. So in this instance, we're not necessarily trying to maximize expansion. We're trying to understand and identify the differences in phenotype. Now, there are also like conversations we've been having as to how do we then apply this principle of viscoelasticity in situations where you also maximize expansion. So that work is ongoing. But for now, the question is, can we identify viscoelasticity and demonstrate that this principle is important? We don't necessarily maximize the expansion. So they don't expand as well as the Dynabeads. So in terms of ligands, we have not seen differences in the ligands, but it is possible that the ligand cluster might be different. Maybe the T cells might be pushing and pulling the strands a bit differently. The fast relaxin, maybe they might be able to pull it a lot better than the slow relaxin. We have not done detailed imaging studies. That's also something that a few folks in the lab are interested in. And so we might be able to answer that question later. But for this study, we didn't. We don't know what the answer is. In the 3D, yes, they do. Yeah, we've done some time lapse to show that they grab onto the collagen very actively. They migrate through. They migrate slightly less in the slow relaxin than in the fast relaxin. But you can see them actively engaging with the collagen matrix. We just collagenase type 4. Yeah.
Hi, thank you. It was a great talk. I have 2 questions as well. What kind of tumors were present in the mice, and have you thought about looking at how effective these are versus the slow and fast at different types of tumors, as well as how did you provide the mechanical stimulation in one of your previous slides?
All right, so let's look at the type of tumors we've tried. So we've tried 2. We've tried the Raji tumor model, which is the liquid tumor, and we've also done mouse B16, F10 melanoma, and we see the same trends. Your second question was, how do we provide mechanical stimulation?
What kind of mechanical stimulation? Do you stretch it, compress it, provide what kind of forces?
Okay, so I'm not sure if you're talking about for the T cells or for the matrix. So for the matrices, we identify changes in viscoelasticity by performing rheology.
You had a slide previously where you had the low stimulation, medium stimulation, and high stimulation. So I was wondering what simulation you were talking about.
Sorry, so these are different Dynabead stimulations. So we wanted to see what happens if you activate these T cells at different Dynabead densities before you put them in the matrices, if that level of stimulation has any effect.
Thank you.
Right.
Are there any more questions? All right, if there are no more questions, please join me in thanking Kwesi. And the final talk for our symposium will be delivered by Claire Hivros, who is the Director of Research at the INSERM and Team Leader Institute Curie. And the floor is yours.
So thanks a lot. It's a pleasure to be here. And I really want to thank the organizer for inviting me. It's my first time in Philadelphia. I enjoyed it a lot. In fact, I enjoyed the symposium because I didn't get the chance to visit yet. And it's difficult to give the last talk because so many great talks have been given. I hope that I can add something to what I have heard. And I was really impressed by the young people and bright scientists that presented during this symposium. It's nice when you're getting old to see all these young people. So I will start by thanking the people that did the work. So especially Stéphanie Dogno and Noémie Paillon, as well as Juan José Saez in my team, and in the past, Anna Savika, Michael Saitakis. And I really want to thank my main collaborators, two incredible biophysicists, Julien Husson and Atef Astasios, who are working in Paris. So I think, okay, just thank you. So I think that we heard before that immune cells are really patrolling the body, and it's important for the immune response. Here I will talk only about T lymphocytes. And so the T lymphocytes are always interacting with cells, so rolling on endothelial cells during their journey in the vessels, and then they attach in response to inflammation, they extravasate in the tissues, they migrate in these tissues and interact with cells and extracellular matrix. But the thing that I'm really interested in is the formation of this structure. So when the T lymphocyte encounter an antigen-presenting cells, these antigen-presenting cells being professional antigen-presenting cell, like the dendritic cells, or a target cells that will be killed, for example, for a cytotoxic T cells in the tissue to get rid of cells that are, for example, of cancer cells, for example. So I've been interested since 2000, in fact, when the immune synapse was found by this structure. Why? Because in fact, this structure will control the signal that T lymphocyte will receive, and we heard that before. So a classical immunologist will say that you need 3 signals to get activated for a T lymphocyte. The signal 1, which is given by the TCR by recognition of the peptide MHC. The signal 2, which is given by co-stimulation, co-stimulatory receptors such as CD28. And a third signal, which is given by instructive cytokines that are released by antigen-presenting cells, for example, in the lymph node, and that will modify the devenir, the shape and the function of the T lymphocytes. And looking at these immune synapses, especially in live, it was really it was striking that the formation of this immune synapse is dynamic, and that the cells move on the antigen-presenting cells. So the antigen-presenting cells can give some forces towards the T lymphocyte, but the T lymphocytes also exert forces on the antigen-presenting cells, and we heard about that. And of course, this is due to the remodeling, the striking remodeling of the cytoskeleton, here the actin in the contact zone, that is happening in T lymphocytes. So we wanted to measure these forces and try to understand what was the role of these forces. So I contacted Julien Husson in École Polytechnique, who set up a model to measure, a tool to measure the forces exerted by cells. So it's the micropipette force probe. So there's one pipette holding gently here a T lymphocyte. It will be human primary CD4 positive T lymphocyte, and a long pipette of non-rigidity holding a bead, which is covered with antibodies of interest or molecule of interest. And then you bring the bead close to the T lymphocyte, and you look what happened. And by looking at the displacement of this thin micropipette, and knowing the rigidity, the K rigidity of this pipette, you can measure the force exerted by the T cell on this micropipette. And that is what happened. Normally, we should see it was working. It was working before, so. But I was doing that. Anyway, you can't see it. So you have to believe me, and we will see on the next, perhaps on the next slide. Yes. So at the beginning, not much happens. So there's a lag phase. And so the time trace of the bead micropipette tip reveals is very characteristic. So you will see always this time trace with the cells from one donor, the T lymphocyte from one donor. And it's also found among donors, it will be always the same time trace. So this lag phase where nothing seems to happen, but yet we know that the T cell is increasing its stiffness. Probably the cytoskeleton is mobilized towards the development of forces after. Then you have this phase of pushing, and you can see this protrusion, which is pushing. Here you can see it by scanning electron microscopy. What you can notice is that normally the T lymphocyte has a lot of microvilli, and here it's completely bald. Because all the membrane has been used to form this protrusion. And during the growing of this protrusion, we can see an increased calcium. And then the protrusion retracts, and there's a lag phase again, and then a phase of pooling and the formation of this cap, phagocytic cap-like domain. Here, the T cell try to envelope the bead. So what we wanted to know is if it was this kind of time trace and the forces that were exerted could happen when we were putting a bead with antibodies against other things than the TCR. So what we used here is anti-CD45 antibodies, because the CD45 is highly expressed by T lymphocyte. And we looked at the time trace, and what you can see here is that the time trace is completely flat. There's no induction of this protrusion and pushing force, neither of the pulling force that we can see with anti-CD3 coated bead. So the mere attachment of the bead to the cell that you can see here, CD45 coated bead, with this really good attachment, does not induce forces on T lymphocyte. So then we wanted to know what was the mechanism underneath the development of these forces. And of course, the usual suspect was the remodeling of actin at the immunological synapse. And I took this image from a really nice review from Janice Burkhardt. And so what you can see is that in the distal area, there's branched actin, which is dependent of ARP2/3. Then you have in the lamella, in the periphery here, bundles of actin that are formin dependent and actomyosin. So here you will find the myosin 2A, and also in the middle you have these foci that are wasp dependent. So what we did is we inhibited, so to look at the role of the different networks in force development, we inhibited each of these networks. So we used ML7 to inhibit the phosphorylation of the myosin light chain, and we looked at the time trace and the forces. What you can see is that ML7 inhibits the pooling forces exerted by the T lymphocyte on anti-CD3 beads. So the pooling forces depends on the actomyosin. They depend also on formin, because if we inhibit formins with SMIF-H2, you can see also that the max pooling force are diminished from 230 to 170, from 0.23 nanonewton to 0.17 nanonewton. And now, if we inhibit the branched actin formation by inhibiting ARP2/3 with the CK666, what you can see is that you inhibit the maximum pushing forces from 0.33 to 0.14. And indeed, this ARP2/3 is very important for the pushing force. If now we look at with the different inhibitors at the maximum pushing forces, you can see that only the CK666 is inhibiting this. So now what is really good with this model from this tool developed by Julien is that you can change the rigidity of this long thin pipette which holds the bead. And so that's what we did to see if the forces were affected by that. And what you can see is that indeed, if you increase the rigidity of this pipette, then the pushing and pulling forces, pushing in orange and pulling forces, increase with the rigidity, and also the force rate are increasing with the rigidity of the pipette. So indeed, upon TCR activation, T lymphocytes exert pushing and pulling forces. Actin remodeling is important to control these force development, and the forces can adapt to the rigidity of the substrate. And of course, we wanted to know if the rigidity of the substrate can change T cell function. So it's hard to come after Landscam and also Quasi. But here is what we did. So of course, each model has its own bias, I guess. But what we use is polyacrylamide gels of measured rigidity containing polyacrylamide streptavidin that we coated with activating molecules, so biotinylated CD3, CD28 antibodies, as well as ICAM1 Fc chimeric molecules. I must say that the human primary T lymphocyte that we used are blasts, because in this condition, we were unable to see flattening of the T cell on the polyacrylamide gel with naive T cells or non-activated T cells. So these cells have been activated for 6 days with anti-CD3 and anti-CD28 plus IL-2. And then we put these blasts on the substrate of different rigidity. We looked at the activation of cells in different conditions, purified the mRNA, did an Affymetrix analysis, and this was done with 4 different donors. And so what you can see on this presentation of the principal component analysis is that the rigidity here, we use 500 Pascal, 6.4 kilopascal, or 100 kilopascal. If we only put anti-CD28 plus ICAM1, we don't see much difference in the induction of gene expression. But as soon as we put anti-CD3 on this substrate of different rigidity, now we see differences in transcription in the T cells exposed to this different rigidity of the substrate. So the response requires TCR stimulation. This mechanical response requires TCR stimulation. And here what we looked at, what I'm showing is that so activation markers such as CD40 ligand, CD69 and others are activated upon an increase upon substrate stiffness. And this is the same for cytokines, for also for transcription factors that are important in T cell activation, such as FOXP3 and MYC. And also what is striking here is that the laminase gene is also increased upon stiffness, which is the case for many cells. So T cells is not an exception. So the stiffness of substrate modulates TCR-induced T cell activation. And what we notice is that a lot of genes involved in metabolism were induced by increased substrate when activated by anti-CD3. And here I'm showing you the pentofructokinase and the pyruvate kinase. And when we looked at all the genes, and to cut a long story short, but for example, if we look at the genes encoding enzymes or receptors that are important for the aerobic glycolysis, what we can see is that upon activation, all these genes are, upon activation, the stiffness increases the expression of all these genes. So the mechanical environment can rewire metabolism in T cells. What has been shown also by several groups, but in particular by the Morgan Hughes group, is that these forces are very important for cytotoxicity because they exert forces on the target cells. And what has been proposed by Morgan is that this will help inserting the perforin inside the target cells. And more recently, what has been shown also is that the T-cell cytoskeletal force shape the curvature of both the T-cell membrane and the target cell membrane, and that this is important for the unidirectionality of killing. So the T-cell does not kill itself, but kills the target. And remember these topological things also for the rest. So what we propose, and I think that many of you are proposing also, is that the signal 1, which is antigen specific, is gaining also some instructive information from the mechanical environment. So finally, there's a signal 2, which is sensing the mechanic of the antigen presenting cells. And then, of course, you have the co-stimulation and the cytokine that change the T lymphocyte function and the veneer. And of course, because we are immunologists, we wanted to know if important receptors that are the checkpoint inhibitors could modulate the fourth generation in the synapse. And we got specifically interested by one immune checkpoint inhibitor, PD-1, that I guess that all the immunologists here know. Why? Because PD-1 has been a target in immunotherapy, both in autoimmunity and in cancer therapy, because these PD-1 molecules inhibits T-cell activation. It has been shown to inhibit signaling by TCR and CD28, production of cytokines, cytotoxic activity of CD8 positive T cells, but it has been shown also to inhibit the metabolic switch from oxphosphorylation to aerobic glycolysis. So we wanted to know if PD-1 could modify somehow first the immune synapse and also the force generated. And indeed, it is really interesting because although antibodies against PD-1 and PD-L1 have been used in the clinic, the mechanisms that are involved in this inhibition are not well known. So we embarked to look at that. And we did that by using two models. First, a model of true synapse with engineered antigen presenting cells that can be equipped with anti-CD3 with antibodies, and we use anti-CD3 and anti-CD28 to quote somehow the engineered antigen presenting cells. And these antigen presenting cells express or not PD-L1. And then for T cells, we use either primary CD4 or CD8 positive T cells that have been activated for 6 days in the presence of interferon alpha, because this way they do express PD-1, otherwise at the resting state they don't express PD-1. Or we use germ cells that have been transduced with an antiviral vector encoding PD-1. And the second model that we use is a coated glass slide with anti-CD3 plus anti-CD28 plus IgG1 as a control, or PD-L1 chimeric molecules to bind, so to engage the PD-1, which is at the surface of the T cell. So here I'm presenting the results on the conjugate formation, let's say true synapse. This antigen presenting cell is not presenting, neither present activating antibodies or PD-L1. What you can see is that you still find some conjugate, because these antigen presenting cells express ligand for CD2 and for LFA1. PD-1 is all around the cortex, and there's no concentration in the contact zone. Now, if the antigen presenting cell is expressing PD-L1, what you can see is that the conjugate forms, but there's no remodeling of the actin in this contact zone. Again, there's no activation, and PD-1 is really strongly concentrated in the contact zone. This is the classical immune synapse that you all know. So in the presence of activating antibodies, you can see the formation of this shape with recruitment of filamentous actin in the zone, with this central zone, which is hypodense, and this is important for the release, for example, of cytotoxic granules. And what you can see is that more or less the PD-1 is following, in fact, the cortex of the cell. Now, if you have PD-L1 on the surface of the antigen-presenting cells, it's completely different. There's no remodeling of the actin in the zone. And you have so this PD-1 recruitment at the synapse, and there's no deformation, this phagocytic cap-like deformation of the T cell in the contact. So we measured, thank you. We measured the concentration of PD-1. So this is a mean PD-1 profile on a lot of cells. And what you can see is that in the absence of PD-L1, so PD-1 is all around. It's present here because there's a much more membrane. And in the presence of PD-L1, it's recruited, and we measured that, the mean PD-1 density in the zone. We then looked at the F-actin enrichment at the synapse, and you can see that as soon as you engage PD-L1, you have less actin recruited in the contact zone. And this is true, in fact, when the cell is activated, but there's also a little bit less recruitment in the cells that are not activated. And as a proxy of measuring this change in morphology, we looked at the solidity index. And what you can see is that you lose the T cell deformation when PD-L1 is present and on activated T cells, which is clear here. So you don't have this two patch and this phagocytic-like cap formation. What was interesting, you won't see it. Ah, yes, you can see it. Is that the PD-1 inhibition effect on the formation of the deformations, the morphology and so on, is really local, because here you have one T cell interacting with two antigen-presenting cells, one which express PD-L1. You can see the nice recruitment of PD-1 here, and the other antigen-presenting cells does not express PD-L1. And here the T cell can really form a nice synapse. So in fact, the inhibition is really local and dependent of the presence of PD-L1 on the antigen-presenting cell. So now we use the other model, so the spreading assay. And what you can see here on CD4 positive and CD8 positive primary T cells activated so that they can express PD-1. So what you can see is that there's a decrease of spreading in the spreading area. There's also a decrease of mean F-actin intensity in both the CD4 positive and the CD8 positive T cells when PD-1 is engaged. And we looked more carefully by structure, by 3D SIM microscopy at the contact. What you can see here again is like a bit like in the schematic representation that I showed you from the paper of Adu-Berchie, that you have this branched actin distal formation of this branched actin in cells where PD-1 was not engaged. And you can appreciate at the immune synapse plane that you don't have this remodeling, this clear remodeling of actin in this zone. So this suggests that branched actin is the target of PD-1. And in fact, we looked also at the recruitment of WASP in the conjugate. For sake of time, I don't present that, but we didn't see any recruitment of WASP. So now we wanted to look at the force finally. And what we did is we looked at the phosphorylation of K-cell. Why? Because K-cell interact with the actin cytoskeleton. It's phosphorylated in response to actin tension. And the phosphorylation of Cas-L has been shown by this in this nice paper from Sudha Kumari. So the phosphorylation depends on branched actin. It is inhibited by CK666. And because we knew that PD-1 was affecting the actin remodeling, and especially the branched actin, we looked at the phosphorylation of Cas-L. And what you can see here, looking at the cluster density, is that PD-L1 engagement is inhibiting the phosphorylation of Cas-L and the number of clusters of phosphorylated Cas-L. And this suggests that PD-1 engagement inhibits the fourth generation by T cells. And I'm finished. So finally, what we have seen is that T cells develop forces. These forces mechanically enhance the killing, as shown by the nice work of Morgan Hughes and others. It also affects the T cell activation, and as we have shown, the metabolism switch. So when PD-1 is engaged, this affects actin remodeling. It affects also the cell morphology. You don't see this hat shape of the T cell. The cell stays round, and it affects the fourth generation. For sake of time, I didn't show you that, in fact, these effects are not dependent of the signaling tyrosine of PD-1. So there are two tyrosine in the intracellular domain of PD-1, which bind SHP2, and that have been shown to control T cell activation, at least some effects of T cell activation. But in this case, the effect on actin remodeling does not depend on these two tyrosine. And so now what we would like to know is which are the properties of PD-1 that are involved in inhibition of actin remodeling, especially the size, the affinity of PD-1 for PD-L1, the molecular flexibility of PD-1, its density. We would like to look at if these inhibitory effects are shared by other immune checkpoint inhibitors, and how actin remodeling is affected, and which are the T cell function affected by the signaling independent PD-1 effects. And we have some preliminary data showing that PD-1 can really inhibit the rapid metabolic switch of T cells in a signaling independent manner. And with that, I want to thank you again, thank the organizer and all of you, and the people that have done the experiment, and yes, of course, the funding. Thank you.
Thank you very much. Any questions?
Oh, hi, great talk. I wanted to ask if the suction force coming from the vacuum generated by the micropipette affects the activation.
So it's difficult to really tackle that, but as far as we have seen, it does not, because we change a bit the pressure on in this pipette to aspire more or less the T cells, and we didn't see any effect.
Thank you.
That, of course, each model has its own bias, so I cannot.
Yeah, a wonderful talk. I was specifically fascinated by the observation that the PD-1 interaction is local. That if you have APCs with or without PD-1, that you see very different interactions. I'm curious to know what the net effect ends up being. So in terms of the net effects, today, I guess you are looking at independent PD-1 signaling, but I'm curious to know, would the cells end up still shutting down TCR interaction overall if they have if they're interacting with two APCs? And then also for metabolism as well, do you still see that switch? If you have
So this is a difficult question. But what I can say is that I don't know if some of you have looked at the inhibitory effect of PD-1. They do exist in vitro. It's not a black and white effect. And my interpretation of the fact that it's not a black and white effect is that the T cells will be activated by APC that are in this test. Because I think that some people thought that they were freezed by the interaction of PD-1 with PD-L1 because the signaling was extinguished somehow so that they could not be activated by another cells. But I think it's not true because they can still form a normal immune synapse. And it is, yeah, we should look more, you know, it's serendipity. I mean, it was because we were looking at all these cells that we see these double synapse effect. And I've been trying to tell the PhD student working with me to look more carefully at that with no success for the moment.
Hi, I love the talk. Thank you. So you shared some really nice data about how inhibiting different parts of the actin cytoskeleton decreases the force, both pushing and pulling, that T cells exert. Do you have any insight into whether that decrease in force translates to anything downstream in terms of signaling and activation? Does that just mean there's no signaling or activation, or is it a diminished but still present level of activation? Do you have any thoughts about that?
So we didn't do that, but some people did. And indeed, I think that people looked at the phosphorylation of ZAP70 and that this phosphorylation was increased with stiffness. I can't remember the name of the person who did, or the group who did that. I'm sorry if he's here. We didn't do that, although we are working on signaling, so I should.
Thank you.
There is one more question there.
Hi, I really enjoy your talk. And I wonder if you have looked at the target stiffness. For example, cells that are stiffer, is it easier to form the synapse or easier to kill? Because in a tumor microenvironment, when you have a stiff matrix, usually cells become stiffer because they have a better cytoskeleton. And I wonder if the T cells can better anchor onto like a stiffer cell body when the cells are stiffer, and then perhaps improve the chances of forming immune synapse, or when they're softer, it's easier to pull so that it's easier to kill for the cell.
So we didn't do that. Again, some people, so Morgan Hughes looked at the killing of the cells. And if I remember correctly, the cells, I'm sure, in fact, the cells, the stiffer, the stiff cells will be more killed than soft cells. What we did is we looked, and I didn't present that for sake of time, with Atef Asnasios, we looked at the rigidity of different antigen presenting cells that have been activated by inflammatory cytokines such as interferon gamma or TNF. And indeed, this increased the stiffness of the antigen presenting cells. The dendritic cells, for example, they are around 500 Pascal when they are at steady state, non-immature, and they go to 1,500 Pascal in response to interferon gamma or TNF. So, of course, the stiffness increased, but a lot of other things also are changed. So we couldn't attribute the better activation by different mature dendritic cells to the mechanical properties of these cells.
Okay, thank you.
All right, any more questions? Okay, so if there are no questions, please join me in thanking Dr. Hivros and also the other speakers of the symposium. It was a wonderful experience. Being a plant biologist, I learned a lot and got new research ideas. So be ready. I'll be shooting emails to the speakers for collaboration. And now I would like to call upon Dr. Vivek Shenoy, the director of CEMB, for closing remarks.
Yeah, so it's a really great symposium. So when we sort of thought about immunology and epigenetics, we didn't really know how it would come together, but it seems it came together really, really well. So a lot of the immunology talks also had a lot of gene expression data, RNA-seq, and so going forward, how mechanics sort of couples to that would be a great thing to look into. So I want to thank all the speakers, very great talks, the students and postdocs for their for presenting, and for you all for sticking around till the very end. So see you next year. We'll have a new topic. Okay, thanks.